that you communicate, right? And the second, when there's time, and I want to do this as an interactive exercise, thinking of your own personal brand as a leader. And
Sennheiser test 1 2. Sennheiser mic test 1 2 2 2 2. Sennheiser mic test 1 2. Test, test, test. Hello, 1, 2. Test, test, test. 1, 2, 2, 2, 2, 2. Handheld 1, 2, 2. Test. Test, test, 1, 2. Test 1.
All right, good morning, everyone. We're going to get started in just a moment, so I'd like to ask everyone to take their seats, get comfortable. Boy, that got quiet quickly. I can tell this group is enthusiastic. All right, I'd like to welcome you all to this event on scientific and regulatory considerations for the analytical validation of assays used in the qualification of biomarkers in biological matrices. I'm Mark McClellan. We're very pleased to be supporting this event under a cooperative agreement with the FDA and the Duke-Margolis Center for Health Policy, and in very close collaboration with the Critical Path Institute, a group that we've partnered with in the past to deal with important technical issues relevant to drug development and other aspects of medical product development that also have some important policy implications. And that's certainly the case here. So we're very pleased that you're able to join us for what we hope will be 2 days of productive and important back and forth on advancing this topic of biomarker qualification. We appreciate as well everybody who is joining us online for this event, as well as those who are participating here in the room. As many of you know, the meeting is in the development of the white paper framework that accompanied it has been over a year in the making. It's involved a lot of collaboration from our partners at Critical Path and FDA, and also many other contributors to the effort. I want to particularly thank the strong leadership of the Critical Path Institute, without whom this white paper would not have been possible. Before we kick off the discussion today, I want to provide just a little bit of background. As you all know, there is a lot of interest in biomarkers as a means of advancing the process of drug development, biomarkers as important drug development tools. This has been highlighted as recently as the 21st Century Cures legislation. It's been under discussion in FDA's user fee negotiations that are and that process is coming to conclusion now because this is a fundamentally important issue in getting to faster, more reliable development of safe and effective medical treatments. And the characterization and analytical validation of biomarker assay performance is a fundamentally important aspect of biomarker qualification. There are a number of complex analytical factors that need to be taken into account when assessing the robustness and the reliability of a biomarker assay. Biases introduced in the conduct and interpretation of the assay results will affect the biomarker's predictive accuracy and thus its evaluation as a useful drug development tool. And the application of current bioanalytic validation methods for biomarkers has proven to be challenging, particularly because the guidelines developed for PK assays and in vitro diagnostics that are used in clinical practice are not universally applicable to biomarker assays. It's an issue of fitness for use. Analytical validation of biomarker assays used during biomarker qualification must fit the proposed context for which they are ultimately going to be used, the proposed context for use. Although the development and interpretation of the FDA biomarker qualification pathway has made some important and continuing progress, clarity on the scientific and regulatory expectations for biomarker assay performance characteristics and validation is needed to guide biomarker qualification studies. Further, because there is no one set approach that can be applied to all biomarker assays, this framework needs to embrace scientific flexibility while establishing sufficient guidance for experts that they can then apply the unique guidance to the unique attributes of a biomarker being qualified under a specific context of use. So for all of these reasons, the Biomarker Assay Collaborative Evidentiary Considerations Writing Group, under the leadership of the Critical Path Institute, has developed a draft framework. This was circulated to all of you last week, outlining key criteria and minimal expectations for the validation of fluid-based biomarker assays used in biomarker qualifications, maybe with some implications for other types of assays that could be used in biomarker qualification as well. I know many of you in the room today have played a key role in developing this framework, and we very much look forward to continuing that conversation and building on the work that's been developed to date. Our main objectives for both today's discussion as well as tomorrow's are to, first of all, review the components of the draft framework, then explore how the framework can be applied in real-world contexts through the use of some case studies, and then gain broad stakeholder feedback to identify any remaining gaps or areas of disagreement around the framework or other ways to improve it, and then to discuss strategies to ensure the dissemination and uptake of the framework across the scientific community, back to that practical impact on drug development. As you can see from our agenda, we do have a lot of ground to cover. I'm going to turn this introduction over now to Greg Daniel, our Deputy Director here at the Center, who's going to take a few minutes to walk through the agenda for today, as well as go over a few housekeeping announcements. So Greg, thank you very much.
Thanks, Mark. Good morning, everybody. Allow me to echo Mark's thanks to all of you for joining us today. As you'll note in the agenda, each of our sessions will be kicked off with either an opening presentation and or a series of panel remarks, which are intended to frame up the ensuing conversation. Although substantial time has been set for all of our sessions to allow for broad stakeholder Q&A input from all of you on the discussion for that session. We'll begin by opening remarks from FDA, which will provide further background and help set the context for the day's discussion. We'll also have a brief presentation on some of the key terminology that is used in the white paper that you all received. We'll then go into sessions 1A and 1B, which will cover various sections of the actual draft framework itself. Each of those sessions will include an initial presentation followed by moderated Q&A with the panel and with all of you in the audience. After those discussions, we'll take a break for lunch. Following lunch, session 2 will begin at 1:45, and that session will focus on our first case study, which is the including biomarkers for drug-induced nephrotoxicity, which will illustrate how the framework might be applied in the real-world context. So, after that, session 3 will be our final session for today, and that will provide an opportunity to reflect back on the day's discussion and facilitate discussion of the framework in a broader context. I want to stress that there will be plenty of time reserved, as I mentioned, for Q&A throughout the day, and we look forward to all of your thoughtful comments. Before we get kicked off, just a few more housekeeping items. I want to remind everybody that this is a public meeting. The event is being live broadcast online, so everything you say will be part of the record. Just so that you know, materials and the video itself would be available after this workshop online on our website. As note to speakers and panelists, you'll know we have many speakers, many panelists in sort of a short amount of time. So we will be on you in terms of the number of minutes, and we have staff. Staff will be somewhere. They'll figure it out, but you'll see them because they'll be waving big pieces of paper with big numbers on it, so you know exactly how much time is left. For those of you in the room, we do have mics in the aisles for any of the comments or questions that you have throughout the sessions. For those of you who are joining us via the webcast, you can ask your question in two ways. First, you can ask, one way is to ask question through the comments or live chat section on YouTube. Where we have staff monitoring that, and so we'll be able to get your questions right away. If not, you can also send questions to the Margolis Events email address, which is margolisevents@duke.edu. We're also monitoring that, so we'll make sure to incorporate your questions via that route as well. We'll do our best to do all of that. We will be live tweeting as well, and you can join us with our hashtag biomarker assay validation, okay? So that is, that's what I thought too. It's a lot of characters, and so you might be able to get a few more words into that tweet after putting that hashtag in. So as you'll note, a few other things, we've built in breaks throughout the day. We do have coffee, tea, water to my left over here for your convenience. Lunch will be on your own, although there are a number of restaurants and food trucks in the area, so we have a list of those. And if you have any questions throughout the day, please just find one of the Duke staff members to help with that. We'll now hear from opening comments from Dr. Shavre Buckman-Garner, who is the Director of the Office of Translational Sciences and CDER at the US FDA, and she'll provide further context and an overview of FDA's goals as well as the goals for this two-day meeting. And with that, I'll turn things over to Shavre.
Thank you so much. Good morning, everyone. Very nice to see you. I try and talk quickly, and I'm going to see if I can move my slides. I'm Shavre Buckman-Garner. I'm the Director of the Office of Translational Sciences in CDER, and I'm also the co-director for the Biomarker Qualification Program. And I'm going to talk to you a little bit about where we are, where we've been, and where we hope to be going, and hopefully infuse a tremendous amount of excitement around the opportunities that we have in front of us. So before we get started, I really want to offer a huge thank you to the Duke Margolis Group as well as the CPATH Institute for tremendous efforts in helping us with over a year of planning to try and get us to this day. We even had a pre-meeting, I think, back in November. This has been a tremendous amount of effort, and I see a lot of familiar faces that were at that pre-meet. I also want to thank Steve Piccoli and all the contributing authors for this writing group, for this tremendous white paper. This was an enormous amount of work. And it's not very often that you go to a workshop like this where you have some of this material right in front of you to react to. Oftentimes you have to come out of the workshop and then people have to develop things. So we have tremendous starting material, and I'm hoping that will help us broker a wonderful conversation today. So why are we here? We're here to help review and define a draft set of best practices and performance characteristics for biomarker assay validation for the purpose of qualification. We want to be able to use case studies to explore methodologic implications of the framework that's been created and identify any remaining gaps. And really what I want to be able to help do is help us define a path forward. So I'll be coming back at the last session because those who know me know that I'm a very concrete person, and I want to make sure we have a path forward and we have things that we can start to implement and do to move this forward. So that's where we are. So this is a rather busy slide, but it really goes to show you that we've been working for well over a decade now in this biomarker development and qualification space. This launched back in 2006 from the FDA perspective with the Critical Path Opportunities List and report, where we started to articulate the need for biomarker development and qualification to help streamline drug development, to help unclog that path towards getting new products to market, and help us increase our confidence and reliability in the types of medical products that are being developed. So since that time, we've invested in a variety of efforts, including developing guidances and manuals of policies and procedures, collaborating with our international colleagues, launching our critical path innovation meetings, which allow us an opportunity to just have a scientific discussion with others around potential areas for biomarker development. We've launched a letter of support program where we've been able to identify promising biomarkers for qualification. We've qualified multiple packages of biomarkers for both preclinical and clinical context of use. And we have also launched a number of different workshops, surveys both internally and externally, surveys with pharma, trying to understand the landscape and where opportunities for development are, right? So there's been a tremendous amount of effort, but as Dr. McClellan spoke to, one of the big looming issues has to do with what's the bar? What are the evidentiary criteria? How much is enough for qualification, and what does that entail? And that's the area that we really need to continue to dig into. So what have we learned? This concept that the specific context of use for a biomarker has to drive the extent of evidence needed for qualification. That is a critical concept that drives everything that we're doing in this space. And we've learned that this analytical validation piece has to come very early in the qualification process. It needs to precede the clinical validation piece. We need to be able to have confidence in the performance characteristics, acceptance criteria, efforts around reference ranges, decision points, pre-analytical and assay performance characteristics, rigor, reproducibility, sample handling, stability. All of these things we know are part and parcel to that analytical validation context we need. And then the other piece that we need that we're not discussing today is the clinical validation piece. And that has to do with how we establish that a biomarker acceptably identifies, measures, and supports a context of use. And that has to do with study design acceptability, clinical meaningfulness and decision points, and benefit risk assessments. So we see that both of these components are critical for us to be able to make decisions. So as you all know, and Dr. McClellan spoke to, we have 21st century cures. As of December 2016, we now have new legislation that helps us refine and shine a bright light on the biomarker qualification activities. And with that, we have now 3 main time points that are focused on how we're looking at qualification. So the letter of intent that we receive, the qualification plan, and the full qualification package, right? And during that time, the goal is antecedent to the letter of intent. We're anticipating that there's a lot of biomarker discovery and development that goes on, that we're already getting a good foothold in terms of the analytical validation issues that might come to pass, and that by the time we get the qualification plan, we have a pretty good handle on the analytical validation issues, right? And so the goal is really hoping to streamline this process, hoping to make sure that the consultation and advice phases that we've had in the past don't become interminable, right? And that we're able to move forward with clear packages that we're able to make clear decisions on and be able to move forward. So this goal of having clarity around what is the bar and what are those evidentiary considerations when we look at analytical validation becomes critical in this process. So with that, now that we do have this new Section 507, we're in the process, as I've talked to Martha, we're kind of flying the plane and changing out the parts at the same time. So you'll see when you look at our websites, you will see that there's increasing information that's going to be going up to try and clarify what are these time points in the process, what do we need, and then there's a whole push towards transparency. What are we sharing in the public domain in terms of what have we received at the letter of intent phase, what have we received at the qualification plan phase, full reviews when we issue a qualification determination, so that the rest of the world understands what our thinking is and that we're best able to communicate where these development efforts lie. So why is the timing of this meeting so critical? Well, if you look at the 21st Century Cures Act, and I read it again last night, there really is a focus on helping to develop, through a transparent public process, standards and scientific approaches to data collection accepted by the medical and clinical research community for the purposes of qualifying drug development tools. And that's where we are, right? Is really how do we develop that rigor and reproducibility and understanding of what these assays mean. So I spoke with my colleagues, Martha and John Michael and our Duke Margolis colleagues, and I emailed them and I said, what do you think would constitute a successful workshop? Because when I kick things off, I want to think about what's the end in mind? Where are we headed? What's going to make this successful? And so Martha, who I quoted here, said, we understand that we as a research community own this. The more of the concepts for necessary analytical validation that can be incorporated in the earliest experiments, trials, et cetera, then the more expeditiously we can achieve qualification. It's not only the companies who own this or those who undertake biomarker qualification. It's the entire community, meaning each of us has a contribution. And that's this call to action today, is how we step forward and collaboratively figure out how to move this effort. we begin to articulate core expectations for the validation of a fluid biomarker assay used in qualification. We understand how benefit risk and context of use defines additional assay performance characteristics and requirements and expectations, and that we develop a greater understanding of a potential framework that may be applied while considering the unique attributes of a biomarker being qualified under a specific context of use, and understanding that there needs to be scientific flexibility, right? That there's not a hard, fast, if you don't fit in this small box, this is not acceptable. We have to understand that there's flexibility, but we need a framework to be able to work from that we all can align with. So with that, I want to point out to you, because none of us have name tags on, who's in the room with you from FDA, and I may not name everyone. Shashi Amar, who's our scientific coordinator, she's there in the back. Stand up for a second so we know who she is. Aloka Chakravarti, who may not be with us, who is with our Office of Biostatistics. Ruchen, who's one of our staff fellows. Kylie Haskins, who's our lead for the Letter of Support program. There she is in the back. Stand up, Kylie, so people at least know, because no name tags, so you don't know who these folks are. Lori Muldowney, who's our Associate Director for Medical Policy in the Office of Translational Sciences. Mary Ann Noon, who's our Project Manager for the Biomarker Qualification Program. Mike Pakinowski, who's our Associate Director for Pharmacogenomics in the Office of Translational Sciences. Amita Parikh, who's our Scientific Coordinator, who is also our liaison to the Predictive Safety Testing Consortium. Sharmistha Sanyal, raise your hand, stand up, who's one of our important individuals who's part of our biomarker qualification team. Sujane Wong, Sujane, who's not raising her hand, who is one of our important leads in the Office of Biostatistics for the biomarker qualification activities. In Office of New Drugs, Elizabeth Hausner, who's, I don't know if she's here with us. She is not, who's with our Pharm Tox group. Chris Leptek, who you'll hear from next, who's our co-director for the Biomarker Qualification Program. Aliza Thompson. I saw Aliza just a minute ago right here, who's been a tremendous lead with our cardiorenal division with a lot of our activities in biomarker qualification. Of course, Dr. Woodcock, who couldn't be here with us, but it has been a tremendous push in terms of how we move forward in this space. Bob Becker from CDRH. I see Bob is here. Dan Kraniak may not be here with us. Dan is here. Fantastic. Vasum Paris, who I may be mistaking his name, but he was also part of the writing group. Susie McCune, who's been part of the Office of Pediatric Therapeutics and before was helping us lead the qualification efforts with OTS. So I want you to see the number of people from FDA that are vested in trying to move this forward. I don't like to waste my time. I know you all don't either, and we really want to be able to make progress and make sure that we have some traction to move forward from this meeting. So I appreciate your time and attention, and I think Chris Leptek is up next, and he will take a much deeper dive than me. So thank you.
Excellent. So I will also lend my welcome to everyone for being here today and follow up on some of the comments that we've had thus far. If whoever's going to tee up the slides, if you could do so. Okay. There we go. Certainly there's a lot of familiar faces in the audience from the various engagements that we've had over the last year and a half. I also see some new folks. So one of the things that we want to start off with is just making sure that we all understand that we're talking the same language, that we can take some of the broad concepts that Chavre put forward and then speak about how the analytics fit into that bigger picture around qualification. So ultimately, if you think about precision medicine, there's a lot of public attention to that, and there has been some time. Ultimately, I call it the parade of the rights, the right therapeutic for the right patient, at the right dose, for the right duration, at the right time of engagement, and for the right outcome. All of those rights ultimately can be determined by biomarkers. So it certainly has a pivotal role as part of translating the scientific understanding of disease as we now are coming to understand it and how we can make better choices for patients, which is the ultimate outcome of all the communities. When you look at how biomarkers integrated as part of a drug development program, whether or not it's 4 circles or 6, it can be debated, but ultimately you can see that there's distinct components of how biomarkers are integrated as part of a clinical trial design. So certainly the finding the right biomarker and understanding that we're all speaking of the same entity in the same way is exceedingly important. So that's a key component of the process moving forward. When you think about the analytics, are we talking about biomarkers in the same way? What is the algorithm for the determination of the biomarker? And ultimately, what is the decision that's made based on that biomarker information? So that's kind of one key component. The second is around the assay. You can have a great biomarker, and if you're not reliably measuring it, you could throw out a good biomarker for not good reason or vice versa. So the assay is critical and part of this process. And certainly, as Chavray pointed up forward, we've certainly have come to an understanding as we've had the biomarker qualification in place for a while, that the analytics and understanding those early and often are really critical to know whether or not you're really investing your efforts in the right way. Because we all have limited resources, so we'll make sure that we're devoting them to things that make sense and that can ultimately be successful. The right patient population. Certainly, as we're getting a better understanding of the molecular pathogenesis of disease, what used to be descriptive in a constellation of signs and symptoms, we're now describing at a molecular level. So as patients get subsetted based on biomarker status into smaller groups, you want to make sure that you're rolling the right patient based on your target of a mechanism of action for your therapeutic. And then finally, the clinical trial design elements. Certainly, biomarkers are used as enrichment strategies, prediction of patient response that can be used for endpoints. So certainly, the biomarkers all come into play there. Each of these characteristics, if you will, of a biomarker-based drug development program are important. And certainly, when there's success, that sort of small integration, great. You know you did something right, that at least you had an outcome that you have faith in, that this could be beneficial to patients based on how the study was designed. It's when things don't go well, it's trying to figure out which of these 4 components was the one that was problematic. Was it just that you didn't have the right biomarker, or that your assay wasn't good, or you hadn't enrolled the right patient population, or maybe you just didn't have the right endpoint. So I think what the analytics really drive home as the central character of the importance that piece has to this overall success. From a regulatory perspective, certainly we're defining biomarkers as a defined characteristic of a normal or an abnormal process or response to a therapeutic, kind of the abridged version. Certainly we define it broadly in that it's not just something that you measure in the blood, which is kind of I think how many of the lay public view it, that we interpret it much more broadly. So we're talking about imaging biomarkers, different types of algorithms that can use to assess and come to a determination. For today's purposes, for the analytics, we're talking about the fluid biomarker class. We're not discussing analytics for the imaging, and that would likely be a subsequent workshop. The key thing to say here is that the biomarkers are not how a patient feels, functions, or survives. This is not the standard for drug approval. Now, some biomarkers may reach that, and there are the small subset known as surrogates. But in general, if you take that class aside, that's not what a biomarker is intended to do as part of an FDA assessment of a drug in a clinical trial. And then ultimately, there's many communities that use biomarkers. There's the regulatory, there's the scientific, there's the clinical communities. And one of the things we get asked at the FDA is, if you get acceptance in one community, why can't all communities universally accept it? And ultimately, it comes down to what was the biomarker developed for, what data was brought forward, and what is the potential risks and benefit based on that information. That certainly within the scientific community, the goal is usually publication and sharing with your peers your ideas around the science. The downside, if you're not right, is that maybe your peers can't reproduce your data. That's problematic, but that doesn't mean a person's going to get harmed. And it's also very different for the clinical person who's sitting across with a patient, and the two of them can negotiate based on what's available, what might be tried to see if that could help that patient for that particular problem. When we approve products, we're approving it for populations. So it's a very different scenario. So just to make that distinction. Ultimately, there was an interagency proposal that was put forward, I guess, almost 2 years ago between the FDA and NIH. It was led by our previous commissioner, Dr. Califf. And ultimately, a number of the folks in the audience got together and hashed out definitions around the biomarker space so that ultimately we can be using the terms with the same intention. We certainly found that over time, you could have meetings and after an hour of discussion realize that how people were using predictive or prognostic was at opposite ends of the spectrum. And so you come out of the meeting, think you understood something, where the people at the table came away with very different outcomes because they had different interpretations. So whether or not you like the best definitions, you think they're the best definitions, that could all be debated, and it's certainly a living document, and there's a number of updates that are going up, and I leave the website there for people to provide comment. Ultimately, in addition to defining a biomarker and distinguishing it from clinical outcome assessments and endpoints, it also discusses subclasses of biomarkers. Again, whether 7 is the right number, you could argue. We basically tried to come up with distinct classes that had meaning for the various stakeholder communities, whether that be regulatory, scientific, or pharma. And so these are the 7 classes. If you put them into a sort of Robbins pathogenesis of disease, I think pictorially it helps you figure out what is the meaning that we're trying to go for. So ultimately, when you have normal physiology, we all know that there's normal variation between people and within a given person, even over the course of the day or over the course of their lifetime. So having biomarkers to assess that normal variability and know what its ranges are can be beneficial, especially if that biomarker is going to ultimately look for changes over time to see, well, what level of change may be actually useful or not. Ultimately, the normal physiology changes. It results in some type of a biologic change in the body that ultimately can become physiologic and result in what we would classify as a disease. That classification, at least historically, has been more descriptive. It's now becoming more molecular. And so then we get into a whole different set of classes of biomarkers where you're basically trying to use biomarkers to diagnose the condition or define the disease or a subset of patients with that disease, monitor the progress of that disease over time. All this can be without a therapy. And then ultimately, what is the prognosis of that individual? What's the likelihood that they would develop a future event of clinical interest, whether it be transplant, hospitalization, failure? And then once you get a therapy that's in play, then you're looking at a different subset of biomarkers. Here, we're looking for a change in physiologic function. That may just be at the molecular level, may have no bearing on terms of clinical benefit initially. And these are the pharmacodynamic response biomarker class, also for safety and prediction of response, whether it be advantageous or deleterious. Ultimately, if you can show that there's no progression of the symptoms or there's a reversal, then we're starting to get into that clinical benefit realm, and then we could be thinking about a response or an endpoint. And then ultimately, if that response or endpoint biomarker is truly predictive of a clinical benefit, then we're into the smaller subset of the surrogates. Ultimately, when it comes to how biomarker information comes into the agency, we have different pathways. Again, whether it's 3 or however you want to define it, I think it's more important to just know that there's different opportunities that biomarker information comes in to help us with regulatory decisions. Certainly, there's the drug-specific process under IND that's been long-standing. There's scientific community consensus in that we have examples where professional groups have gotten together and looked very critically at a problem, especially in the oncology community, and over a period of time, they come up with an understanding that they then can support and have value, bring it to us as part of an IND, and then we can look at it from a regulatory perspective. And then finally, the biomarker qualification program, certainly been around for a decade, but it's still the new kid on the block when you look at in respect to the other 2. So certainly we're talking today about analytical validation for biomarker qualification, but as Chevray pointed out, I think that the lessons learned for that particular space can have benefit to the other 2 as well. These are just more for reference that we have the websites that Chevray had put up and various documents that are linked there. Ultimately, initially we were looking, when the biomarker qualification came into being, mostly non-clinical, mostly safety, but over time we're now seeing a nice dispersion, and that there's almost equal interest in safety and efficacy, and non-clinical and clinical, as well as some of these other classes coming forward. So we're really getting a nice representation of all those best biomarker classes in our qualification work streams. This is just a list of the qualified biomarkers to date. As Chevray had pointed out, that we've had successes in non-clinical and clinical. And just to be clear that these are qualification efforts, not individual biomarkers. So each of these, many of these also have multiple biomarkers that were done as part of that qualification effort. So not to have that be misinterpreted. Ultimately, Chevray pointed out the timeline that we've been doing in the qualification space and all of the activities that have occurred over the last 10 years. I think it's important for us to take a step back and think about how that fits into what have we been doing in drug development? Drug development has been going on for decades. And certainly there's a lot of understandings that have accrued over that period of time. So certainly biomarker regulation and thinking is relatively new. And I think we need to kind of keep that in perspective of how we can get to the understanding necessary that yes, time is important, but we've got to keep it in perspective, that certainly we didn't learn as quickly in the drug space as what we maybe would have liked. Ultimately, it comes down to context of use. So that is ultimately, in a very simplistic way, what is the best biomarker class and what is the clinical trial question that the biomarker is intended to address. These are some examples of how that can be included, whether it be from inclusion-exclusion criteria to endpoints to safety monitoring to efficacy and response. And then at the bottom is an example of a context of use from a recent qualification for the total kidney volume. So you certainly can get a sense that there's a lot of descriptors so that once a biomarker is qualified for a given context of use, that any group then that wants to use that and incorporate it in their drug program, it tells them at least enough detail that they know how to do that. So the need for stakeholder engagement, certainly this is one important meeting. There's been multiples that have preceded it. And the FDA has been learning as part of this process, as have the communities with Critical Path and FNIH, et cetera. Much like Martha's quote around that we all own this. This is a paraphrase of a Dr. Woodcock response I had heard in the past, that basically it takes a village to read a biomarker. FDA cannot do this alone. Everybody is participating their piece to the puzzle. And then in 2015 and 16, there were a number of workshops. This is just a listing of those. Everything from a CIRSE meeting with the University of Maryland, a Duke Margolis meeting around biomarker development, the National Biomarker Development Alliance put on a meeting that was a case study approach. There was the FNIH meeting, and then we have the workshop today. So I think what really this list shows is how engaged the various people in this community have come together to say that this is important. We are willing to stay at the table and we're willing to help. And so that's all bodes well, I think, for the future of us having greater understanding in this space. An outcome of the FNIH meeting was a framework of how to think about a biomarker development effort. So not to go through all the details, but ultimately you have to start, well, what is the need that you're trying to fill? What is the drug development need specifically that you're trying to fill? If you're going to invest resources in a biomarker development, if we are going to invest resources, we want to know that you're going after something that's going to be impactful. So really stating up front what your objectives are and what you think would be the benefit should that biomarker be successful is critical as a first step. From there, you go into what is the context of use, what is the question the biomarker is intending to address. Then you put it in the perspective of the clinical trial and the benefit and risks of that decision, ultimately leading to what level of evidence will need to be brought forward. As you can see under the evidentiary criteria, the second to last bullet is the assay performance. So this is that piece to this overall puzzle or framework. And just to draw attention that there's also a stats working group that's working on the statistics of this. So again, each group is taking on their piece of this overall effort. So the thought for the day, what are the distinctions between analytical validation for assays used in clinical practice versus those that are used in the regulatory space? I think it's useful to think about of the different types of analytics that are out there. Many different communities are using them for different purposes. What is it that we can learn from those adjacent efforts to distinguish what is the same for qualification and what is different, and why is it different? Think about the impact of the best categories of biomarkers in the context of use to better understand what analytical performance characteristics are important. Certainly there's a laundry list as outlaid in the white paper. So I think one of the goals that we're trying today and over the course of today and tomorrow is to come up with, well, what are the unique subsets based on the context of use and the benefit and risk that we would like to see around the assay analytics. It's not a one size fits all. You're not going to be doing the full analytics for every context of use. So trying to figure out how you subset that, I think, could be a worthwhile goal. Ultimately, it comes down to level of certainty and uncertainty. And so that depends on the context of use and the benefit and risk. How does the phase of drug development affect what type of analytical and extent of analytical validation is necessary? We certainly acknowledge that when you're developing an assay, it's an expense, it's an investment. So if you're doing an early phase exploratory biomarker development, Are you going to do a full court press for a CDRH level clearance of that analytic? Probably not. But what level of analytics would we like to see? And how can you prepare for the future if that biomarker then progresses in the drug development program? And then finally, what parallels can we see between how diagnostics are used in the IND space for clinical claims from CDRH and then ultimately qualification, trying to figure out that intersection. So with that, I will turn it over to Steve, who will walk us through the elements of the white paper.
Thank you all. I would like to give you a little bit of a history of the white paper, and I will touch on some of the topics that Chris has already mentioned, but hopefully to reinforce them. So here we are, overview of the analytical validation white paper objectives and some key terminology. We will go through the first part. So this is actually a collection of some of the meetings that Chris has already described. You can see the particular players. I won't go through this in detail since he's already done a nice job of that, but look at the output down at the bottom on the left and the right. The Best Biomarkers Terminology Guide, which is available online and you are strongly encouraged to use. And on the right hand is the paper put out by the FNIH Writing Group on evidentiary criteria for biomarker qualification. An important document which is also freely available. Those are two of the things which are actually foundations for us moving forward here. One of the things, though, here is that in these series of meetings, there was not an overwhelming amount of pharmaceutical industry participation. On the next slide here, this happened rather independently from all of those meetings, although there was some small amount of overlap. Here you can see a number of the groups that have been involved in doing this from an industrial perspective. The American Association of Pharmaceutical Scientists, CFABs, and the workshops on recent issues in bioanalysis meetings, Global CRO Consortium, the European Bioanalytical Forum. And a lot of these things came together in a series of meetings which were known as the Crystal City 5 and Crystal City 6 meetings, at which there were many participants here, there as well. But what we brought out of Crystal City 6, along with the revelation that biomarkers are not PK assays, was a ill-defined path forward with a lot of questions still remaining about how we deal with biomarker validation. And actually, out of the evidentiary considerations meetings grew the task to do this. So these are the pertinent FDA guidances which exist for biomarkers in some sense or other. Not all of them actually mention the term biomarker, which makes it really difficult to bring specific concepts from these documents into biomarker validation. The BMV of 2001 does not use the word, neither do a number of the others. So how does this give us guidance to go forward, to do something to set up a assay development and validation scheme to qualify biomarkers and then provide data for drug development submissions? So these are the meetings again. I won't rehash them. There have been a lot of things. Most of the output from these, it's important to note, is publicly available. Most of these, by going to the websites where the meetings were, have transcripts and or slides. The white paper outputs are also available for these things. They're very valuable resources to this, but they aren't codified, so they don't come directly from the FDA. These are collaborative concerns that have put these together. Here you see the Crystal City workshops, the Brookings Institute, and at the bottom, the FNIH Biomarkers Consortium workshop. It was there that we were really given the charge to go ahead and do this, to provide a scientific consensus, a framework for assay validation for qualification of biomarkers with the specific context of safety biomarkers, which we will talk about later this afternoon. So the rationale for this white paper. Characterization and validation of biomarker assay performance as fundamentally important to biomarker qualification. I'm sure everybody agrees with that. Multiple guidance documents, as you've seen already, have been published for in vitro diagnostic and pharmacokinetic assay development and validation. However, Though these documents contain nearly all the fundamental concepts necessary for the development and validation of biomarker assays, they are not specifically designed for use as drug development tools. They have the concepts, but they have not been put together in a package that is directly applicable to drug development tools. However, the direct application of these concepts has not yet been codified into guidance for biomarker assay validation. So how do we get there? The pathway. At the FNIH meeting, Shavre said, we need to put together a white paper and get this out as a consortium effort of people to develop the scientific consensus necessary for that. So, very good. You now know how this started. That was a great idea, energy efficient, as you can see. And on to the next step, which is the stakeholder initial meeting, which happened at the beginning of last November at the Duke Margolis Institute at the other site before the new one here, which is really very nice. And from that, we started to develop the ideas behind this public workshop. Just to make no doubt, you are here now. This is where we are. So this is open to the public. We are here for comment and an iterative process of developing these guidelines going forward. So this is what we hope to leave here with, a goal of iterative refinement of the white paper, of discussion between the regulatory and the industrial concerns. Scientific input on both sides has been very important so far, and taking this to the next step where we have what we can agree on is a good scientific framework for assay development and validation that is appropriate for everyone's needs in drug development. So the primary goal as a collaboration between the scientists at the FDA and pharmaceutical and biotech scientists, the goal of this effort is to define the scientific and regulatory considerations for the validation of soluble biomarker assays used in the qualification of drug development tools. That leaves out a lot of things, but it actually narrows it down to something that we can manage and is very, very necessary. The stakeholders are not just the FDA and the pharmaceutical industry scientists, as you can see here, but it is rather a conglomeration of regulatory agencies and industry, but academia, federal partners, advocacies, foundations, societies as well, and various consortia that are coming together to put this together. And the end goal in the center there, biomarker evaluation, qualification, and utilization. So there are a lot of people who have input into this because there are a lot of people who get benefit and need to have this work well. The requisites for the points to consider white paper. We detailed these out before we started this. We need to obtain consensus on the experimental characterization of the biomarker assays used in qualification, which is the assay consideration step. The approach to defining the requisite assay performance and acceptance criteria, the assay validation acceptance criteria, and we would use examples of safety biomarkers of nephrotoxicity. This is important because one thing that's not been detailed so far yet is that although there have been a number of qualification submissions, most of those are not public. They are part of a drug development project for a company, and that material is proprietary. One of the things that we had intended to do here is qualify a class of biomarkers as well as make that information publicly available so that you have the ability to see what actually was done down to every level of detail that you would like and show that that was acceptable in a biomarker qualification to really set the public bar for doing this. So assumptions that we made to do this, and you've heard this before, but the validation expectations for biomarker qualification assays are not identical to the expectations outlined for PK or TK assays in drug development. Everyone has managed to agree on this for a couple of years now. The performance characteristics of a biomarker qualification assay are in line with the context of use and ultimately the clinical application of the biomarker in drug development. They are distinctly linked. So we have to link the performance to the context of use. Although an IVD, an in vitro diagnostic assay, is not required in biomarker qualification efforts, adequate assay performance and validation is essential for support of that qualification's COU. So you can use a RUO test, but you have to show that that supports your final context of use. Biomarker qualification assays are not sufficient for and are not intended to be used as de facto substitutes for an in vitro diagnostic device, that is PMA approvals or 510K clearances by CDRH. They are designed for the qualification of that biomarker, and that's it. Qualified biomarkers and their associated defined assay performance expectations are suitable for use in drug development and regulatory systems, that's our intended purpose here, but are not assumed to be directly applicable in or transferable to clinical practice, those laboratories regulated by CLIA. So this is not designing this assay to run these things commercially in a CLIA-regulated laboratory. Only single-plex soluble biomarkers measured by ligand-binding assays and liquid chromatography mass spectrometry technologies are within scope of this paper. There are far too many with different criteria for judging than we were able to do. This is our first step, and we're limiting it to 2 popular analytical technologies, but we're distinctly limiting to those 2. As well as soluble biomarkers and singleplex. We're taking baby steps here to get this done, and we will advance into other fields and multiplex assays at some point in the future. So all defined categories of biomarkers are within scope, in line with the COU, and they are listed there, and the definitions, again, are in the best resource. So the structure is actually, since you've all seen it, this can be fairly quick, an overview, biomarker qualification in the context of use, analytical validation versus clinical validation. We are talking about analytical validation here of the assay, not the clinical validation of the biomarker. Biomarker assay validation and how fit for purpose plays into this. Our preferred term is context of use. History of guidance documents relevant to the assay validation so that you can see the progression of how this came to be. Moving on, assay design and technology selection for biomarker assays, the things that you need to consider before you develop this assay. The pre-analytical considerations where many things are less well controlled than you would like to have them believe, and they're very important for generating high quality, robust, and reproducible biomarker data. Analytical performance requirements. We all know in developing assays that we can always make it better. All it takes is another 6 months and another $400,000, and we can do it, and we can do it just great. What is enough, and how do we know that we have established that analytical bar of what is sufficient for our context of use? Then assay performance key parameters. Those are the things that tie into that context of use that say this is generation of the appropriate data in the appropriate amount to lead us to the context of use. Not too much, not too little. A lot like Goldilocks and the Three Bears, if you will. Hitting it right. The assay validation acceptance criteria will, the criteria listed here, all of those should be painfully familiar to everyone in the audience. But those are the things that we considered were the key parameters that we needed to do for any assay that we were developing so that we had faith in the data that was generated on any level, regardless of the context of use. And as the end, case studies that we would link. I already mentioned the kidney safety biomarker clinical validation, which is a panel of 6 biomarkers we'll talk about extensively in the afternoon with a composite measure output. And also a case study of liver safety biomarker clinical validation, GLDH, which is a single marker and is also coming through the biomarker qualification process. So this is the document which you all have in your hands now. This is not a final version. I think it still has draft watermarked on it in very large letters. So one of the purposes of this meeting today is to obtain more comment from more stakeholders and drive this to the point where we really do have complete consensus among everyone that I showed on the stakeholders slide, and then can move that forward as a framework for biomarker qualification. So we thought that it was necessary, as Chris mentioned before. There have been meetings where we've argued more than shades of nuance about terminology. We need to know exactly what the words mean. They have precise defined definitions. And it's obvious that we need to do a little bit better job of defining what it is we are speaking about before we open our mouths and do so, to eliminate confusion. So I'm going to go through briefly a few of these. It would be a little pedantic to go through the entire list that we have, but to show you that we are looking at these things in terms of a very regulated definition. So these are the drug development process qualification, drug development tools, DDT, the qualification terms that are listed in BESS. And I urge you to use that as your best friend in biomarkers. It is updated. I do believe the last update was just before Christmas last year, early December. It has evolved and will continue to evolve as the white paper will. We actually do have the ability to add things to that, to change definitions based on output from this meeting or comments to the FDA. That's the, quote, living, breathing document that can be a very helpful place to use as a resource. Biomarker, if you don't already know this, you shouldn't be here. But here's the definition. The important thing here, context of use. A context of use is defined as a statement that fully and clearly describes the way the medical product development tool is to be used and the medical product development related purpose of the use. So that's what and why. We're going to talk about how for the analytical parts. But that's a little bit more detailed than fit for purpose, which we will go to in a moment. But the context of use is what drives all of this process. So as Chris pointed out, a few of the things that are necessary to determine for the context of use, these things have to be done as the first step, not the last step in the assay development process. Before one starts to go into the laboratory and design experiments to do something, you have to know what the end goal is. That is all dependent on the context of use. That's our most important concept to get over today. Qualification is a conclusion based on a formal regulatory process that within the stated context of use, there's that word again, a medical product development tool can be relied upon to have a specific interpretation and application in medical product development and regulatory review. Pretty clear? Now, analytical validation, we have a tendency to confuse analytical and clinical validations. So we are putting those two in separate definitions here. Analytical, establishing that the performance characteristics of a test tool or instrument are acceptable in terms of sensitivity, specificity, accuracy, precision, and other relevant performance characteristics using a specified technical protocol. This is a validation of the test, tools, or instruments' technical performance, but is not validation of the item's usefulness. An important end piece to that. Clinical validation is establishing that the test, tool, or instrument acceptably identifies, measures, or predicts the concept of interest. Those are very different to the regulatory people in the room, and therefore should be very different to the rest of us. Non-drug development tool terms. These are largely the definitions that we use in the industry to construct, develop, and put assays into our workflow process. So you can see here, although everybody thinks, yeah, I know what accuracy is, this is actually a formal definition accepted as a global standard referenced here. And the accuracy of measurement is the closeness of agreement between a measured quantity value and a true quantity value of a measurand. That has some important words in it, measurand, not analyte or target. And accuracy is actually something in the biomarker world which we do not have. It is nearly impossible to have defined accuracy and absolute quantitation. And we have this here as a definite term to compare with accuracy relative. The next line down, the closeness of agreement between the result of a measurement and the true value of the measurement, which sounds a lot like the first, but it's not quite the same thing. In practice, an accepted reference value is substituted for the true value. This is where we live in the biomarker world. We don't have, quote, gold standards. We don't have true certified reference materials. Those don't exist. 3 or 4 of them do, but that's it for the biomarker world. And this is where we cannot quantitate precisely what we want to do. We don't have appropriate in vivo standards. Everyone knows this, but we need to recognize this and quit saying that we are dealing with accurate measurements when that, in fact, is not what we are doing. Allowable error is a concept that we needed to flag as part of the development of assays in relation to the context of use. If you are going to prove that this assay is suitable for that, how do you know how much assay you can tolerate, how much error you can tolerate in the assay and still obtain clinically relevant information? And so it is important to understand exactly how much error you can tolerate without invalidating the medical usefulness of the result. Analytical sensitivity and specificity, I think those are actually pretty well dispersed in the arena. Bias. Bias is something that we tend to overlook because bias plays into our relative accuracy. Bias is any systematic error that contributes to the difference between the mean of a large number of tests and an accepted reference value. So this is where failing to have appropriate certified reference material comes into play. We don't know how big our deviation from an in vivo molecule being measured to our reference standard or manufacturer's suggested reference material. We don't know what that is, and that plays into our ability to accurately measure biomarkers in a patient population. Detection limit. This is actually something that we thought was necessary to define, along with limit of detection at the bottom. The smallest quantity, everyone is reasonably familiar with this, but it's important to keep in mind that the limit of detection is not analytical sensitivity. We need to be very precise about the terms, and precise, I guess that's a pun. Method development, qualification, and validation for exploratory purposes. Our reference for that is the Jean Lee paper, which everyone is probably quite familiar with, and that has been accepted largely in the community as a white paper, as a recognized standard. And these are for exploratory purposes. We are not discussing exploratory biomarker qualification here. We are moving beyond that, but this is actually the first stage down that road. Parallelism is the extent to which the dose-response relationship between two materials, the calibrator versus your unknown, is constant for the examined range of concentrations. The CLSI document there is a good reference for how one determines that. Reportable range is actually the whole linear range that you can report based on the final assay construction. Selectivity and interference, the ability of the assay to determine the identity of the analyte definitively in the presence of other materials present in the matrix. If the lack of selectivity comes from a known source, it is interference. If it comes from an unknown source, it is matrix effect. Spike recovery is the process of comparing the amount of analyte present in a sample after a standard has been added to and extracted from as compared to the true concentration. This is where we get into difficulty assuming that a spike recovery is actually an analytical methodology which allows us to determine accuracy in the original sample. It does not. It is comparing a false standard against an in vivo standard. And though it has some use, it is not sufficient to determine that the assay is suitable for its context of use. Trueness, at the end, trueness of measurement is the closeness of agreement between the average value obtained from a large series of test results and an accepted reference value. So it's really the opposite of bias. How close are we getting to truth? And these are pretty much the basic things that we have gone through in here. I hope that I did not bore everyone to tears with these, but it is important that we all agree on these. There's a much more extensive list in the paper. We have a much more extensive list actually in a glossary in the paper as well, but it's important that we all speak the same language. So here are what we have used as the sources for our definitions. The best biomarkers, endpoints, and other tools. If you don't have the thing, don't Google best biomarkers. You'll get such nonsense coming down for things. Here it is. It's extremely helpful and settles arguments with people about whether or not what you're doing is prognostic or predictive. The guidance for industry and FDA staff qualification process for drug development tools, January 2014 issued guidance, is available from the FDA's website. That is the drug development tools. That is where a number of the definitions first appeared before they were put into BEST. It is how to qualify all of your drug development tools, and it is applicable to biomarkers, although not distinctly dedicated to biomarkers. It is an excellent source for how to do a number of the things that are listed. And as a third, the CLSI Harmonized Terminology Database. CLSI is an organization which produces international standards which are harmonized between government regulators, industry, academia, and set out as guidances for clinical laboratories. Again, not what we are discussing here. They are, however, an invaluable source of methodology and definitions about how to do the things that we may want to do here. So, with that being said, I think that is the end of my talk. If there are any questions, I'd be happy to entertain them. Any comments or suggestions at this point? Yes, sir.
Do you already have a plan to extend these guidelines to other analytes like microRNA, effects, analytics?
Someday. We need to get this one through first, and that's why we limited to the targets that we had. So this was something that we could achieve, and we'd like to get this done, but other things will be next.
Just a second question, with the LCMS approach, you always work in a multiplex mode. So you mentioned you don't look into multiplex assays.
So a good point, but it is a multiplex analysis for a single measurand, and that is a difference. And actually, thank you for bringing that up, because the examples that we showed today are all based on LBA. We actually did not use mass spectrometry methods in the development of the two programs that we'll talk about today. They're still in scope. Any other questions or comments? If not, then I will turn the podium over to Meena Subramaniam, who has recently moved from Biogen to Takeda, and we'll talk about assay design, development, and validation. Thank you.
Thank you. So good morning, everybody. If you need to get up and stretch, feel free to, because if you're going to be ramming more of the concepts again. So thank you for the previous speakers for setting the stage as to what this framework is all about. And the good news is assays are being taken very, very seriously, right? For all the assay scientists who toil away in the basement of buildings and trying to justify why they need more time to get things done, I think this is a very welcome change where there is broad recognition that methods are foundational, and you having the right methods is what is going to enable the qualification of very important drug development tools, which we are referring to as biomarkers here. So my job here is to walk you through what is contained in the framework with regards to assay design, development, technology selection, and some of the pre-analytical factors that need to be considered. So when you look at the framework, there are two significant areas where we want to have consensus and agreement by the end of the meeting. So one is in the experimental characterization of the methods, which you can refer to as assay development, and the other aspect is assay validation. Okay? So the way this is structured is, I'm going to give you a quick overview of what is contained in the framework, and then we're going to have an exciting discussion with panelists. So please line up your questions, and the panelists for the first session are Sashi, Robert, Amanda, and Jean Lee, who will come up after I finish my brief overview of what's contained in the section. Okay, so the reason why I am going to redefine what the biomarker qualification program is, when I got involved with this committee, there was clearly confusion in my own mind because of the use of the term qualification. In the bioanalytical world, we use the term qualification to say when an assay has been developed and partially validated. We used to say a qualified biomarker test, which is very different from qualifying a biomarker for clinical use. So I just want to reiterate that qualification comes in two stages. There's analytical validation, and so development of the methods, and qualifying the method for its capability and capacity, which is the validation of it, and then demonstrating the utility of a biomarker for making clinical decisions, which is the clinical validation. So it was a good learning exercise for me, and so it changed the lingo we started using within my own laboratory back at Biogen, because we were using this term pretty loosely as to what we qualified as a qualified marker or a qualified test. So another definition here, you heard what biomarkers are, and Steve said you wouldn't be here if you didn't know what they are. But I just wanted to point out that biomarkers are actually endogenous molecules or entities. We are used to thinking about drug development in the context of exogenously administered products, which you're trying to measure. But in this case, it's really an endogenous measure of a change in an endogenous molecule or an entity. And so the US FDA biomarker qualification program, really what was the purpose of it is to provide an opportunity for external stakeholders to work with CDER to discuss the utilization of these biomarkers in drug development, the value of them, say, for disease diagnosis, for staging disease, or to support the development of a therapeutic. So the goals of the platform are to qualify these biomarkers and to make this information publicly available. And a good example of that is the nephrotoxicity markers that we'll be discussing at length this afternoon. And also to facilitate the uptake of these qualified markers. The purpose of the program is really to enable the industry and the academic community to utilize these markers to make better decisions and to make faster decisions, right? So there's one thing about qualifying markers, but if it's not broadly adopted, then the value of this really is, you're leaving the value on the table. So I just want to reaffirm that that is the goal here. And also, besides the known markers, there's a desire to encourage the identification of newer markers. So let's talk about then what is it that we are discussing within the framework of this white paper. We are talking about biomarker assays, which are typically measuring an increase or a decrease in the endogenous level of a measurand or an entity or a molecule. And so one thing that we need to recognize and acknowledge is that the level of these molecules can change, and they are affected by individual variability in physiology, in the disease biology, maybe in the disease pathology, and that's the reason why you're interested in measuring this marker, comorbidities, treatment. So these are all pointers that we need to keep in mind when we think about measuring these markers and trying to apply them in the context of a clinical decision. So that's something that is highlighted. And then the next thing is we really need to keep in mind when we develop these methods, what is the molecule that we are trying to measure? Is it a protein? Is it a lipid? And what kinds of decisions? So the context in which we'll be using this methodology and applying it within the clinical studies to make a decision. So the context of use becomes very important. Is it going to be a prognostic marker? Is it going to predict? It's going to tell us about safety events that may occur. Are we going to use it to understand a drug dose response? So there are many different ways in which you could be utilizing this biomarker. You may be using a single biomarker to make multiple decisions. So, but for each of these instances, you have to define the context of use and understand whether the assay's capacity and capability is suitable for that purpose. So that's the fitness for purpose. So what is covered in the guidance? Actually, Steve already covered this very nicely, but I'll quickly walk through this. So really, we are focusing on analytical validation of single-plex ligand immunobinding assays, mass spectrometry, and enzyme-based assays. So to think more broadly about these. However, what is out of scope is the validation of histology assays or flow cytometry assays, genetics and genomic methods, And also what is out of scope is the development and validation of assays that are regulated by CLIA. Okay? But the main point I want to make is these are assay development and validation principles. So these are broadly applicable to exploratory biomarker methods that are currently being developed and implemented as part of drug development. So these are general principles. These are good scientific principles that can be adopted more broadly. And so we don't want to really make this context very narrow in terms of validation principles. So the first thing to remember is, it's like the foundation of a home. So the accuracy and the reproducibility of the data that is generated using the biomarker method is very important to qualify the biomarker. And therefore, we have to pay particular attention to the accuracy and reproducibility of the data, which is, what enables that is the method itself. And so the characterization of the assay capability and its limitations is very, very critical. So there are several areas in the white paper that this is emphasized. And the main reason for this is that any errors that are generated in the measurement of that measurand or the analyte then translates into really biasing the information that is generated in the context of the clinical utility. So we have to be very particular in defining clearly how precisely does this assay measure the particular analyte of interest, okay? And then the performance characteristics, once again, I want to reiterate that is dependent on the context of use because that helps us define how much can we tolerate any deviations in the performance in terms of either its precision or the bias in the assay. So what is the tolerance around that? And that is really dependent on the context of use. And this has already been stated that, even though we don't believe that the parameters that we're going to use to validate these methods are equivalent or identical to what is being done for PK or TK assays, so they are not, a carte blanche use for this. We have used that as a framework because the characteristics that we have to assess are the same, but the criteria for acceptance may be different because of the difference in the molecules that are being measured. And this was also stated previously. We should not assume that just because these methods have now been developed and analytically validated, they automatically become clinical diagnostics. They are not. So these are not cleared by CDRH, either through the PMA pathway or through any other mechanism. So these are typically, they remain as test methods for supporting biomarker qualification, but these then can serve as the prototype for development into a diagnostic in the future. So you should be doing much more evaluation of these same methods to then eventually transform them into a diagnostic device. So the definition for context of use has been provided, but I just want to reiterate that it is really the intended purpose. And as I mentioned, you could take the same biomarker and apply it in very different contexts for qualification of a tool. So in every instance, we need to be certain that the analytical validation supports that context of use. And so the example here is, what is the test population? Is it going to be for non-clinical use, or is this going to be for use in humans? And so then are you comparing the difference in the expression of the marker in healthy humans versus a disease population? If your context of use is to stage disease, do you want to look at stage 1 of the disease versus stage 4? So you really have to plan when you're designing the assays as to what is it that you're trying to demonstrate so that you can understand if the degree of difference that you're measuring between these populations can be supported by the method, right? So that is the distinction that you have to make in terms of the context. And then it is also important to recognize that there are many factors that can impact the measurement of biomarkers. So starting with the collection time. So urine is a classic example, whether you're going to collect it first thing in the morning or the last thing in the evening. The methods that are used to collect in terms of the tubes that are used, the anticoagulants if it's blood, what kind of anticoagulant you're going to use, how are you going to transport the samples, how long can you store them, how are you going to preserve them, right? Do you have the right preservation defined for these samples. And then contamination, how clean does the sample need to be? And a classic example is blood contamination in CSF, which can really throw off your analysis because you'll now be measuring levels that really are not present in this, in the sample of interest, but it's present in the contaminant. So these are some things that you need to pay particular attention to, because this is where you're defining exactly what are the ingredients that you're going to use to create the test. And you have to be very specific about the quality of the ingredients that you want to use, which is essentially your sample. And then the use environment, right? So how is this test going to be used? This is going to be in a very controlled environment, like in a laboratory with very well-defined methodologies and instrumentation, or do you need to use this method in a field like condition, right? So, and very high volume testing versus low volume testing. So all of these sorts of parameters are going to be important in designing how complex or how simple you need to make this test and the readout from the test. So that's what are some of the things that are considered when you're trying to develop the method. And the next point that is highlighted is, do you have professionally trained analysts who are going to be running this, or this is going to be run by a nurse practitioner right at the doctor's office, right? Though there are many measures that can be done very rapidly at a doctor's office. You don't need a very highly trained bioanalytical person running it, but it could be a very skilled operator who's running it, And then long term, will you be making changes to the method based on the data that you're collecting? So, iterative changes that will be made to the method for improving its performance. So these are considerations when you think about the design. So you don't have to have the absolutely perfect method off the gate, but you have to make some critical decisions as to how much data needs to be collected before you think about updating or modifying the method to improve its performance characteristics. So this has to be thought about as a process of continuous improvement until you hit validation. And many times, as you know, validation can also be repeated, right? So we learn certain things after we do clinical analysis, and we then do some addendum to our validation to improve the reproducibility of the method or the stability of the samples or whatever else. So we need to keep that in mind, that this is an ongoing process. And then finally, in terms of the designing the method, establishing the working criteria for the assay is very important. In terms of finalizing your technology, do you have adequate sensitivity? Do you need to change, really the detection method that you're using? All of this is important. So it goes back to the context of use. What is the difference that you're trying to measure between A versus B? And how large is that signal? And if the difference is very small, but it is still meaningful, then you need to have a technology that can support that. So keep these things in mind. And so that's highlighted in the paper. Then let's talk about characterizing the assay capability. So these are the features that should be considered. So the white paper walks through in a very succinct manner things that you need to keep in mind in terms of how do you plan to report the data? Is it a qualitative yes or no, so a binary response, or is it going to be semi-quantitative like a titer level? Right, that you typically report for, say, a viral infection and you're looking at an antibody titer, so that could be a very good biomarker. Or is it going to be quantitative, relative quantitative? So I want to be very clear here, unless you can really do true quantitation. So that's the first level of decision that you're going to make when you think about designing a biomarker test. Then talking about what are we going to use as our calibrators and reference material, right? Are these going to be our recombinant standards that are going to be spiked into human serum or animal serum, depending on what you're developing the methods for? Or are they going to be endogenous materials that are purified and used? So there are different levels of decisions that need to be made when you're designing the assay. And then think about the controls that are going to be used, right? So many a times we end up purchasing kits commercially for use in biomarker tests, but these kits do not always come with representative controls or reference standards that you think is of value. So you may end up throwing every other thing from the kit besides the capture reagents, and then you may create your own calibrators and controls. So when you go through this validation, you have to really think through what is a representative calibrator for me and what will be a good quality control that I can use so that every time I run the assay, I'm convinced that the system suitability criteria are met. So you want to make sure that they are robust samples that can be used. And also scalability. Remember, we talked about large and small volume testing. So you don't want to prep like a 5 ml of your calibrator and then run out of it after you're just a couple of tests. Then you're in this, a situation where you have to bridge now your control lots from A to B. So you want to be aware of that. And then reportable range or the reference interval. So this is one thing that we thought is of value, to highlight, because this is not something that people are typically used to defining when they think about a PK assay. We think about it as, your limit of qualification or quantitation. So in the document, although it's not playing here, the reason it's highlighted is I wanted to point out that each of these terms is clearly defined. What do we mean by this, right? So what is a reference interval? So when you think about healthies versus disease subjects, for each of these groups of patients, you may have a reference interval for the level of your biomarker. So the way you make your decision is there a difference between my healthy versus my disease population using that reference interval? So you'll have an upper and a lower limit for each of these populations for making your decisions. So please check out those references, and I think this is important when you read the definitions. If it is not clear what is meant by the terminology, we'd love to hear from you to make sure that we can clarify what we really mean by those phrases. Specimen quantity, how much should you be collecting? Right? Everybody wants to create a biobank, and you have to take this opportunity when you have the patients, in a phase 0 or your phase 1, 2, 3 trials, or even outside of that, how much of the sample is going to be needed so that you could be, ensuring that the biomarker is being reproducibly measured, not just at a single time point, but even over time. And then allowable analytical precision and total error. I'll get to more about this in my later slides. What is the desired detection sensitivity? So how are you going to make decisions between the value of the biomarker between, say, a healthy and a disease population? So what is the difference? So do you have an assay that can support that? So it's really the putative detection range. And then interference factors. You want to make sure that you have selectivity. You're picking up the right sort of biomarker from this complex matrix. So what other things can interfere in that measurement in this endogenous matrix? So there are several other factors that we'd love for you to consider when you design the assays. I'm not going to go through each one of them, but I want to highlight, in case you missed it in your reading, that all of these are there in the framework document. So let's talk about then you made the complex decisions about what you want to report out, what reagents you're going to use. Now, how do you go about constructing your assay? So here we introduce the concept of DOE, or the design of expert, which is a methodology that is broadly used in the industry. So it's not new, but we want to highlight to you that it's a simple way of looking at multiple factors. And by using a checkerboard or a design of experiment approach, which is a fractional factorial experiment, or you could use a central composite design, you could evaluate what the impact of various different factors may be on the test in a single shot, right? So you could look at the minimum required dilution. How is the minimum required dilution impacted by the reagent concentrations that you're using? What kinds of calibrator levels should I be, thinking about? How long should I be incubating? What kinds of blocking reagents should I be using in order to, determine whether I'm getting any non-specific binding? How many times do I wash? All of this can be done in one shot when you use a design of expert approach for doing this. Okay? And then finally, defining your system suitability. When are you going to be happy that your assay is performing reproducibly over time in the hands of multiple operators? So these are things that need to be considered when you're setting up the test, because this is what is going to then help you move the test into a validation phase. You don't want to get to validation without understanding what your assay capability is, right? So the work that is done a priori is going to really minimize all the headaches that you may face during assay validation. Then finally, let's get to the variation and bias. So if you want to develop quality specifications for your method, the two key parameters are really precision and bias. So the framework document really goes through and provides you some guidance around how can you think about generating this data, right? So the first goal of variance is to understand within subject variance. So when you execute a test using samples from a single subject versus when you're going to take now multiple subjects or between days from the same subject, what sort of variation do you see? And so there's some guidance around how you can execute this evaluation. And obviously, the last bullet point says there, more subjects you use, the better it is, because then you could, determine really what the actual variance is. And the fewer the samples, it's really hard for you to get a good sense for what the variance is. The next component in the total allowable error is bias, and Steve already referred to this, right? And what we need to remember is when you think about quality specifications, bias is computed by the addition of bias and precision in a linear manner. So you need to remember that both of these components contribute to this total allowable error. So if you have large variance, you have to try to minimize the bias, and vice versa, okay? And so I thought this is a simple pictorial will highlight how to think about variance and bias. So in the first row, what you see is when you have low variance, you do really hit the bullseye, right? So you're able to really do the measurement very well when you have low bias and low variance. Now, when you have low bias, you're still within that inner circle, but you see that the individual measurements are all over the map, right? So you have high variance in those individual measurements, but still your bias is well controlled. Now, if you go to the lower row, what you see is to start off with, you do have a higher bias because you're not really hitting that bullseye. You're really outside of that, place where you want to be in terms of where the trueness of the measurement is. You're already outside of that. However, the analytical execution of your method in terms of the variance is very well controlled, right? So the individual measurements are all clustered together, and then you'll see the counterpoint there when the bias is high and your variance is also high. So this assay probably may not, unless your context of use is pretty broad, this method may not be very suitable, so you may need to do some more work in the assay design. So that's how you use these two metrics in order to improve your assay. So that's essentially what we want to communicate with this. So the white paper framework also goes further into suggesting some goals that you can set up when you look at analytical variation and also for bias. So it walks you through what could be considered as an optimal variance, a desirable variance, and a minimal variance. And this is a good way to operate when you're designing your method as well as for bias. Okay? And there are some statistical papers that are referenced within the document. So if you have any questions, we can talk about this in the discussion forum. But the one thing we do want to emphasize is biological variability, right? Even though we design the assays and we execute it with the best of interest, when you go into the real world scenario and when you start looking at longitudinal variability of the marker, even within your control population, you may have to take that into consideration when you eventually think about what are those cutoff values that I'm going to define to make my decisions, whether there is a real change in this biomarker. So we just want to say that during your assay design phase, if you have access to samples that have been collected over time, and depending on your context of use, I think it is very useful to integrate it early on, so it gives you a good feeling for what the variability in this marker may be. So when you set your acceptance criteria, it'll already be incorporating this biological variability. Then finally, technology selection. I refer to this, in terms of sensitivity of your method. If you really have very small changes, but you truly believe that it is clinically meaningful, then sometimes technology can come to your aid. And also technology selection is important depending on whether you mean to use this methodology in the field versus in a highly controlled environment of a laboratory. So you don't want to be using highly cutting edge technologies if this is not broadly available in multiple laboratories or sites. So all of these considerations need to be taken in place a priori before you design the assay. So you don't want to choose an exotic optical method for which the machine is so expensive that laboratories across the world may not be able to afford this technology, right? So you want to be very pragmatic about how you choose this. So there are so many methods that are available these days, but what we need to consider is, how do we go about selecting the technology? So there is a little bit of discussion around that, where you use a controlled set of reagents once you have a preliminary assay design, and then you evaluate the various technologies. So as an illustration, I'm showing you an example from a publication here where they were using the sensitivity metric in order to select their, in order to evaluate various technologies. And what you see here listed is the various technologies that they were looked at, and they were measuring cytokines here. As I said, there could be different sorts of biomarkers that you're measuring, but in this particular instance, you're looking at a variety of cytokines. And here their goal was they wanted to measure all of these cytokines using a single technology. So they wanted to see which one was sensitive enough. So in this case, they used two different types of controls to assess the capability of the technology. So the first table here shows the cytokines that were produced from stimulated peripheral blood mononuclear cells. And they used the supernatant and they spiked it into human serum because that's the ultimate target population they were going after. And then they looked at the capabilities of these various technologies. The numbers don't matter because it's all relative. Within a technology, are you able to see a difference or not, right? So I don't want you to focus on that. And then what they did was to extend this analysis by using recombinant proteins. So they said, okay, what if, I don't want to be endlessly producing PBMC supernatants and spiking it, because then I have to show the continuity of, the controls and the calibrators and all that. So what if I use recombinant protein? Will I still be able to use the same sorts of technologies to measure the cytokines? And then finally, they used what is called as a feed analysis, which is the frequency of endogenous analyte detection. So they used samples from a number of subjects, and they said, how many of them did I detect this analyte in, or the biomarker in, right? And then finally, you can see that there were some technologies which were capable of detecting the biomarkers, the various cytokines in many of the subjects, or 100%, and there were some which were not capable of detecting it. So this is just, a suggestion. It's an illustration of how you can think about technology selection, and that we just want to tell you that that's an integral part of designing your assays. Finally, pre-analytical factors. I can't emphasize enough on this, even though I'm addressing it last, but there is something that you have to consider first when you think about your context of use and the sorts of samples that you'll be utilizing to design your assays, right? So there is a table that is dedicated to highlighting all the various, factors that need to be considered when you think about the pre-analytical factors that can impact your biomarker of interest. So you have to apply a lot of rigor in collecting the samples, in storing them, and then to think about what are the factors that may affect the biomarker of interest, like age, gender, ethnicity, exercise, the sorts of things that people eat and drink. And also, how do you then think about the pre-analytical factors and the samples that you use for qualifying your method? Okay, it may also change. The pre-analytical factors may also change across multiple assays and matrices, so that should be kept in mind. So the context of use becomes important again. So I do want to illustrate this with an example. So there are many publications these days that are addressing the importance of pre-analytical factors. So one that is shown here is from a publication by Zhao et al. From Journal of Immunological Methods, where they looked at the measurement of various fluid biomarkers, and they wanted to understand, should we be collecting plasma or serum? Right? And what is the degree of difference that I see from the same subjects when I collect plasma versus serum? And then they also evaluated the use of two different protocols, right? Should I be collecting using a traditional protocol, or what if I modify the way I collect it with, say, a different anticoagulant or, a different storage and shipment condition? How would this work out? And there are differences, right? So what I want to acknowledge is there are distinct differences based on the methodology that you use to collect samples. And the other one that I do want to highlight, because in my work at Biogen, one of the biggest challenges we had with the Alzheimer biomarkers was the way in which CSF was collected, right? So for us, even the utility of the biomarker became in question because we were able to pick it up sometimes and not at other times until we figured out it was all because of the way the samples were being collected, right? So a lot of effort goes into developing very good methods, but then when you don't have the right samples, it all appears to be in vain. So I think we want to emphasize that consideration given to pre-analytical factors really will pay off in the long term, okay? And then finally, going back to where I started about tests that are qualified versus validated, we now refer to this as staged validation. And so the biggest question that everybody faces in the biomarker assay world is, when do you have to have fully validated methods? And we heard clearly that, at the time of letter of intent, when you say that we would like to develop a biomarker for this specific context of use, it's a good idea to have a validated method, because then you start collecting the data to demonstrate its clinical validation. Okay? So, but in a drug development process, that is slightly different, and that's what Afshin will also refer to in the afternoon. So I'm trying to make a link between these two talks in that, mostly it's all exploratory until you hit phase 3 when you're trying to see if this biomarker can be truly used for decision making. But by your phase 2, when you're doing your dose range finding studies, many of us do have validated methods. It may undergo some refinement for phase 3, but we do have validated methods. So the table 1 that is incorporated in the framework, it walks you through what sorts of parameters you can consider in the early exploratory validation versus what is referred to as partial validation and then full validation. Okay? With that, I'm going to stop and I'm going to invite my panel members to come up. Unless you have burning questions for me, which we could also ask off the panel, because they've all contributed to this design section. So may I request my panel members to come? So the first order of business is I'm going to request each of the panel members to introduce themselves, and then we'll open it out for questions from the audience. I also want to say we have some questions, and I wouldn't call them as prepared, but these are questions that we all had when we attended previous biomarker meetings. So I've been collecting these over time, and we also had questions that were contributed by the panel members as good things to bring up and highlight in the conversation here. We'd prefer that it comes from you, but if we are struggling to get the conversation going, I'm happy to start with some questions. So we'll start with the introductions.
Hi, I'm Amanda Baker. I'm from the Critical Path Institute. I've been working with the Kidney Safety Project qualification effort now for over 7 years.
Hi, I'm Shashi Amor. I'm in the biomarker qualification program, and I've been in the program for nearly 6 years now, and I've been in FDA for 12 years.
I'm Bob Becker. I'm a pathologist medical officer with the Center for Devices and Radiological Health, where we deal with the review of in vitro diagnostic devices.
I'm Jean Lee. I've been in biomarker for more than 15 years, I believe. And I was working with a CRO and then worked for Amgen. And then sort of retire, semi-retire, and my BioQualcomm is a consultant.
So thanks, everyone. So we'll open the floor for questions. Anybody has a burning question to start off with? Yes, please. Maybe get a microphone. Okay, I'll repeat it.
Thank you. And so the question is, I think it's a decision between pest and cholera. So I can come up with 3 EDTA plasma samples, right? So the right metrics as the clinical samples later on, but they will not cover the complete calibration range of the assay. And I do not know if my patient samples may have higher levels than the samples that I can get, obviously only from healthy volunteers, because I do not have access to patient samples. So this would be one limitation. The other limitation would be I could spike in calibration material to get, let's say, a high sample, However, a lot of times we have difficulties because the calibration material is not stable for a long time, has other difficulties. So especially when we're talking about relative accuracy, that's going to be an issue. So is there any kind of advice how to do that?
Repeat that question.
So if I were to summarize it in one statement, it's a challenge with calibrators when you don't have the right patient samples, and also to simulate high levels of the calibrator, right? So the spiked samples don't have the stability that they are looking for. So what sorts of approaches can be used?
So the first thing I'd like to just recommend is if you're working toward a qualification, those efforts are really done best in collaboration. And so, of course, the ideal is if you can get samples from a collaborator that represents the population that you'll be studying for your context of use. So that doesn't solve your problem of not having current access, but I'd say reach out to the community, and because those are going to be extremely valuable during your validation process for not only calibrators, but also for understanding your pre-analytical factors and potentially the differences in your biomarker in your disease population or population of interest versus your normal healthies. So I think you have to find a solution to get those samples.
So nothing takes the place of the samples themselves. I think that in the white paper that there is a recognition of the difficulty in sometimes getting those samples, and that you might need to take some intermediate steps that will get you at least part way down the road. Realizing that it's only part way down. If you're able to get at materials that can be introduced into a matrix of the kind that you're dealing with, and that can be of some help, but it doesn't solve the problem that you're looking at. It just helps get you a hint as to whether you're maybe going off track early.
I also want to comment because the validation is a continuum. So, I mean, before you probably would have already pilot study that involve patient population, and it will be usually during that time it will allow you to collect some samples that are of higher level. And so that will prepare you to have those validation samples. And also it is very important to really plan ahead to find a supplier that will provide you with the calibrator standard in a larger quantity and with the stability studied.
So did that address your question? Yeah. So I think.
No, I think important is that validation is the continuum. So
Yeah.
I can start off with a validation after the first study, getting the samples, and then do experiments that were not possible before, right? So if it's not available before start of the study, then keep the samples and then do it later, right?
Correct. So it is important to acknowledge that that is the situation when you're, so you're not going to make a final decision on the utility of the marker yet. But what you're going to add is an addendum to your validation to say, when I have access to these, samples with high concentrations of my analyte or measurand of interest, this is how the assay behaves, and this is the sorts of decisions we can make. Yes.
You ready now? All right, first I'd like to sing a song. Russ Wiener, Daiichi Sankyo, been involved like Gene, 20 plus years, responsible for bioanalytical biomarkers and companion diagnostic at Daiichi Sankyo. I just want to ask the audience, that individual, a question. Are we allowed to go cross-audience as well?
Of course, Ross.
All right. So that particular assay that you were developing, is that for early development or one of the qualified type of assays we're here? Because I want to make sure we don't squeeze and sneak back and forth, because that's when the confusion is going to come in and we will not come to consensus later.
Okay, so I mean,
Right behind you.
We anticipated this problem, actually.
well, that's when, well, okay. So you start off with the idea all the pre-consideration of we want to go for an assay validation for a marker that makes an impact. How far are we going to go? We don't know in the beginning. So, but we are thinking about it. And then the question is what? So one general aspect could be, what can I leave out to start with a very small validation? Or what actually can I do best and all to start off to get to this point, right? So I think a lot of times actually the biomarker may not meet then the expectations and will not get the state of a qualified biomarker. However, we want to enforce the biomarkers to be able to get to this point, right? So yes and no.
So the way I see it is getting back to what Jean said, and to deal with your specific problem of maybe not having high enough concentrations, is that in the early stage, you may not, but you are going to run pilot studies. You're going to look at those biomarkers in your clinical study. You're going to get a hint. You're going to, hey, this is really going to do it. And that would move it from that early exploratory world to the world that we came here to discuss. So again, I would ask all the moderators to make sure that when the questions are asked, we put it in the context of where it is in the drug development pathway. Thanks.
Yeah, but really, let me interject something quickly. When you're talking about early development, I assume you're talking about early development of the biomarker for a stated context of use, rather than early development of a specific drug.
Yes.
And it'll be really essential to keep these things very, very straight, okay? The whole idea of this is to be able to get at the validation of the biomarker that can be applied across a whole range of drug development programs. Very different from saying now you're going to actually apply what you believe is the qualified biomarker in the context of developing one or more specific drugs.
Completely
Always
agree.
keep that straight.
Great, thank you.
Also, I want to add another comment is that you need to plan strategically. So what you are asking the question, what is the most important thing? I think in your case, I would say the calibrator is the most important. If you don't have anything that your calibration is relied on, and also to make spike samples to test, that will be really very dangerous. Also, another thing to consider is that don't bank on only one biomarker if you can help. There may be other proximal biomarker that you can test, and by that time you will compare the risk and benefit, and then you will take the one that will be most beneficial to you.
Okay, next question.
John Allinson, LGC. I've been working in biomarkers over 40 years in different areas. And just to address that one point in particular about high concentration samples, I agree with Robert, nothing can be better than using samples themselves to prove the characteristics of the method. But in situations, I think, when even in the diagnostic arena, when we see really high patient samples, concentrations that we may not have witnessed before, I think it's really important to get out of the mindset of regulated bioanalysis where, you know, it's almost you cannot do a repeat on a sample without jumping through a lot of hoops. From an ethical perspective, I find it very difficult as a clinical scientist to actually report the data without repeating it in a characteristic parallelism type experiment to prove that we've not got an artifact in that individual patient samples. Depending on which area of pathology you work in, you can get really high concentrations of molecules you've never tested in the assay itself. And it's not unusual to find individual patients that have artifacts that give you misleading high concentrations due to interferences you just don't know about.
I think that's a key point about interference, right? And how well is the selectivity of the method characterized? So it's an excellent point that you raised, John.
Actually, God, this question's almost been beaten to death already, but I just wanted to, Russell Grant, I was one of the writing group, been in diagnostics for 15 years. So I just wanted to draw your attention to a couple of things that are in the framework. We talked about two nested ANOVA with healthy and disease and actually getting acquisition of the specimens up front.
Yep.
I think we got the concept of in-study parallelism and dilution, And I'm always reminded in working with analytical technologies of Donald Rumsfeld's quote, right? That we know, we don't know, and then there's the unknown unknowns. And an example of all of our best intention and finding marker measurements that are way outside analytical measurable range, you almost treat those, in my experience now, as do I really feel that I've asked enough questions of the selectivity of our assays? Because specificity is 100% selectivity, and we never have specificity. We're always doing trade-offs. So the in-study dilution, spike and recovery of calibration, and the two-nested ANOVA may be your best crutches. But let's always remember that we're trying to do perfection in an imperfect world, and human beings generally are quite disgusting. Present company accepted.
There's a question there in the audience.
So in the guidance, Frank Sestair from Merck. So in the guidance, we're talking about taking a blood sample, processing it, and measuring it, essentially. Why are we limiting it to just a single analyte type measurement? Why not multiplexes? Why not a tissue sample that's extracted and looking at RNA? You know, why are we limiting it? Because the principles are standard, are constant throughout. They're good principles. We can argue about, what level of performance each of those needs to meet, but I don't think the document's going to solve that problem either. That's always going to be an individual decision. So I would encourage broadening it, because I think we're doing a disservice to assay developers who can develop good multiplex assays. And you heard, I think one of the first questions out of the box was, hey, what about all these other things we're trying to do? These are really important to do. So I understand the risk of trying to take too big of a bite, but there's risk of taking too small of a bite too. There's consequences, I should say.
I think the answer to that gets to be that we wanted to get started with something we knew we would accomplish with a minimum of diversion, which multiplex assays tend to be. Because when we try and define all of these characteristics for one assay, we think we can do that pretty well. For defining half a dozen done simultaneously with interference and back to what is selectivity and specificity and how you relate those to the other molecules you're measuring simultaneously. This brought up a slew of problems that we didn't want to deal with on the first round. If we nail this down for single plexing, we will have a good foundation to go ahead and do multiplexing. We realized that multiplexing is a very valuable way to get a lot of information quickly, but we didn't think there was an easy path to get there for consensus, first off, without getting single-plex consensus.
But if you put the onus on the assay developer to say, if you can do this in a multiplex, you can show specificity, you can show all these parameters, bring it on, right?
So the problem with showing specificity is that you can't show specificity. You can show lack of interference up to a point.
I mean,
and that's as far as you can go. And when you are looking at one analyte, you have an array of one. When you're looking at 6 analytes, you have an array that's 6 by 6. It becomes a 36-fold bigger problem to look at that. And so that's why we started with one that we were sure we could get through. And then we can apply.
I don't know if others agree, but I think we should push the authors a little bit further.
Next year, Frank, multiplexing.
Let me add one more item to this. I think let's go back to that question after we talk about acceptance criteria. If we can, by the end of tomorrow, get this audience to agree on 70 or 80% of the acceptance criteria as a team, we come up with the acceptance criteria we can all live, then I would say we open it up to add other areas. The other thing is we've got to think about the stakeholders, people that are doing the RNA work or doing the immunohistochemistry or other things. I think in order to be able to expand it beyond what we're covering today, we've got to get those stakeholders to be involved and provide the input. So
I think that is something we want to emphasize, as we did talk about it, but the question was, when you're talking about multiplexing, are you talking about multiplexing the same types of molecules, Are you talking about multiplexing a protein with a nucleic acid endpoint, so on and so forth? So we got caught up in these discussions. That's when Steve brought us back and said, can we just agree on doing one thing, which is single plex? And then we can talk about, because it may be just for multiplex of the same molecule. Right. These are the same criteria that will apply, and it's a matter of integrating that. But then once again, it goes back to data reporting. And you want every one of them to meet the criteria or not. So go ahead.
And I'd also say, Frank, that the pre-analytical handling might be quite different.
It just has to be specified.
Yeah.
I think it just has to be specified. And the thing about acceptance criteria, I look forward to that discussion. But again, if the acceptance criteria are based on principles as opposed to an exact number, right? Yeah, exact number. I don't know if this guidance is going to try to do that or just talk about the principles of the acceptance criteria must be sufficient for the performance that you're seeking. Then it works for everything.
Yes, please.
This is Li Ching Ko from Sangamo Therapeutic. So just want to set the stage of what the company is doing. So we are a gene therapy company, so the calibrator doesn't exist for any biomarker assays. So we are using a recombinant version of the protein for all the biomarker assays. So the ultimate readout is the activity. So based on that, how do you actually set the criteria using activity, or do you using the enzyme concentration? How wide can you set your criteria because it's fit for purpose, and what is the tolerance based on the guidance, because there's nothing there to say, is it 30% or 20%, or how do you see from here?
So anybody in the...
You should have an early assessment of the total allowable error. And then you go from there and say, what is it that, for example, your patient population versus the normal, what are your enzyme activity? So, if mostly they are very huge differences, then you can estimate your total allowable error and go from there.
So do you, because do you accept the assay based on your recombinant protein concentrations and then translate that to enzyme activity? What if they don't align and you have your enzyme concentration, you go with, say, 20% just at the PK, and then your enzyme activity, can you set the different criteria for that? And do you use a normal donor as your controls, or do you use recombinant because it doesn't represent the true populations?
Right, so the one thing that you need to do is to characterize your recombinant material very well in terms of its enzymatic activity, right? So maybe the protein concentration does not translate into 100% enzyme activity. That is the first step you need to do. And then you articulated all the other right things that you would do, having a normal donor, right? So having multiple layers of decision making and being able to track how this, endogenously activated molecule, so you're trying to translate it into some normalized units of activity. It's not exact units of enzyme activity, but it's a normalization either to your recombinant control or to a normal sample. At least that will be my perspective to see how reproducible your data is going to be.
Okay, thank you.
Again, your performance characteristics would be, as Steve mentioned, linked to the context of use. What are you trying to get at?
So would that means that if we say context of use, so if we set 30%, will the regulatory in terms of acceptance, whether there will have issues saying your assay is not good enough and you have to redo your qualification and validation?
So I think we need to move away from that mindset of regulatory will not accept it. This is where we are saying you have to position what you bring forth in the context of use. If it is enabling you to make decisions confidently over time in multiple patients, you have to justify your assay performance as acceptable, right? The total allowable performance based on how you plan to apply it.
Thank you.
So Mina, let me just address this with an example. There is already an example that has already gone through the regulatory agency approval. If you look at the alpha-1 protease inhibitor, alpha-1 PI, it's an assay that's been around for 25 years, right? The discrepancy between the potency and the content, one reads 1 mg per ml, the other one reads 0.7 mg per ml. The initial assay that was developed, and it was used by all the manufacturers out there, and they're dosing the patient based on that, is at 0.7 mg per ml, right? So if you go do the content, it varies by about 25-30%. That is exactly as you said. You basically have gone in now, and they have actually characterized the difference between potency and the content. Now, in terms of acceptance criteria, you have an enzyme activity assay, which is photometric or fluorescent, and you end up with a CV that is very tight. So the acceptance criteria that you set up, or these manufacturers have set up, and the clinical use they have set up, is much tighter than a content assay, which is based on based on an ELISA assay, right? Now, same content assay is put on immunonephrolometer, which is an automated instrument. So the spec around that is much tighter. So it really comes back to exactly what Mina was saying. Depending on the instrumentation, the platform you use, the context of use, all those come into play. I hope that if you read around the alpha-1 PI history, some review papers, it really goes through what was asked.
I think with respect to both of those, it's important to keep in mind that this was never intended to be a proscriptive document with limits set. So 30% is good enough or not good enough by rule. And going back to the context of use, that determines what is good enough. So can you measure the clinical change of interest in the assay system you have at hand? That's the key point. That tells you whether or not it's good enough. I personally have taken 510K applications to CDRH that have CV on a year-to-year basis of 1% to 2% and 30 to 50%. Now, your immediate reaction is 30 to 50% CV in an assay. That's terrible. It was accepted. It was a little bit harder argument than the 1% to 2%, but we justified that based on the context of use. And we demonstrated rather convincingly that at a CV of 30 to 50%, we could get the information out of this test and make it medically actionable and have that accepted by physicians and the FDA. And that is an approved test, and that is sufficient. So the mindset that it has to be 20 or less to be good is really just inappropriate. It's what is it in the end you're trying to measure? That's not a number. That's a clinical issue. You need to determine, is there a clinical state or not? And those determine the numbers. So a lot of that can be done with the recombinant version of the protein. You can flesh out all of the variabilities and then adapt those, bring those into patient samples, and show that there is correlation or at least parallel activity between those two. And that's about as good as you can do at that point. But it's not a numbers game. It's a clinical outcomes game.
So actually my question relates, I'm Mark Rose, I'm from CHDI Foundation, and my question actually relates to what Steve was just talking about. By the way, a great document. I think it's a giant leap forward. I like the context of use, the fact that we're actually kind of cozying up a little bit to where we define accuracy and relative accuracy, and I really appreciate that. But what I'm worried about and what I'm seeing is this idea of context of use, and there are kind of two situations. One is a situation where you have a very early biomarker assay, and then the other situation is where we expect to measure a giant change, right? And what they kind of lead analytical chemists, they lead them to a place of carte blanche as far as using assays that might not be very good, that have high error. And I'm worried about this because I now work with laboratories, and you guys are talking about rigorous validation of a plus or minus 50% assay, but some people aren't going to interpret it that way. And is there any way that you could build into this document a way that something that emphasizes that an assay still has to be under control? We know based on the limits of the technology what an assay can do. And so if you have 1,000% change that you expect, is it okay to have an assay that's plus or minus 200%? even though we know the technology, gives us 20%. Is there anything that you guys have thought about to be able to do that? Because I don't like this idea of giving somebody the ability to, and it doesn't protect our chemists either, because we have customers and they're like, why are you spending so much time on something where we're going to measure 1,000% change? It's going to be no big deal. And again, question to you, can we build something into the document to make sure this doesn't happen?
So Mark, a quick question for you. We talk about system suitability criteria, right? That every time an assay is run, it has to reproducibly work a certain way. So your point is somebody cannot set that system suitability criteria to be plus or minus 50% just because they have a big signal in the biomarker, right? Is that the point you're trying to make?
Yeah, that's exactly it. And I didn't see that anywhere in the document, which I think is, again, a very good document, but I didn't see this kind of, but I've seen people kind of.
I think it didn't cross our mind that somebody would do that. Just on purpose, not develop the method well enough. I'm just kidding. It's a good point. So how can we address that?
Can I address that, Steve? Sure. I would argue that perhaps the stats that you put up, the CVI, CVG, the two nested ANOVA, is defining precision and bias standards to address fold changes. The two groups of disease and healthy, and the numbers you put up from Fraser in 97, if it's not been interpreted that way, that was the intent, to get to how good would it need to be when fold changes are enormous or tiny, at least in a provisional manner. So if it wasn't interpreted that way, that was certainly the intent of that section in the assay specs, Mark.
So I agree with that.
I agree with you, actually, but I think it should be more defined in the document. So that's kind of my point.
Tighten the language, because you cannot use context of use as an excuse to have an assay that doesn't meet the criteria. I understand what you're saying.
It is implicit from the context of use. It is not specifically stated in the classical sense of assay in control, out of control, according to Levy-Jennings, like you might think. But if it is in the context of use, then that has to follow backwards, that the assay is in control. So again, it comes from there. Russ is right in the determination of error. We have to understand what we define to be in control for this particular context of use. We can emphasize that, but again, that comes back to understanding the clinical implications of the COU and working backwards. So how much control do you have to need? If you are doing a singleton measurement at one point, do you need year-to-year reproducibility or inter-laboratory variability studies? And that will determine whether or not you're in control. So we can expand on that, but that's what we're here for to discuss. If that's a designated need, we will certainly do that.
I think Shavri has a point to add.
So, as I said at the beginning, I'm pretty concrete. So, would you be willing to give us an edit of what you think that additional language should look like and maybe send it to Steve so that we can incorporate exactly, so it won't be a guess what I'm thinking exercise, but it gives you an opportunity to add that additional language, because you make an excellent point, but could you send it to Steve?
Yes, I could.
Okay, excellent. Thank you.
I would enjoy that.
Awesome.
And we know him, so we can chase after him too.
Okay.
So I think Shavri brings up a good point. This is a document that we want to be broadly adopted. So please feel free to send your commentary and the specific language that you think will enhance what's already there on paper. We appreciate that. Lauren?
Lauren Stevenson from Biogen. I guess I just wanted to understand from the regulators, given Mark's concern about how frequently we see this kind of behavior. Like, what is the likelihood that this is a problem? Mark, in my 10 years of biomarkers, I've never met a scientist who is like, oh, it doesn't need to be good assays, we'll make a crappy assay.
Well, no, see, you guys are from organizations.
Best assays we can, regardless. You bring your method to its best state. Just because I only need to measure a 200% change doesn't mean I say, I'm going to develop to 200% and then I'm done. Good, throw that one, and we'll file that. So I really just want to understand from the regulators, because I certainly have no opposition to there being a concrete statement in the document, but it'd be interesting to hear from them, like what is the frequency with which you see this kind of concern that people have kind of just said, we're just not making our best effort because it's good enough.
Shashi?
Yeah, thanks. We haven't seen that kind of a variation, thankfully. But it doesn't mean we'll never see it, so I think the language would be useful. But we have seen up to, say, 30% and, or 40% and a justification, and sometimes it's justifiable, sometimes sometimes it's justifiable, sometimes it's not. So then we talk to them, make sure that all the issues are addressed, such as pre-analytical considerations, etc. So, we try to help them in that space.
But I do want to say that we have heard from others in the regulatory teams that if it's a smaller outfit or if it's a company with very few scientists with less experience, they do struggle with how to improve what they have. So they just come with what they have, right? But then the guidance from the agencies in terms of how to improve the methods is always there. So it's a two-way street.
My experience is limited to biomarker qualification efforts, so I can't speak for the entire drug development scenario.
But it's a good thing to keep in
Disclaimer
mind.
here.
Yeah.
Yeah, so these issues play out over time to a degree, and sometimes it's a question of the availability or the invention of new methodologies that might give you a better assay than what you had at the start off.
Yes.
So that you have always the question of whether to migrate to that perhaps more expensive but better way of doing it. At the same time, there are changes that happen with respect to cost and with respect to conceivably the context of use that you're going to be able to apply the assay to that have to be kept under control. So I think what Steve was describing there is that for the context of use, a well-defined context of use, making sure that the assay that you're dealing with is good enough to support that. That's the key point for it. Realizing that there's always some debate as to what is good enough.
And I just wanted to add, I'm Kylie Haskins on the Biomarker Qualification Program. One component we didn't talk about in the paper is the conditions for qualified use, which is very important because oftentimes when we look at assays and the level of control, it's for very specific uses that aren't always defined in the use statement, but we have an additional document that provides limitations of how this would explicitly be used in drug development. And I think this point is very important because within the analytical validation expertise that we have in the room, it's more understood, but in the broader drug development community, those nuances aren't as well understood. So I think just providing education and maybe including some statements in the white paper is very important.
Hi, Devangi Mehta from Biogen. So I would like to actually hear the panel discuss some specific points in the white paper, particularly around Table 1, where there's, you had that up earlier in the slide, Mina, where you're discussing both exploratory, partial validation, and full validation. Now, everything I've been hearing today is really about having an appropriately validated assay for biomarker qualification. So this is a late-stage biomarker where we can carry this biomarker across multiple programs. So I want to understand the value of discussing the exploratory and partial, because as long as we all understand that those principles are ultimately need to be addressed, can we not leave it up to the scientific community to determine what's needed for their exploratory biomarker assays, and instead of prescribing that in this? Because I think we run into the situation where the exploratory that's listed may not be enough, even in an exploratory fashion, to really get you to that next stage of validation. So perhaps just focus on what's really, truly needed at that biomarker qualification level. So I'd like to hear your thoughts on that.
A lot of these have to be decided on a case-by-case basis for that particular biomarker, for that category of biomarker, for the context of use proposed, etc. I think the table was just to give a general idea of what you might need, and it's not prescriptive. Steve, correct me if I'm off the mark, but that's what it was meant to be. And that's why the case studies might be helpful too, that you'll probably hear tomorrow. So.
Yeah. And I would just say, as part of our case study with the kidney safety biomarkers, we used exploratory assays when we had enlisted over 20 biomarkers to narrow down. And we looked very carefully at their validation characteristics when we prioritized the biomarkers that we wanted to take further into the qualification process. So that was a very critical foundation for a qualification effort. So I think that continuum that Jean mentioned earlier, is important to keep in mind, because, you start usually with a fairly large body of evidence before you want us to invest in the qualification effort.
You have to realize that in the so-called exploratory, it is actually a very wide spectrum, from even almost no requirement in the preclinical animal tests to the increase of rigor.
So I
And
guess my
then
question is
finally to the qualification.
So I guess my
purpose.
question is more about what is the scope of the document? So everything I've been hearing about is the scope is at this biomarker qualification process, which requires both analytical and clinical validation, right?
That's correct.
And so I guess I don't want people to be swayed by, what's recommended in the exploratory setting, because I think, like you said, it varies widely. And so do we want to be more aligned on what it is required for that full analytical validation, as opposed to worrying about what's happening upstream of that, which will require a dialogue, ongoing dialogue between the scientists and the regulatory bodies?
Right. Our intent was just to highlight what the journey may look like. So that's why, and remember the context is we are hoping this white paper is going to be broadly used by people who are well versed in the art of biomarkers and the ones who are novices who are entering the space. So it's just to give a preview of what something may look like, what characteristics you may want to consider before you rank order your biomarkers of interest. But if it does create confusion, we'd love to hear from folks, because we don't want it to give mixed messages. That's not the idea. And it's not, it cannot be comprehensive either, right? It can only provide you, really an outline of what should you be considering. Really, it's rightly called points for consideration. So,
I agree, and that's exactly what it's meant for, analytical considerations. And then the case studies hopefully will help you understand how it can be applied in two different scenarios, and then we can probably have more discussion at that time about other cases we have encountered.
So just one more comment on that. I think one of the things we're struggling with is we want to provide that additional information for the less experienced scientists out there. But sort of circling back to what Mark said and what Devangi alluded to, by putting any specifics in there in the pre-stage we're really talking about, then you run the risk that less experienced people in the exploratory phase are going to go, check, I did those 3 things they listed in that column, when it's not appropriate for the use that they are doing their exploration for. So I think it's just a cautionary, think about what you put in there and how it's going to be interpreted with from people with different levels of experience. Some people are going to say, but I don't need to do that, and now am I going to have to explain why I don't need to do that in my exploratory phase? But then on the other end of the spectrum, people checking 3 boxes and saying, well, I did what I had to do, and then they could end up making a spend and an investment going down a non-viable path as a result.
So adding a note under the table would be what you're suggesting.
I think a neon sign over the table.
Thank you.
So there's another
Hi.
question. Yep.
Lisa McShane. I'm a statistician at the National Cancer Institute, and I wanted to just make a few pesky statistician comments.
Please.
We've been talking about exploratory versus, later in the process of qualifying. We've been talking about different sources of variability in the sense of different technical aspects or biological variability. One thing that always gives statisticians heartburn is looking at some of these analytical performance studies and looking at the number of samples that are used to estimate these all-important elements of variability or the magnitude of these variances. And so I think we need to keep that in mind as well when we talk about exploratory, that we say, well, all right, in exploratory phases, we might be willing to accept an assay that has a fair amount of variability, maybe a CV of 50% or whatever. But if you actually sat down and calculated the variability of your estimate, it would be huge.
Yep.
So that's another danger that you have to keep in mind, that you may have estimated it at 50%, but the confidence limits around that thing may be enormous. So I think we have to factor that into our development process as well, that we need to accumulate additional data on the analytical performance of the assay as we go.
Please.
So we do have a soft area in the document.
Yes.
And that was when we got to the end of the pre-validation criteria. We got to the power functions and the ends to confidently assign change. So we have some room for somebody to add to the document, don't we?
We do. We should. I think
We
these
got-
are good points that are being brought up.
We sort of drew a line there, but we could perhaps expand that further at comments well received.
I'll have to look back at that particular figure, but usually the way we do those things, when we do sample size calculations, for example, we take an estimate and we plug it in as though that's the truth. So in fact, if there's a power estimate that depends on your variance estimates, you ought to be inflating those variance estimates that you're plugging into that power calculation to really understand how well you have characterized the situation.
Following up on Shore's lead, Lisa, can I ask you to send us a comment so that, we can consider that to be added to the document? Really thankful to you.
Just a quick comment. By the way, as you have seen, there's a trend. If you make a comment and a suggestion, they're going to ask you to
Action item for the...
Please let that not deter anybody.
And then we're going to discuss it for another 2 months. But in any case, no. So Steve going to make the final call. But I just want to make sure that I think there was a point that was raised up, which is nice. I want to make sure we are all in agreement. So, and we have the same understanding. The way it works out here is that for the parameters that we want to test, right, what we have included in the paper, and we're going to talk about the acceptance criteria, we're putting sort of an anchor point for biomarker qualification, clinical biomarker qualification. The whole slew of biomarkers that we work in at the preclinical stage, some just for fun, some for exploratory, some like to use 20 and 50,000 plexes out there. But at some point, these things trickle down. What we're trying to do here today and tomorrow is to set this anchor point and make sure we get your comment and hopefully consensus as what is that going to look like, right? Now, in terms of, early on, there are companies that are going to still take that kit exploratory and they're going to use the commercial, controls, and they're going to run that once, and they're going to say, I'm ready to go and not test some samples. And there are companies that early on, they're going to fully validate that almost to the standard of what we're talking about. That's a company decision, sort of, right? But we're trying here today, and correct me if I'm wrong again, any of the panels, we're trying to say clinical biomarker qualification. What is the core? What is the minimum that we need to establish, okay?
Right.
In order for us to be on the same page, at least know what the expectation is and what to work toward.
Yes, so I want to say I want to agree that this is the gold standard, and if we think back to the first FDA guidance on bioanalytical method validation, people picked up that document, and it was a revelation to a lot of people. I have to do this, I have to do that, and I would like the ability for people. I don't want to get rid of the early stage expectations. I'd like to see this evolution, and I'd like the implication that some of these things could actually be done very early, because we make $1 million decisions on some of these early stage biomarker studies. And if somebody really wants to do a good job of it, I want them to be able to look at this document and say, okay, here's what I need to do. Here's what scientifically they think makes a good biomarker assay. And all you people sitting in this room, you're way up here, and there's people working in a lab that are they're low skill level that are going to look to this document. So just don't forget that, and even early stage.
Chris?
So certainly the comments of late, especially around driving home the difference between use in early phase studies and late phase studies, because ultimately the context of use is in a drug development program. Yes, the context of use is around the biomarker, but it's how the biomarker is used in drug development. So I completely agree we need to have the stake in the stand, but that stake would be a minimum based on the easiest context of use, and then could be expanded from there. So to the extent that this group could come to consensus of what that lowest bar is around all the analytics, what is the lowest bar, and then what would you add based on the benefit and risk of the context of use?
Yeah. Yes, Frank?
I was similarly confused in Table 1, Table 9, and Table 10. What is the message that the authors were sending here? One message is there's variability, and there's a progression. One message from the authors is that there is a continuum from early stages to a full validation. We understand that. And then Table 9 says there's a lot of different options out there. They look at all these different guidances that show all these different approaches. And then you go to Table 10 and say, this is how a 410K is done. And so we're like, okay, is that what we have to do for biomarker qualification? I'm a little confused as a reader. What is the expectation? Is there like 600 examples or whatever the number is in that table 10? Because that's the last table we see.
So if we can reserve this question for the next session, because Afshin is going to be addressing some of that in the conversation around validation. So we'll tell you why and what is the utility of table 9 and 10.
So we're very much not saying that that is not appropriate for a biomarker qualification. That is by far the upper ceiling
Right.
of what it would take. Because at
The
that
high
point,
end.
it's a diagnostic.
So we just want to give you the flavor from easy to the toughest bar. So any other questions from the floor?
So I just had another comment. So you put up, a biomarker qualification will not equate to a clinical diagnostic. But I think we also should have the discussion that a clinical diagnostic may not be useful for a biomarker that we're qualifying, right? And that clinical diagnostic may not be fit to qualify a biomarker, depending on the context of use. So it works both ways.
Absolutely. Fair point.
That is stated in the document. Even if the correlation is not correlated, that is stated.
Okay. Yes, it may be the final question, or we can go to one more question from our question bank.
Sorry. I would like to ask one question specifically for the table where you have exploratory, middle, and then full validation, because at the lowest point there is semi-quantitative and qualitatively quantified assays mentioned for exploratory. And actually, I circulated this document within my group, and the understanding was actually that semi-quantitative and qualitative assays requiring exploratory validation requirements, and so it is by assay divided. So I just wanted to make this as a comment that may be a little bit misleading to have then the assays mentioned.
I see. So they interpreted it as only semi-quantitative and qualitative assays can go through an exploratory validation.
Exactly.
I see. So EG was not seen. Okay, for example. God, it's a good point. I think there's somebody who's taking notes, so these are things that we need to really address in the guidance. Thank you.
I have a question for the audience. Was the table useful to you? I mean, the whole purpose we had was to give a framework and give a relative idea. So you think the table should be there, but we should have the disclaimers or, explanations.
Hey, one person gives 2 thumbs up, just so that if you can't see it.
Yeah.
Okay. I wanted to bring up table 1 as well, but there were enough voices, so I didn't bring it up. And we will have a brief discussion on that tomorrow. I think it is useful. I guess that's my answer. However, as I think it's fair to say, and let's open up the conversation, based on my experience over many years now, what I've been hearing within the industry is there are 2 schools of thought. There are people who are involved in biomarker assay development validation who really would prefer a prescriptive guidance on exactly what criteria to use. And I go in the lab and I pick up these reagents and develop these markers. There is another group of people who appreciate the context of use and rather have this whole leave it all open, where you might not even, and that's where you're hearing the concern when you see exploratory and you lay out the specifics. Do I have to stick to this? So that's what they're asking. So we have to find a middle ground, and I think you guys have done a great job. I have to say this is not a trivial task to really satisfy all parties involved to clearly understand this. But I do think it's a balanced message to go along with the table based on the discussion that's happening here. I think that would be very useful, where it could be very useful to have these specific guidance, but at the same time, don't use it as it is. You've said it already, I know it, but I think we have to say it more clearly as well in the document.
And just remember, this is a white paper, not a guidance.
Right.
Thank you. Thank you.
But we shouldn't forget what Shavri said, which is what are the next steps, right? And the question is, will this white paper evolve into a guidance? So if individuals can really submit statements which will clarify the intent, because now we told you what our intent is. We will appreciate it because we can then take into consideration what language resonates with the majority of you in terms of, adding the disclaimer or a clarification at the to the table.
Right. I mean, I think that is. So, I mean, some of us have heard this, right? So this would form the basis for the future guidance. It's certainly something many of us have heard about already. And secondly, of course, guidance is also a guidance and not a requirement, right?
Absolutely.
So,
absolutely.
Right. It's all the level of scrutiny that people pay.
Degrees.
So thank you all so much for your active participation. So now we'll leave the floor to Afshin to start talking about, is it? No, it's a break. It's a break, coffee break, okay? And really, thank you for the participation. Okay, we have 15 minutes for the coffee break, so please be back in time. Thank you.
Okay, yes.
And I called you also
Yes.
once for.
I know.
So,
Yeah,
okay.
that's okay. Good. How are you?
I'm good, and you?
Good. Yes. Yeah. Keeping busy. Tired.
Yeah, you're traveling a lot.
Absolutely. Yeah.
You
Yeah.
are at all conferences.
It's
right?
crazy. Yes. But this is a very important one, so
Yeah.
I have to beat this one. So yeah.
Test, test, test. 1, 2, 2, 2.
Folks that are looking at it with the webinar
From
from Europe or.
To be honest, it's been difficult to find out. I think there's a lot of people that actually wanted to come but haven't. I know I even got a message a couple of days ago from someone, and I haven't had a reply back yet because I asked him, if
Test,
he's
test,
coming
test.
along.
1,
I don't think he is.
2, 2, 2, 2.
Other people in Europe that are going to be missing.
Test, test, test. 1, 2.
They've got it on a live webcast, so a lot of them will be just watching that. Yeah. So it'll be interesting to see if any of the questions coming in come from Europe. I'm sure there will be some. Right, I'm just going to try and get a coffee. Yeah, good to see you.
Should we wait until lunch?
How's that? 1, 2. Test, test, test. 1, 2. 2, 2, 2. Test.
Okay, we're going to go ahead and start the next session. If I could ask everybody to take your seats. Is this even working? Hello? Oh, is it working? Okay. It looks, it's red. Okay. Okay, we're going to go ahead and start the next session. If I could ask you all to take your seats. You can't hear it. It's not on. It's not, is it on? Can you grab that, Mike?
It's not on.
Hello? Oh, hi. We're going to go ahead and start the next session. If I could ask you all to please take your seats. Thank you. Hello? Okay, that's right. Is that you want to, Liz? Okay, welcome back, everybody. Welcome back to this next session. Yeah. Liz. Yeah. Yeah, okay. Everybody, welcome back. We're going to go ahead and start the next session. If I could ask you, please take your seats. Welcome back, everybody. There's a very lively and active crowd, so this is going to be challenging to get everybody to go ahead and take your seats. We're going to move on to the next session where we'll continue exploring the key components of the white paper. Afshin Zavavi is founder and global chief scientific officer at Bioagilitics Labs and we'll kick off this next session with a presentation focusing on assay performance and assay validation acceptance criteria. Before we go ahead and move into that session, I'd like to remind everybody in the room and those of you who are joining us via the web that we have an email address up on the slides now, which is margolisevents@duke.edu. That's the email address that you'll use for any edits, comments, feedback on the white paper and the framework that you have access to. I'll go ahead and turn things over to Afshin, and after his presentation, I'll go ahead and moderate a discussion among the panelists and audience on these framework components. One note that I do want to ask everyone that's on the panel is to go ahead and speak very closely into the microphone. I think we had some challenges in the last session hearing everybody, and we want to make sure that we can hear all of you. So thanks, Ashton.
Great. Good morning, everybody. Again, it's a pleasure to be standing in front of you, and my team has volunteered me to talk to you about the most controversial part of this document, perhaps. We checked for Rotten Tomatoes before everybody got here. Just to let you guys know also, tremendous effort has gone into getting this paper together, at least, for us to get it started. And I hope that every one of us is committed that before we leave tomorrow, get in the plane, what is that, 3:30, 4 o'clock, 5, whenever we're leaving, that we have come to some level of consensus. Because if we don't, we're going to be sitting here a year from now and having another philosophical discussion, and I think we want to avoid that. And if we can go ahead and anchor, the flag somewhere, to move all the discussion that we've had for past several years to the next level, that would be amazing. Now, another item to just share with you: the same number of people, approximately 100 plus people that are actually same number as the number of people that we have in the room. They're actually calling in by webinar, WebEx. So they're listening, and they're from all over Europe, all over US, and all over Japan, and probably other countries that you guys probably can tell me. So this is, and we're going to be getting their questions through email as well, and we're going to discuss those. Just a couple points. My apologies that some of the slides that I show you, there's a lot of verbiage in it. I really wanted to stick with the content of the paper, so I used the same words that was used. It's a little bit, again, maybe the slides are a little bit boring. I'm going to try to make it a little bit more exciting and get your involvement. The second thing, the reason, again, there's so much verbiage, because before my CEO said, is FDA going to be in the room? I said yes. They said, behave with those slides. So I'm going to behave and stick with the content of the paper. Okay, so after the session, we're going to have actually a group of panelists here that are much smarter than me to help me answer your questions and get the discussion going. We have Steve, Robert, and Kylie, and I do actually want to go ahead and definitely personally thank Dr. Jean Lee, who with Steve's help and some of the other panels, folks' help, we actually pulled her out of her semi-retirement to come back and work with us. So again, thank you very much, Jean, for your contribution. Okay. So in terms of the, setting up the validation acceptance criteria, I think this is the most challenging exercise we're going to go through. Okay. And if I can get consensus on most of these, we're going to be high-fiving by the end of tomorrow. We know that unlike the predefined acceptance criteria established for small and large molecule PK assays, the acceptance criteria for biomarker assays are dependent upon each biomarker physiological behavior. So let me just let you know, anybody who's looking for a cookbook, a complete recipe for biomarker work, you're out of luck. We're not going to be able to ever provide you that. I can provide you 9 examples. That's how we should do it, and that's what we should set in stone. And you're going to find somebody in the audience who's going to come up with another example that's going to discredit, what was proposed. So we're trying to come up with at least the core of what is needed. I think, that was already brought up around the most difficult question around this validation is that appropriate validation samples. You're going to look at a biomarker validation that you want to that maybe all you have is just there's no blank. You find all these endogenous samples with very, very high level. On the other hand, you basically are looking for endogenous level because you don't want to use the recombinant protein as a reference material. And guess what? All you find is nothing, 0, basal level. So it is again, there are a lot of challenges, and a lot of our challenge comes from just a lack of reference material or the proper sample sometimes to do the validation. If we had those, guess what? We would have been in a PK world, and it would have been a little bit more structured. In terms of our goal, we have spent countless hours within the team, and there have been some what I say, argument in a good way, as in terms of what should we propose? How much detail should we put in this acceptance criteria? If we put too much, then we're tying people's hand. If we put too little, well, does it give enough structure? And that's what we're looking for your feedback. So again, the main papers, there are a lot of good papers out there, white papers, the ones that we have really used, try to anchor these core parameters that we want to go and set up some acceptance criteria around, are coming from the 2 guidances from FDA 2001 and 2013, the Crystal City 5 and 6. I think they were really good discussion, especially Crystal City 6. At least we came to agreement. PK and biomarkers are not the same. That was nice. And we also talked about relative accuracy. That was even nicer at the last Crystal City. And we started hearing about the CLSI guidelines. Those of us in the BMV world, we were, had set of guidances that, or not guidances, set of, white papers and how we learn and we grow up, and then we hear about the CLSI guidelines, and we actually looked at those, and you're going to see some influence of what we have put in the white paper when it's appropriate from the CLSI documents. Again, and we hope that all of this is going to provoke a good discussion. I think by now we know that the fit for purpose status of the biomarker method is acceptable if the assay is capable of discriminating changes that are statistically significant. Okay, can we see the difference? Can we see the pre versus post dose? Can we see the normal versus disease? Again, for whatever indication that you're looking for, can you see the change that you're looking for? Example, I also want to stress this. There are situations that we might allow actually a 40% total allowable error, right, for an assay, and that might be good enough. There are instances that you can actually have maybe a 10% total allowable error, and that's not going to be good enough for intended use. So the context of use becomes very important, and we're going to talk about that in more detail. Okay. The slides are not changing. Okay, I'll push it 3 times. Let's see what happens now. So let me just. This is something that I think it was sort of brought in the last hour, right? This journey of the biomarker validation, exploratory, moving toward biomarker qualification. Most of the people that I know in this industry, in their company, they look at a continuous process for biomarker validation, okay? It is really for the intended use, the context of use, although they don't necessarily say it's a context of use. And you're all familiar with the fit for purpose, right? So there are, again, individuals that they do minimal, looking at few parameters. They set up some loose acceptance criteria on these parameters at the early exploratory stage. And then they move on, and eventually, at some point, this biomarker may go toward becoming, or decision is made to go toward a clinical biomarker qualification, okay? What drives a lot of this decision early on as what to do? It is really the risk benefit. And the risk benefit varies from company practices. I think I mentioned earlier that there are companies, major pharmas, that early on for exploratory biomarkers, they basically take the kit as is. They request you not to make quality controls in biologicals because it costs them extra, and take it and run it as is, the kit as a component. And then there are companies that they actually go to the extent that during phase 1, actually, they make every component and they own every component of what goes into their biomarker validation. And they do the full-blown biomarker validation, every parameter that you can imagine, right, at phase 1. So that's a company practice, company culture. But the one thing that I want to share with you here is, and I got that from my friend Kylie at Cedar, the assay should be established and analytically validated as soon as possible in the biomarker qualification process. At a minimum, this should be completed prior to submitting the qualification plan to ensure the assay's acceptability prior to its use in the confirmatory clinical study. At some point, when you want to go to the FDA now, and you want to go ahead and say this is a biomarker qualification, you got to have your assay nailed down, and it got to be fully validated, okay? Now, when is an assay considered validated? We actually had a meeting, I think it was in November here, and with the folks involved in writing the paper, and we sat around and we went, what are parameters we want to include? All these publications, the white papers. Steve mentioned again the GCCs, the EBF, the AAPS. I mean, there's so much good documents out there. But at the end, we decided that there's going to be a core set of parameters that these need to be validated analytically in order to be able to perform biomarker qualification in the clinic. And these are relative accuracy, reproducibility, analytical measurement range, parallelism, specificity, selectivity, and stability. Now, we already talked about relative accuracy. We know why we call it relative accuracy because the reality is 99% plus of the biomarkers that we work with, we have to use a recombinant protein in order to be able to quantify the samples. We don't know if that recombinant protein is 100% identical to the endogenous molecule that we're measuring. And that's why we call it relative accuracy. And that's something that I think all of us, we need to do a better job sometime internally, make sure our users internally are aware of that as well. In terms of the accuracy can be expressed as percent recovery or bias, established over 6 run. And again, the formula that you see out there is what we're using to calculate accuracy. Sorry, there's a delay from the time I click by the time it... Now, in terms of the analytical measurement range, the analytical measurement range is the range of the analyte values that a method can directly measure on the specimen without any dilution, concentration, or any pretreatment. AMR validation is the process of confirming that the assay system will correctly recover the concentration or activity of the analyte over the AMR. So for the assays that can test a specimen without dilution, it is LLOQ up to the ULOQ multiplied by the maximum validated dilution. Is the microphone on? In terms of the, for the assay that require a specimen dilution prior to analysis, it is the LLOQ times minimum required dilution up to the ULOQ times maximum validated dilution. Some of you are going to be looking at this and say, I am used to the LLOQ, ULOQ, LODs. What's going on with this AMR terminology? Don't freak out. Don't be scared. It is almost the same, except what this is doing is telling you that you got to take into account your minimum required dilution and the maximum validated dilution that you also do. We already do that, most of us. We just got to go in and, again, if you want to classify it under this AMR. In terms of the analytical measurement, again, the key that was brought up earlier, is this going to be a qualitative or quantitative? Based on that, you're going to make a decision how much emphasis you're going to put on your LOD. Most of us, we actually do measure our LLOQ and ULOQ, and we know we do that by spike recovery. Hopefully, if you can find a blank sample, right? But again, the LOD, it is the response signal plus 3 STs, and minimum number of blank sample to use is 10 plus. Now, let's talk about the analytical measurement range and whole idea of how do we go about setting up acceptance criteria. So we know that with the biomarkers, both the assay and the biomarkers' intrinsic physiological behavior are the primary source of variability. So what you see here is what we're putting here, what we're outlining over the next 2 or 3 slides, and you will see it in a paper. It is the minimum performance standard is what we're introducing here. So the performance standard is defined by the total allowable error for the biomarker at the decision level. Now, the analytical measurement range is, so the total error is sum of the systematic bias and variance. And the bias is so total error is a bias plus the intermediate precision that contributes. And so the performance is acceptable when the observed analytical total error is less than the performance standard. And the performance is not acceptable when observed analytical total error is greater than the performance standard. So let me just tell you what this means. In simple language, what it means is that biomarkers with a high degree of biological variability plus lower amplitude of response to stimulates require an assay with relatively low total error, and vice versa, right? If you have a lot of variability in your population, you better come up with an assay that is tighter, right? However you want to define tighter, and we talk about it. Then that way you can actually the assay going to be useful. I think I broke the projector. Let me see. Tell you what, I will ask you guys to advance from now on. So with the parallelism, they have been actually, I want to point your, make sure you guys, there are 2 white papers that have been published in the past 12 months, Lindsay King and Laura Stevenson. Those are great white papers for parallelism studies. CLSI guidelines documents also talk about, parallelism studies. The bottom line here with parallelism, what you're trying to do is that you want to make sure that as you dilute your sample, that there is no bias. You don't get an increase or decrease in signal or in the concentration that you're getting. And the beauty of it here is that, here's one advantage we have over the PK folks, that we actually can go in and buy these samples from vendors or actually get them ourselves. And we don't have to wait to have these samples until the clinical trial is done. For most parts. So no need to wait to get the incurred sample. Next slide, please. In terms of evaluation, I will throw several items at you, and I expect that there will be some good discussion around this. For us, we believe in a tier approach of evaluation of parallelism, right? The best thing is that if you can find some samples at the endogenous samples at high level, and you can use those. In the event that you cannot find the large number of or enough endogenous samples, you're sometimes going to end up actually just get some blank matrix and spike it with the endogenous samples. Is it true parallelism? No. Is it the best you can do? Yeah, maybe you can combine that with a couple samples that you can get your hand on with a high endogenous level. But at some point, remember, we're going to come back to this theme. Validation is a continuous process here. So once you run your phase 1, at that point or phase 2, at some point you will get your hands on some samples with a high endogenous level. So you can come back as an amendment and add additional parallelism studies to your study. One of the typical ways that we do that is that you screen the samples, and then you take minimum of maybe 4 samples. In the industry, you see anywhere from 2 to 10. So somewhere about minimum 4 samples that you do 1.5 to 2-fold serial dilution, and you use that to establish your parallelism. Another method that is I think is more from a CLSI document is a 5-point admixing procedure, which allows you to actually look at it and look at both imprecision and bias evaluation. Next slide. So in terms of assessment, there is no consensus in the industry as how you would go and assess parallelism. One way, again, the one at least that I've seen mostly is that you look at the mean of the diluted corrected concentration, and you have certain CV range that you have set up for that as acceptable. And those CV range that you normally see is that for 15% for LCMS, 20 to 25% for hybrid method, and 25 to 30% for LBA assays. But I want to stress this, that depending, again, the context of use, the acceptance criteria that you set up for your study, it can vary. A lot of time we see people actually get dragged into, they have a beautiful assay, and it's a pretty tight assay, but they don't have parallelism. Maybe in order to, maybe the parallelism CV expand to 40%. Does it mean the assay is not useful? So again, this is what we're trying to do. What we see as an industry use or standard, but remember, you have the COU to drive your fit for purpose and eventually your acceptance criteria. Next slide, please. So, how do you go ahead and visually show parallelism in your reports or for your data analysis? These are three schematic drawing that we've seen out there. One is the observed concentration versus 1 over dilution factor on a log scale, which you're looking for a slope of 1. That's the first example you see. The second example, that's the one that I personally use in the lab, is that you do a dilution-corrected concentration, and you apply that over a log scale of 1 over dilution factor, and you look at for a slope of 0. And I think we can understand why these experiments are called parallelism. And then the third one is the average bias per dilution level versus the dilution-corrected result for each sample, and you look for a slope of 0. So next slide, please. Now, let's talk about the minimum required dilution and Prozone effect. The data that you collect from your parallelism studies, you're going to set up, look at it, and you're going to set up your minimum required dilution. The example, sorry, I don't have a pointer here, but if you look at the line that I've drawn here, is that the minimum required dilution that you use, let's say you were setting up the percent recovery for your assay at plus or minus 20%. That's what your assay required, right? So this means you got to do a 20-fold dilution. But in the same example, the hypothetical example that I'm showing here is that, let's say your method can actually go in and withstand, a 30 plus or minus, so 70 to 130% recovery, right? Because the sensitivity is there. You don't need to worry about that. Your LLOQ, ULOQ, everything looks good. So in that case, you can actually get by 1 to 10 dilution. So those are the decisions. We do not want to tie your hands to say how to go about, setting up some of these acceptance criteria. A lot of that, again, going to go back to context of you fit for purpose. In terms of the Prozone effect, this is when you get a falsely low result on a sample with a high concentration. This is very similar to actually what you do for a PK study, that you take the sample, you spike it at 100x above your ULOQ, and then you do the dilution to bring it back to your ULOQ and see if you recover properly. Next slide, please. In terms of reproducibility, again, we're talking about relative accuracy, right? So it's but again, what you want to do in this case, the best thing is that if you use patient samples to establish your reproducibility, right? Within run and between run. Now, depending on your use, you may want to go ahead and run it in multiple labs, or you might want to have multiple operators. So depending on how you're going to actually conduct this study, how many labs going to be involved, how many operators going to be involved, that's what you need to establish in terms of for the reproducibility. Next slide, please. For selectivity, this is where, our recommendation is you take 10 normal individuals and 10 disease when you can get your hands on the disease samples, and you spike them at high and low level, and you look to see what happens to the recovery. There is there's a pre-specified sufficient proportion of the test samples that should be found acceptable in terms of the total error that you have set up. Although it may not be needed at the early stages, I want to stress that at the early stages of your biomarker validation, at the exploratory stage, to look at the hemolysis sample or lipemic samples. But as your drug development proceeds, and by the time you get to the validation part for your biomarker qualification, clinical biomarker qualification, you got to go ahead and actually have tested the hemolysis and lipemic samples and documented the effect of those samples, type of samples on your recovery. Next slide, please. In terms of specificity, again, for a small molecule, when you're doing the analysis on LCMS, you look at physiochemical characterization, you can establish specificity very nicely for small molecule. For large molecule, we will never, ever be able to establish specificity completely unless we really thoroughly, and actually I take that unless away, because we never know about, again, if these guys are actually have other metabolites, isoforms that we don't even know about, right? So, but at least what you can do is that for specificity, you can go in and use compendial methods such as LCMS to characterize and make sure you feel comfortable with the level of specificity that exists. Next slide. In terms of stability, in an ideal world, we like our time zero for stability is when you take a patient, you poke them with a needle, you take the sample, and you analyze it right away. In the world of the drug development, that is extremely difficult, right? So this is something that we had some discussion within the team, and I think we came to agreement that what we're looking at is really the trend of degradation. Our time one, time zero in essence, is when we have taken that sample, it was sitting in a freezer for a period of time, and then we use that to establish our baseline for stability. So if there was an initial degradation, some issues, we will not know that. But again, that's a limitation, and we just got to be aware of that. Next slide, please. So that's everything that I wanted to share with you very quickly. I'm about to put my helmet on. But no, in all seriousness, I think, again, with the acceptance criteria, that is very important. That's very critical. That's where there's a lot of confusion. We want to go ahead and, again, provide some guidance, some directions, some core, maybe industry standard numbers to stick with. But at the same time, we cannot tie the scientist's hand. This is biomarker world. It's very different than the world of having a fully characterized reference standard with a blank matrix. So we just got to keep that in mind as we move forward. With that being said, I'd like to go ahead and actually ask the panelists to come up here. Maybe they're worried to come up here. Excellent. So if I can go ahead and start by asking the panel, I think the new panelists, if they can go ahead and introduce themselves, please.
Sure. I'm Kylie Haskins. I'm a biologist in the biomarker qualification program at the FDA.
I'm an old panelist.
I'm old.
See, no matter what you say, you're going to get in trouble. But go ahead.
I don't know how to use this. I'm Steve Gutman. I'm a regulatory scientist working for Illumina.
Excellent. So please go ahead.
Okay, my name is Judy Greengard, and I'm from Sangamo Therapeutics. I have a question about a sort of biomarker, which, for example, is measurement of antibodies that have arisen to a therapeutic. And in some cases, there are pre-existing antibodies. What is the definition of a negative matrix? How do you know when you're trying to establish a spiking of a negative matrix that the matrix that you're looking at is truly negative for that analyte?
Panelist?
You test it first and see what background activity you see.
Okay, well, you've looked at your background activity. You see a range of response in your assay. Is that a low positive or is that a true negative?
That's a problem.
Jean, you want to?
What?
Maybe Jean can elaborate a little bit more on that.
Well, if your assay's jumping around a lot, I don't know if you have an assay.
Well, it's not jumping around. For an individual, you get the same result every time, but does that represent an individual with a low level of for example, that antibody, or does it represent an individual who has none?
Yeah, to clarify the question, I think what we're getting at, for example, insulin. There is a certain population that they have actually antibody against insulin. So the question, if we're measuring insulin, we might be measuring, 70% of the free insulin. The other 30% might be bound.
Yeah. Exactly.
And so go ahead, Jean, please.
There are two things you may want to do. Number one, do you have an antibody assay for that?
Well, if what you're measuring is antibody and of course it's going to be polyclonal, I'm not sure how you would test that.
No, it's because you are developing that as a drug candidate.
We're actually trying to look at the development of antibodies to the drug candidate. But we need to develop it relative to what a normal level might be.
So
Go ahead,
I think it's worth remembering that no molecule circulating has a label on it saying I'm an antibody to this drug, or what was the circumstance that elicited its presence. So you have populations you can be looking at that can give you an idea as to whether they have an antibody, a signal that's consistent with being not exposed to the drug. Whether that's the same thing as being clinically not susceptible to having a problem with the drug is a different question. But you, in essence, have the ability to look at samples which will try to give you an idea of what the background is that you might expect for the assay yielding a signal, absent having been exposed to the drug. And then you're still left to interpret what it means if you end up with a signal higher than that, whether it means that you had something which was an antibody that's just cross-reacting, or that was elicited by something which is clinically relevant to what your drug's going to do, or whether it's something which is signaling you don't give this drug. All of that is part of the biology of what your drug does and what the pre-exposure, what the history of the population is that's going to be given that drug.
Let me just maybe add one more thing. So from an analytical perspective, I had done one example of this. And what happened is that this individual, they knew that this molecule, there's a good chance there's antibody against it. So we did acid treatment, right? And we see after the acid treatment, does the level go up and basically creates free biomarker. Now, at the same time, when you do that, you're unfolding a lot of other proteins, and you got to make sure that it's not just a background going up, right? So that's a difficult challenge. In my opinion, you will never, ever know, right? Completely 100% if everything is free. But there's a series of experiments that more falls under discovery mode that you run if you suspect that there is some again, the antibody raised against your biomarker of interest. I have seen again cases, and I've managed to fix one of it, okay, by the acid treatment. But that's not something that I do right away. Okay, good.
If I can say, I think that's a tad out of scope because we are looking at qualifying biomarkers for drug development purposes. Even though a lot of what you are actually asking is pertinent to a biomarker with an in vivo concentration. How do you know what you have blank matrix? The antibody problem is not being qualified as a biomarker, so I'd rather stay away from that as an example of this. Yeah.
Yep. Yeah. Thank you, Steve. Steve's job is to make sure that we keep everybody on track on this. I just got burned by Steve, but go ahead, Laura.
I'm looking at Mark.
I think you were next, Laura. I want to make sure we stay in line. So
He's biased. So I just have one simple question related to prozone selectivity, stability, and lipemic and hemolytic samples. So.
You can sit down for a while.
Do you want Mark to go first now?
Go ahead.
No, actually it's two questions. The first, which relates to prozone and selectivity and stability. Tiny clarification, I'm assuming you didn't say it overtly, that when you were talking about stability, you meant the endogenous molecule, not a spiked sample.
Exactly. Thank you for that clarification. What we do want to, and that's why we have her to keep an eye on us. Exactly. The way you want to set up your stability is with your endogenous samples. We always try to, low, mid, high, or low and high. There are instances that unfortunately you do not find a sample at one of those desired level. And what do? Do you just basically go with the stability at one level? Or do you go ahead and say, okay, you know what, it doesn't represent the true sample, but do I go ahead and spike the endogenous with some recombinant? And
no.
So that's for discussion. Yeah, so that's good, right? No, right? But I can tell you that most of the folks that I work with, they say yes. Okay? I'm not saying that I agree or disagree with them. But that's
great if they want to understand stability of recombinant and matrix.
Exactly. No, good point. Good point.
Okay.
Yeah.
I just want to clarify that. The other is around when it comes to selectivity and prozone. I just want to tie it back to your parallelism point, because I think that was very clearly explained how you would do it with the endogenous molecule. We're all in situations where we may not have appropriate samples. If you're stuck and you want to spike your recombinant on top to perform that analysis, that may help you sleep at night, that your critical reagents in a ligand binding assay are seeing the endogenous and the spike material in a similar fashion. But you very clearly stated, but it doesn't replace it, right? So when you have the appropriate samples, you should go back and do it all with endogenous. So I just want to raise that same issue for selectivity. Spiking on top of background and then recovering that spike material tells you about the recovery of spiked recombinant material in matrix.
Yep.
You know, but if you perform parallelism when you have the appropriate samples available at high enough concentrations in multiple individuals, that's going to be a much more robust selectivity assessment.
I know what's about to come. So you describe the BMV version for the crowd that they do it.
Exactly,
I know
yeah,
what's
and
about
that's
to come
why-
from the
that's why
CLSI, right?
particularly-
The admixing?
So admixing.
Particularly said ligand binding assay.
Yeah.
Well,
in
so I'll get you on
large.
ligand binding. So one of the things that we have to appreciate, the whole concept around MRD, prozone, parallelism in a biological context, depending on whether you believe 6,000 years or 5 billion years of biology, we're still competing with biology. So standard addition or calibrated material addition, one has to allow equilibration to occur with biology within the specimen before one does parallelism. So there's an extra study to even prove you've got free and bound status. So it's even more complicated than just spike and recovery and showing that you can parallelly dilute that out. So there's no better solution than either a priori or in study parallelism. And by the way, I loved your paper.
Oh,
that we need to have that as a reference.
No,
Lauren.
so
Great.
we're completely aligned in study, or if you're lucky enough to have high enough concentration samples pre-study to do purely endogenous parallelism. And I would say, and then for prozone, the same holds true, right?
Yep.
If you have a very high concentration sample, and then John Allenson mentioned earlier, when you see that high concentration sample in study, you really have to go back. I mean, that was effectively a prozone assessment that he addressed. And before I take up all the airtime, I just wanted to mention lipemic and hemolytic samples. I would just like to advocate that that should be a scientifically driven decision based on your biomarker, your pathway, what you anticipate your patient population to be, because it's not actually even in the BMV guidance for PK assays, in the current draft guidance for FDA, nor is it in the Japanese guidance, although I appreciate that EMA still asks for it.
I would say, so actually, I think, Ross, one thing would help is that it is in a paper about the admixing and experiment. And again, a lot of folks are not familiar with that. Do you mind actually talking about it for a couple minutes?
So CLSI EP7, which is methodological interferences in measurement, has a whole slew of techniques and approaches to postulate or actually measure and determine bias and or imprecision by perturbation. So for instance, one perturbation would be a pool of normal and then spiking in commercially available human-derived triglyceride solutions, red blood cells or hemolytic material or bilirubin icterus. And we actually draw reference to a kit and a protocol that can be used with a kit. Another way of thinking about that is actually what we call as a 5-point mixing is two extremes. For instance, a calibrator, whatever reference calibrator you have in whatever diluent as one anchor, and then the other end being a sample, be it a high-level disease or a low-level pool. Admix them 1-to-1, and then the target value then is the average, obviously. You measure the two extremes, and we recommend replication. You admix them 1-to-1, measure, obviously the target then is the average, and then you admix the admix with each end. So you have an end, a 50% mix, and then a 75-25, and a 25-75 mix. You end up with 3 points in the middle of two ends, and we use that experiment quite a lot. You can have a lipemic versus a non-lipemic sample, a hemolytic versus a non-hemolytic, an EDTA tube versus a serum separator tube, a calibration protein in matrix versus a low pool or a high pool. And so all you're really getting into is what are the two ends that I want to use as anchor points to determine the perturbation between those two ends. In examples where it's a upregulation, you generally have a low pool and a high calibrator. In examples where it's a downregulation, you generally have a high pool and a low calibrator, which feels a lot like parallelism anyway. So it is somewhat linked to parallelism, but it's a very easy experiment to actually perturb two ends and come up with the bias from the mean and then those fractional points between. So I hope I'm making this clear. 100% of one, 100% of the other, 50-50 mix, 75-25, and 25-75. And those 3 points in the middle are simple mathematical calculation of averages, and then you compute bias or relative accuracy from those averages. Steve, you stood up. Do you want to add anything to EP7? No. It's a beautiful document and very simple to do.
Stay tuned. We'll use it this afternoon. Thank you, Ross. You went over your 2-minute limit, but that's okay. But guys, again, I hope you guys see here. There's some things that were more straightforward, right? How do you go establish the upper and lower limit of quantification, right? But there are some things that there are multiple ways to try to arrive to the same conclusion. And that's what we try to do in this white paper. How you want to go ahead and illustrate your parallelism? How do you want to go ahead and establish some of these? We try to provide choices. If there are other ways to do it, that's why we have this conversation. We want you to speak up, bring it up. And if they're reasonable and we get a consensus, we will include those as well. So there is not only one road to one destination.
Two quick statements to reinforce 2 points of Lauren's questions and comments before. One, if it's not an in vivo sample, it's not sample stability, period. Two, if it's not an in vivo sample, it's not parallelism, period. Those are immutable. You can do other experiments, but call them what they are. Do not call them what they are not.
So I have a question for the panel. I'm Deb Rasmussen from Janssen. 2 terms that are relatively new to me is parallelism and then AMR. I do a lot of diagnostic work, so I do think that IVD kits that do have intended uses that fit with the context of use would be able to be used. But I don't know that they have parallelism or what you might think would equate to parallelism or AMR in analytical validation. If you have a comment on that.
Yeah, well, I have a comment, but we've already discussed it, that we have the same problem. The language in this document is at odds with the language for some components of IVD work. So unless they've changed, Bob will quality control me here, they don't use the term selectivity outside of maybe microbiology where they're looking at selectivity of bugs. They almost ubiquitously use the term specificity, and they don't use the term parallelism. I'd never seen it until I saw this document. They use the term linearity. Maybe that's close to delusional linearity. They use the term commutability, which sort of relates to some of the information. So there's a language gap here, and it seems to me as we proceed forward with the document, we maybe could do a better job of I think it's understandable because it's coming from the context of a different analytical intellectual line, but I think it's a little bit problematic, particularly if the biomarker once qualified is going to be dressed up and decide to go to the ball being actual clinical use.
Jean, would you like to comment on this?
I think quite a few of these terminology are very much drug-based, right? So like selectivity and also parallelism. I don't know where we came from. Help me if the originator of the white paper. But I remember most of these are actually very much PK-based terminology stem from. And since we are talking about drug development tool, it's not actually I mean, we share some of the IVD, but still it is a drug development tool base. And that's how you have to understand.
Well, I'm not suggesting you firebomb the terms, although that might be my first choice. I'm suggesting that you leave the terms and have footnotes or develop things that sort of better understand the juxtaposition of these differences.
Just one quick comment, and we don't need to go any farther than this, but 510K approved kits, a lot of the diagnostic kits that are being used in the arena that we're talking about, at least for me, I see a lot of problems and issues that they were used not for they were validated, they were put together, there's a whole slew of data generated, a lot of time for different intent, and sometimes different matrix, sometimes different range, and they're unfortunately being used sometimes for different purpose. So I just want to stress that that is something that is very critical. You're going to cut the line?
No, Steve, I just want to, I wasn't cutting
Go
well.
ahead.
She was
Go
very
ahead.
generous. Yeah. So, Steve, linearity, there are a lot of approved kits that don't dilute linearly, so they're not linear.
Yeah.
So the parallelism concept is equivalency in matrix versus calibration system, and that is the key analytic parameter that is being explored.
But the term never appears, so there just needs to be a better explanation.
Well, and parallelism as a concept is a concept from the 1970s in terms of, radioimmunoassay and two-site ligand binding.
Yeah.
And Lindsay King's paper does a very nice historical, which again is another paper needed document, does explain that where that came from, because we used to draw it on chart paper, right? So these aren't new terms functionally, they just twist on different words.
Yeah.
So, good. I guess I wanted to take us out of the weeds for a moment and up, because one of the main goals of the workshop is that stake in the stand that you had talked about earlier. What is the minimum level of analytical validation for the lowest level of context of use that can be imagined? And certainly you guys are, a lot of you are from the diagnostics community. I just want to draw attention that when we talk about context of use, we're not talking about the clinical claim for the diagnostic. Those are not the same. There may be overlap, but we're talking about biomarker qualification for use in a drug development program. So looking at your list of 7, we were at 6 during the fall at our scientific roundtable. Yep. If you're looking at a context of use where it's a proof of concept drug program, early phase, an adjunct or an add-on to currently used biomarkers, there is no change in terms of treatment arms and risks to the patient. Right. That would be the lowest level. Would you still look at all 7 of those?
Panels? Yeah.
Well, I would say yes. That from my perspective, what you can argue whether it's 6 or 5 or 7 or 8, but the concepts of analytical validity are mutable, and you can argue with how many samples and what degree of certainty you want to characterize a particular parameter, but you can't. If you're going to duck it, and I think that there's language in the document that suggests this. If you're going to duck something, explain why you're ducking it.
That's right.
Yeah. So this comes back to the risk-benefit calculations associated with how you're going to use the biomarker and the assay for it. A low-level context of use. The question is, what do you mean by low level? You mean in terms of its clinical implication, or in terms of the phase of the drug development program? What kinds of errors are you willing to tolerate in terms of making the wrong decision based upon the test for the biomarker that you're going after? And these things will acquire much more gravity as you start getting into later aspects of drug development, presumably. And if you're going to go low early on, then you just have to be willing to live with the result. If you can mitigate that by saying, gee, it's one biomarker used among many to try to be able to come to some kind of more gestalt decision about how to proceed from an earlier aspect of drug development. That's not my area. I don't know how you.
Right.
Okay, you figure out how to live with it. Okay, but if you choose not to look at a performance characteristic, then you're basically saying, I don't worry about how being bad in that performance characteristic is going to affect my drug development.
Right. And ultimately, that will be a decision with the drug developer in the IND space. Right. If they're going to roll the dice, to what level of confidence do they feel that the biomarker is of value? Yeah. And that's not really a regulatory question in many respects. And certainly in the IND space for proof of concept, they're not doing this level of rigor because it's all in-house. They're willing to take on that risk. All I'm suggesting is that for the paper, it would be helpful to talk about that lowest case scenario and talk about when you might be able to remove some things and what the arguments might be.
But this is the one point I want to interject something just quickly. The arenas are related but distinct for drug development versus in vitro diagnostic use. In the drug development, you're looking at something prospective. It's not getting out there in the market. Okay, you've got lots of controls. You've got risks that you can try to deal with from a different perspective than trying to take the test and say now it's ready to release into the wild and actually manage patients. So that the terminology differences, I understand they're there. The concepts are sort of rolled up and packaged in different ways between, say, parallelism versus linearity and so forth. You have to be smart enough to be able to tease those out when you want to, say, crosswalk between what might have been an IVD that was put out there, and now you want to repurpose it as a test supporting a biomarker qualification. You just have to be able to do the work necessary to be able to do that, to make that crosswalk happen. But you've got to realize that the fundamental purposes associated with being able to develop the biomarker and the test for it are distinct from those associated with being able to actually manage patients for a test that's actually in the wild. Though I got to tell you, when I think about real-world evidence coming down the road at some point, I'm wondering how soon that stuff's going to emerge. But in our classical development programs for drugs and for devices, what I've just said holds true.
Let me just make it, I'm sorry, go ahead.
I also think it's important to talk about the scope of the context of use and how sometimes that changes based on the level of rigor of the qualification. And often what we see is people have limited access to samples or limited access that, as Chris is talking about, and we approach that using the risk-benefit analysis by reducing the scope that will qualify it for use in drug development. But often those same submitters or qualification requesters are really looking down the road for a broader use. So this conversation is about, as Chris said, a bottom entry level, but also how people can grow those uses, acquire more samples to at later time get a more robust validation, which would then get a more expanded use in drug development.
Thank you. So I just got a sign that it's we have 4 minutes left. But there's going to be a lot more time for discussion. Just one point, guys. Again, I want to go back to what was stated. I really would like this team to come up again with the core parameters that we want to include as the bare minimum, and what sort of acceptance criteria, we want to put out if there are multiple, ways of doing it. I cautious everybody. Be careful again when we're talking about 3 or 4 parameters or whatever. Or we talk about delusional linearity and parallelism and mixing them again. We are here today to really come up and distinguish some of this terminology and what is it that we want to do moving forward. So let's stay focused on those parameters. And if we need to go cut some or add some, this is the time for discussion. So I want to really focus the discussion. We're not going to have much time right now. We have only 2 minutes left. But look, why don't we go ahead, if it's okay, take the 2 folks here quickly. Why don't you go ahead, please?
Yeah, this is Yan Zhang from Bristol Myers Squibb. I will just have a quick comment and also a quick question. So first, it's a well-written document. So I intended to use this as a teaching material. For that purpose, I would just feel it will be more helpful for you add some more example tables, such as if I read table 10A, it's very clear. Not only you have the wording, but that table with the exact number will support, will illustrate what you have written. So if you can have this kind of table for selectivity and also...
Yeah, I think there's a caveat with that those numbers look odd to, I think, some of my colleagues at FDA, and it might be wise if you're... it might be wise to consider them, it depends, and at the best use them as certainly not as yardsticks of targets.
No, it's
I think the numbers are screwy.
Yeah, that's show how show the complexity and also how to assess that because sometimes for scientists they look at a concept. Sometimes it's harder for them to design experiments and show the data, and that kind of it gives even though understand that this is not exactly the only way to do that, because you mentioned that in the wording there are a number of ways, and this.
I think point well taken. I think what you're asking is go ahead and put more examples. I think that's the best way for people to understand, and I think we are now recommendation to move some of these examples to as an appendix. So I think those are 2 good points. Thank you.
Okay.
Chad?
Yeah, I just had one comment around also the case study. I really like the concept of performance standard, total allowable error, but I don't know that it came across in the case studies that you actually applied that. It seemed more of a traditional biomarker validation approach, plus minus 25-30%. So I think that would be useful to bring that forward in the paper. The other question I have though is if you have a biomarker that has demonstrated the use concept, then for those coming after that, let's say IL-6 as an example, but yet it's built off of different antibodies, et cetera, does that require a full validation, or are you, in essence, demonstrating some degree of commutability or comparability
Give you
to...
my opinion.
Okay.
Different antibody for large molecule is a validation over, because who knows where that binding, the stability, you're talking about even the stability of that epitope that is binding to. But
It's really important to note that biomarker qualification doesn't qualify a test. It qualifies a biomarker under specific performance characteristics, and those are stated in the qualified conditions for use. So someone coming in under an IND for the drug approval review would have to provide the performance characteristics for the assay they're choosing to measure the biomarker with, and set specific decision points for their assay in that submission.
Wouldn't you say, though, really you are qualifying the test? Because
So
both the test and the biomarker, it seems to me.
So we're qualifying the biomarker and the performance characteristics to use it to make decisions. So when someone comes in under drug development, they don't have to start at the beginning of why are you even measuring this biomarker.
And Chad, your point regarding the example that was used, yes, it is. When I looked at that as well, it was like the traditional BMV way that we're doing it from a PK, and so point well taken. Yeah. By the way, if you guys have examples for what we're talking about, for better illustration, you know the routine. Send it to us. Please let us know, and we want to incorporate it. Jeff?
Yes, Jeff Selstead, Bioagiletics. I'd like to point out that the term parallelism in a dilutional linearity was in the original bioanalytical method validation guidance years ago, 1980, I think it was. And they were defined as parallelism would only be with incurred samples. So I think you should stick with that. And in the cases with a biomarker that is upregulated, you may need to spike it with a recombinant product early on, because you're dosing up, and especially if it's dose dependent up modulation, you have no other choice to be sure that your assay is legitimate.
You sometimes have to do that to set up MRD. I mean, there's just some limitation, but we got to remember it is not a true. Go ahead, please.
But the point I'm making is let's not redefine those terms.
Yes.
I think it's well established.
Last 2 minutes, Ross.
30 seconds.
30
we
seconds.
have two calculations for total error in the document: bias plus intermediate precision, or bias plus 1.65 times CV, which is, CVA. So we probably, why didn't we change that?
One is defined with an acceptable alpha error and
Got it.
so they are not.
So they're not synonymous, and yet it's the same. One's total error, which is bias plus intermediate precision, and total error allowable. That needs to be fleshed out, because they're almost the same, and yet they're not.
Yeah, we need to, that's correct. I think, again, thank you very much, guys, for your participation. There will be more time for us to go into more detailed discussion. I hope, again, by the end of tomorrow, we can narrow down again if there are any comments, feedback around these 7 parameters that we discussed. So lunch, we're on our own, right? Yep. So would you like to make a quick announcement? So that as when people are coming back?
Okay, thanks everybody. Lunch is on your own. We do have some maps of restaurants and food trucks that are in the area outside. We also have a restaurant that's in this very building. Please come back at 1:45.
And I'd like to thank our panelists up here. Thank you very much.
lunch, anybody? You guys have lunch plan, or? I don't have lunch. We have a break. Sure. Do you have lunch plans? I don't have. You want to go? Just grab some people right away. Whoever comes with us, let's just go grab someone close by. The jacket is off. I know.
Oh, I see. I will find something. Okay. Another way is to explain to pragmatic things that are
Yeah, yeah. Oh, yeah, yeah.
So we have one of the guys on our board. He's the he was the chief counsel for the FDA, right? So he tells the story beautifully. So he tells where all the laws come from that come from the FDA. Yeah, he gave a great presentation.
All right.
No, you guys go get lunch. That's gonna be fun.
Try and get something.
So what are the variability of those? You can calculate.
But the QC is obtained from the same study or different study?
The same study, during the pilot study, I'm saying.
During the pilot, you do the QC on the pilot.
Yeah, you still, I mean, every run, so, okay, first of all, you have, during the exploratory phase, you have some limited analytical validation, pre-study validation. So we did that, looks okay.
But the pre-study validation is also from yet even a smaller panel.
Not necessary. Let's say, I mean, then you could, usually we have a so-called zero phase. Okay, so you have a few samples that allow you to set up the QC sample. So then you run your first study, phase 1. Or even still not yet phase 1 of the pilot. So I mean, let's say, I mean, this is safety test for safety. This is another one is to see any efficacy. I mean, not certain that phase 1 is sort of right? And then you can collect. I mean, I have some sample already collected, and then so you have usually just a low QC, a high QC. So then you just run them. On top of that, I still would have, let's say, I mean, using a surrogate matrix for the standard curve, using a recombinant for QC of low, mid, and high. Those are the analytical parts. I still use those recombinant QC to pass or fail my curve. Okay. But I still continue to start to run those endogenous QC samples.
Left hand pointed back this way. No problem.
Also, in addition, total error now is defined total error defined as bias plus imprecision. CV bias plus right.
So it's 1.65 precision.
Right. And then we also plot on the other axis we plot bias. So then it will actually show
Which axis? The total allowable error on the right.
not just total error, not allowable, because we don't know what's allowed. Total error is from the yeah. Okay, and then this is the QC level concentration.
Chew. Test 1, 2.
H.264 things, and I reduced every hour-long clip to 2.2 minutes or something, and that makes it so much easier for them to work with.
So there's never any like
Chew. Hand on mic. Test 1, 2.
ways, but we got a hold of one of these. Well, we have two.
No, that's what we use.
Oh yeah, those are. That was the best thing you could do.
5 mics close to handheld.
It's the closest to. It's the easiest, best thing.
Clayton. This is Clayton. Yeah, what are you listening to? Are you listening to the camera? Okay. I'm switching to a different mic, different frequencies. Here's another one. Test 1, 2, 3, 4, 5, 6, 7.
I kill my record. I don't kill the stream. If you take the stream down, then you have to stream again. Oh, no, that's fine. No, I brought it down to here, but if it's just like, well, if they've been chatting, you won't hear it. But
Okay, welcome back, everybody. We're going to go ahead and get started. Okay. Welcome back. In this next session, we're going to begin thinking through how the framework might be applied in actual practice and using the real-world example of the Predictive Safety Testing Consortium's nephrotoxicity biomarkers. Our moderator for this session is Steve Piccoli, whom you all know, I think, pretty well. He'll give the initial presentation, and that'll be followed by some panelist remarks and open discussion. So with that, I'll hand it over to Steve.
Thank you very much. And at this point, I have to wonder why I always get the plum position of giving a talk immediately after lunch when people want to go to sleep. I will help make that easier. Okay, actually, this is what I think is the meat of this meeting. We have an example here that we can use to anchor further work and discussions around the white paper. And keep in mind a few things based on that, is that I will not be showing all of the data that we have generated in this, but I will show you what was done to do that and a number of things, because the qualification is still in submission. But this is, I think, the most fun aspect of this meeting. And if you can't have fun with biomarkers, well, crenellated red blood cells, that's a biomarker. Guess the disease. You have until the end of the talk to figure it out. So we will go. And here we go. Applying the framework, biomarkers of drug-induced nephrotoxicity. So I'd like to give you a little bit of background on exactly how we set this up. This has been a long process. Bear with me. We are going to go through some history, but it all has a bearing on the end game and the submission that is in process now. For the panelists, we will have Eliza Thompson, Kelly Kelm, Frank Sister, Meena Subramaniam, and Amanda Baker, who will join me up here after this part of the presentation. Two of those people have been involved with this qualification effort for 8, 9 years, something like that, as well as Kelly is one of the people who has reviewed this material internally. So we have actually a very good panel to ask questions of. Yeah, we're not doing this terribly well this time. There we go. So the Kidney Safety Biomarker Project summary. This began as a collaboration between the FNIH Biomarkers Consortium and the Critical Path Institute, specifically the PSTC, a working group to develop this project and bring it through. A lot of this work has been previously presented to the FDA, the EMA, and the PMDA. This is for extending support for the translational utility of 5 urinary kidney safety biomarkers listed here. Each of these biomarkers has been previously qualified by the FDA, EMA and PMDA as well, for use in rat studies in drug development. This all happened in about 2008. This qualification submission also is intended to support the clinical utility of 3 additional biomarkers, that is NAG, NGAL, and osteopontin. So the initial briefing package for this was submitted in 2011. I did mention that this has been a long, ongoing process. Work for this, if I remember correctly, started somewhere around 2008, about the time, maybe earlier, but shortly after, or maybe at the same time as the qualification for the rat markers was given. So it's been almost 10 years. They told me to point left and the other left. Current qualification status. Okay, so this is where it sits now. The 2008 PSTC qualification was for albumin, KIM1, cystatin C, clusterin, beta-2, microglobulin, total protein, and TFF3, okay? Oh, this is not going to go well. And just to remind you, it's in rats. I do know how to punch buttons. but apparently not well. A short time after, HESI qualified 3 biomarkers for use in the same situation, that is also in rodents: clusterin, alpha-GST, and RPA1, renal papillary antigen. Now, alpha-GST was not recommended for qualification by the FDA. You will note that there is one biomarker that's... I hope this is as funny for you guys as it is for me. Okay. Originally, that arrow pointed from clusterin to clusterin. No longer. But, okay, clusterin was the only one that crossed over in those 2. There's also the 2014 PSTC letter of support for NGAL and osteopontin. So within that entire collection, you can see the biomarkers that we picked. And a word here, there were 25, 28 biomarkers that we initially went into this with the potential option of looking at. We had to whittle this down to a number based a little bit on finances, based a little bit on test availability, based a little bit on samples. So we actually looked at a much larger sampling of biomarkers than even these to begin this project. But let me show you exactly what we did. So you can't tell the players without a scorecard. Here's the scorecard. The title of the application is Qualification of Translational Safety Biomarkers for Monitoring Renal Safety in Clinical Drug Development Research Trials. Remember, this is clinical drug development research. This is not laboratory medicine, and this is not exploratory medicine either, or exploratory biomarkers. The context of use, and this is the thing that we scribed out years ago to kick this off. Qualified renal safety biomarkers are proposed to be used together with conventional kidney biomarker monitoring, serum creatinine and blood urea nitrogen in early clinical drug development research to support conclusions as to whether a drug is likely or unlikely to have caused a mild injury response in the renal tubule at the tested dose and duration. Okay? That's it. So that sets the population parameters, that sets the test parameters right there. So we know that anything outside that is outside the scope of qualification of the biomarker. These biomarkers can be qualified for a different purpose, a different COU later. The study population is healthy volunteers and patients with normal renal function, taking into account age and gender. This is the initial package. There are more for more complex situations that will follow. So this is just the picture of looking at where these things are. The red dots are the highlighted biomarkers which we're using. You note that they may be expressed in different parts of the nephron. And back to the players in the scorecards here. Here are the biomarkers which we examined, as well as their potential areas of expression. One of the other criteria for selecting these was to try to get a cross-section of injury areas so that we were not restricted to simply the proximal tubules or the distal tubules, but we wanted entire kidney injury. So this is part of the selection reason, and you can see that the expected response for everything listed up on the screen. Here we go. So the strategy for doing this, we started off with a learning phase. The PSTC Healthy Volunteers study, which was an observational study conducted at the Jasper Clinic in Kalamazoo, Michigan, 81 subjects between May 2011 and October 2012. These samples were taken and later measured. The other part of this, the intraoperative intrathoracic cisplatin surgery study on mesothelioma patients. So these were patients treated with surgical resection, intraoperative cisplatin placed in the cavity, 59 people between August 2008 and December 2010. So these are the baselines for learning how to deal with our biomarkers. The confirmatory phase, which is actually subjects of a more complex context, an extended context of use, with these biomarkers is an aminoglycoside study in cystic fibrosis with 100 patients, adult CF patients with acute pulmonary infection, treated with tobramycin not to cause kidney injury, but capable of causing kidney injury. Controls are 25 CF patients treated with a different non-nephrotoxic drug and 25 adult CF patients with no infection. Also a cisplatin study in cancer, 100 patients, adult head and neck squamous cell carcinoma. Other cancers treated with cisplatin as a single agent or part of a chemo cocktail. 50 controls, which were other cancer patients receiving non-cisplatin therapy or radiation. So this is the setup for all of the data I will discuss today. Yeah, we got it. The assay comparison phase. So when trying to determine what we were actually going to assay in these, we did assay comparisons. So this is actually, I think, a fairly extensive review of what was capable technically of being done at the time. We looked at 20 normal healthy volunteer random urines with a multiplex Luminex panel for as many of these as we could, a multiplex mesoscale discovery panel, and singleplex assays at 2 different CROs. We also looked at approximately 40 volunteers and 22 cancers, standard of care treated, random urines, again with a multiplex Luminex panel and singleplex assays, this time at a single or singleplex, if you will, CRO. The characteristics that we assessed at the time were the platform, the precision, the dilutional linearity, spike recovery, stability as measured in freeze-thaw, and pre-analytical handling. So it was based on the data generated from this that we made the decision to go ahead and use certain collections of biomarkers based on the technical specs and the data that we obtained from the learning phase. The assays that we utilized for this are all listed here. You can see the analytes on the left, the matrix we were testing, the regulatory status of the tests, the technology, and who actually made them. So the top half of this are comparative assays and standard of care that we did at the same time. The bottom half dozen separated there are the biomarkers which are under qualification here. You can see that we used, they're obviously all urine biomarkers. 5 of the 6 were RUO kits available on the market. One was a test in a CLIA lab on a Roche platform, 510K approved. And so we had 5 essentially ligand binding assays, 1 enzyme activity assay for NAG. These kits are available from the manufacturers on the left. These were the ones that we decided to go ahead with and finally measure. So here's the analytical validation summary of basically what we did with the 6 markers under the kit conditions that I just showed you. Sample type, collection preparation, stability, long and short term, pre-analytical questions, the reference material, and stability, analytical measurement range, the sensitivity and limits of quantitation, standard curve, range and model, precision, inter and intra-assay, accuracy, recovery, parallelism, dilution verification, linearity, specificity, matrix qualification, assay format, and detection system. So now, starting off, pre-analytical variation here, validation was a bit of an interesting process. There are a number of different ways to process urine. These have different effects on the stability of the biomarkers and the subsequent samples, how they can be tested. So we looked at processing samples with and without centrifugation to clear cellular debris, with and without stabilizers. It had been a well-known fact at that time that Arbutus, a company which made kits for urinary biomarkers and looked at some slightly esoteric markers at the time, required a stabilization buffer. We used that as well. That was for some of the GSTs that were notoriously finicky to deal with. But we looked at addition of protease inhibitor cocktail, fairly standard 6-fold protease inhibitors as well as the Arbutus stabilization buffer and exogenous protein buffered as well to be added to the samples. We also needed to look at desalting the samples before testing. So all of these things were examined back and forth. Keep in mind that this was done for 6 different markers, and we had to find a way that we could process these samples efficiently that would allow us to measure all 6 reliably, reproducibly. You know the whole story here. So this was actually a great deal of work to find out the right conditions to be able to begin measuring the samples. Then at this point, we can set up the collection and processing protocols and go ahead, collect the samples, and start to generate the data. So here are the things that we looked at in detail. Sample stability, at least 2 samples, low and high in the assay range, replicates 3 at each concentration. They were stored for at least 24 hours at -80 before being thawed. Acceptability for change was set at less than 20%. Freeze-thaw stability, we looked at a minimum of 3 freeze-thaw cycles, and we did this to mimic intended sample handling conditions used during sample analysis and for long-term stability. Also did benchtop stability, again, to mimic handling on a benchtop. One thing I should say here, though I hadn't explicitly mentioned it, we did not have the ability to do native sampling. So when the samples were obtained from the patients, they were processed and frozen. We do not have time zero data on this. This is a common fault of fairly esoteric tests that requires a minimum of 24 hours to get these things stabilized, sent somewhere. And when you're dribbling in samples at a low rate of speed, you don't want to be testing 1 sample at a time. So we actually have a requirement that these are only analyzable on frozen samples. These tests will not be qualified for fresh samples, which is a bit of a potential problem. We don't know what the fresh values are on those patients. We simply can't measure them. So these are done, and the requirement is that there is at least 1 freeze before sample analysis. Reference material validation. This one was the only easy thing I think that we got to do because we bought the kits, the manufacturer's COA and reagent stability studies served as our background for this. So that's all I have to say for that. Functional sensitivity. Our LLOQ was established by at least 5 samples, and again, keep in mind that these numbers of at least 5 are pertaining to the entire collection of 6 biomarkers. Some may be more. Generated by dilution of QCs or calibrators. The matrix, appropriate matrix, was used for dilution depending on the test. Minimum of 5 analyses over a minimum of 6 analytical runs were performed. We calculated the values, and the LLOQ is defined as the back-calculated concentration of the lowest calibrator that does not exceed a 20% CV, with recovery at plus or minus 25%. So those were our standards. The upper limit of quantification, again, established by at least 5 assay runs of the highest standard curve calibrator, and defined as the back-calculated concentration of the lowest calibrator that does not exceed 20% CV. Standard curve assessment, we went through the usual analyses of the standard curve forms. The standards were prepared according to the kit manufacturer's instructions. Each curve was made to have at least 6 non-zero points, analyzed in duplicate, covering the entire reportable range of the assay, and excluding blanks. We fit this to what we thought would be the simplest regression model. In almost every case, that turned out to be 5PL for these, but we did examine individually, and this was based on a minimum of 6 runs conducted over a couple of days. The acceptance criteria was that the standard should be 25% of nominal at the LLOQ, 20% at other concentrations, and at least 3/4 of the non-zero standard should meet the above criteria. Total error less than or equal to 30%. Some of this should sound pretty familiar to people doing this on a daily basis. QC samples, minimum of 3 concentrations of QCs measured in duplicate per run, and with a minimum number per analysis. Normal donor urines were prepared by standard protocol, collected, centrifuged, aliquoted. Remember, these are our controls for half a dozen different biomarkers, not a single one. So this involved a little bit more complexity than you might be used to. The endogenous analyte concentration was determined for each donor sample prior to pooling. They were pooled to form the low QC, middle QC, and high QC. And we, because we analyzed those before we pooled them, had the ability to adjust those to an appropriate range along the standard curve. In some, we did not have sufficient amount or quantity in the sample, high enough level, to be able to do this. So our fallback position for those was to use recombinant protein for each biomarker spiked into the urine. Whenever possible, we use the in vivo, the incurred samples to do this. Run acceptance, the run was passed if greater than or equal to 2/3 of the QCs were within 20% of theoretical, and at least one of the QCs, or greater than 50% at each level, must be within 20%. It almost worked. Here we go. Now, we did this, our incoming precision data. I believe Lisa mentioned before about how many of these you get. Well, the incoming precision data from the manufacturers were sometimes 3 and sometimes 2 samples were measured to generate precision data. We decided to do more. Those were measured in 1 or 2 matrices, which were not always the matrix of choice that we were going ahead with here, because again, we had to process these in a particular fashion, and sometimes they were not qualified in urine at all. They were done with 2 to 20 replicates, depending on the assay. The inter-assay precisions were 3 to 14%, and the inter-assay precision 4 to 8%. So those numbers look pretty good, but as anyone who has ever done this and pulled a kit off the marketplace, cell culture supernatant and a real-world biological sample, there's just a world of difference, and those precision estimates will only go up in real live samples. But that's what we started with. So we had to verify that those were accurate or establish those under our own conditions for what was being done. So we did. We generated a precision assessment on our own with, again, a minimum of 3 QCs. Remember, those are in vivo samples in the range of expected study sample concentrations. We determined the precision at all 3 of those points. Cutoff was 20% CV and 25% at the LLOQ. Spike recovery, or relative accuracy, was determined independently of the manufacturer, measured using a minimum of 5 determinations at 3 different concentrations of known spike materials. Again, if we had appropriate biological material to work with or biological samples, we used those. In most cases, that was actually not possible, so again, it was recombinant spiking here. In the range of the sample concentrations, acceptable 20% plus or minus, and at the LLOQ 25%. Dilutional linearity, we did a minimum of 2 urine samples appropriately diluted over the range. We created a series of 7 to 11 evenly distributed samples that cover the assay range, measured them in duplicate, and showed that recovery for those analytes in those samples should be plus or minus 20% of the expected values which we had previously measured. So interference studies, and here fortunately is the CLSI EP07A2, which is a document which I've been painfully familiar with for many years. It's incredibly valuable in teaching you how to do interference studies and find out what is important and what is not. This is, we took this with a great deal of seriousness because it is difficult to measure differences in unknown biomarkers and relate that back to, is it clinically significant or not? We picked a number of 5 replicates to be tested, plus or minus 20% acceptance again. So we took 5 normal urine samples, pooled and analyzed for each biomarker. We created subpools and spiked exogenous analyte where necessary to get a single appropriate pool that we can use for analysis rather than having individual pools for each biomarker. The subpools were split into control pools and test pools. Testing was by addition of drug interference at highest expected urine concentration into the test pools. 5 aliquots of each of the 2 test subpools and 5 aliquots of control pool were tested in duplicate non-consecutively. The observed interference is calculated as the difference, obviously, and the acceptance was plus or minus 20%. Now, so what were those things that we looked at here? So this is a point where we get to say that lipemia and icterus don't matter. It's urine. We did look for total protein, albumin, hemoglobin as lysed erythrocytes, and erythrocytes as intact erythrocytes. Looked for interference from each of these substances. We did some of these with dipsticks, some of them with automated cell counting, looking for intact erythrocytes, and some for standard clinical chemistries on the slides I showed you on the table a couple of slides ago. So now, this gets us to the point where this is what we would have wanted to do had we had the information at the time. So you've seen our TAE calculations before. Here I will note that I'm using imprecision as I rather than CV sub A, a difference in nomenclature, but the desirable specifications are there. And the last line there is our total error desirable from the between subject and within subject coefficients of variation, which we were able to measure in the NHV population. Given the bottom, and here you see our calculation setting the acceptable error level, the risk with which we are looking at the specimens. So now the total error assessment. This was all done post hoc because we had to generate the data and go backwards. We actually, again, keep in mind that these samples were collected as early as 2010 and being analyzed in that time frame before we went to the more rigorous studies for the expanded context of use. We did not have an idea of what the normal intra or inter-individual variation was, so we started to generate that. So the data I will show you here is actually unfortunately not scribed down in terms of the biomarker because this data is still under review by the FDA. But I will show you what we did manage to measure in the normal human volunteers. So these are the 8 biomarkers, the 6 that we looked at, as well as total protein and albumin here. And these are simply the ordered values of the individual CV of each of those 8 biomarkers that we looked at. You can see that ranges from 30 to 65% or so. And that's the intra-individual variation. Can be pretty serious. The inter-individual variation here, and again, don't read anything into the colors. These were sorted from low to high. It doesn't mean that the brown one is the bad one in each case. But these ranged from 30% to over 120% within the group. That's a fairly large amount of variation in these biomarkers. So the total CV from a simple calculation of these brings it to 50% to 140% total CV in the measurement population. So this is what we learned we would expect when we looked at these biomarkers in a normal healthy volunteer population. That's a lot. Our desirable imprecision calculated from the test before is actually much smaller. Desirable bias, much smaller than we were able to obtain there. And desirable TE is shown at the end. So those are the things that we wanted to hit. Clearly, from an analytical standpoint, even if we had 30% error here, we would not be able to measure these things in a fully quantitative fashion. So we did get around that. Just to make a point here, published total error data. There are population databases. We mentioned these before. Carmen Ricos at the bottom, and the paper talks about this. The Westgard database is where the repository was of this information until 2014. The desirable total error specs for things that were published that might have some bearing on this range in the 30 to 60% range, so we're not out of whack here. Serum cystatin C is obviously the outlier here and much more tightly controlled than anything else. So the desired outcome for this, the submission is now in final review, a decision hopefully expected shortly. Output is a multiplex analysis rather than singlet values. Yes, all the assays were done singly, and the results mathematically combined. They were not multiplex tested. So the output is a composite measure of the urinary biomarkers proposed to be qualified as a safety biomarker of kidney tubular injury response for use in NHV trials and drug development. So our limited context of use from this human study is intended to promote use and experience with the kidney safety biomarkers on certain development programs while minimizing perceived risks to patients and sponsors and provide public dissemination of procedures and results. That last part is the big bonus here because when this is approved, we will release the data and we will have it available for public use. This is the anchor that we can use to set for this particular context of use. This is appropriate amount of work to be done. And so remember that we are qualifying the biomarker, not the test, but this gives us an idea of how rigorously we have to develop the test to get that level of qualification in the biomarker. Back one. Okay. Learning points. Now, a glimpse of the blindingly obvious. We have a nearly ubiquitous lack of certified reference materials. Everybody knows this. We can't do serious absolute quantitation. Because of the nature of this, we had an inability to routinely use in vivo samples as controls. We did that as much as possible, but again, for 6 different markers, that's an extraordinary undertaking. Broad guidances didn't help. They don't work in cases like this. We must take individual analytical factors into account for each individual assay and make them work. The most important thing is to define a controlled setting for the collection and testing analysis of this so you can minimize risk of uncertainty associated with the individual assays. The consequences of doing this will cost you money. They will impact your drug development decisions. So the learning points: to ensure accurate and reliable drug development conclusions, the level of analytical rigor and quantity of generated data must be based primarily on the biomarker-specific context of use. You've heard that before today, and the implication of the benefit-risk profile associated with that. Therefore, the assay performance and validation characteristics must necessarily be directly derived from the context of use. So here is a listing of the collaborators who had a big hand in that. There are many people from the FNIH and our colleagues at the Food and Drug Administration who have been working with us for a number of years. And I say us for a number of years. I think I started on this in 2007. It's been a long time. And with that, that is the overview of the project, but it's time to move on. And so I'm going to walk into the panel discussion with a quote from one of my favorite philosophers who, as it turns out, knew something about kidney safety biomarkers. So this is Kurt Donald Cobain, who would have turned 50 this year had he not passed away unseemly. And whereas he is justifiably famous for making us smell like Teen Spirit, he also had a quote which is pertinent. We have no right to express an opinion until we know all of the answers. So that's what we're going to do now. Ask the questions. We will give you the answers, we will discuss the answers, we will develop the answers, and then we can form our opinions about how to go forward. So with that, if there are any direct questions for me, I'd be happy to take them, but if not, I would like to call the panel up to begin the discussion session. Mark, a surprise.
So that was a really good presentation. So it's important to me day to day to show that my laboratories can generate the same result from the same sample 2 days in a row, right? And I see a lot of times where they can't do that. And now we start to get into the incurred sample reproducibility. I noticed that you didn't have a formal evaluation of that, but you did use QCs that were from patient samples. So how do you recommend that we move forward with this, knowing that for a person like me, that's very important, that I like to prove that with all the different labs that we work with, that their assays are, are rugged.
So you can do that with the long-term stability done in reverse, the wrong way. But you will do your analyses on sequential days and show whether or not they give the same answer. So you can do that. There was day-to-day data generated, which I did not talk about. We didn't get to all of the things because of time limitation. But it is very important, my lab as well, that we know that tomorrow we're going to get the same result on the same sample as today. So
But do you think they should make that should you make it a formal requirement in this document?
So it plays into robustness, ruggedness, reliability. They are different. One of those actually is not a defined term. Yes, it does. Even if you are doing single point analyses to determine whatever it is you want to determine, we need to know that the assay is performing today in patient X the same as it is tomorrow in patient Y. So it's a little bit of the circular race to try and define that, but that's the problem that we always have before we get certified reference materials to do the standardization of that. So we do essentially round robins within a lab. This was also only done in one facility, so this is not in many cases, not even an inter-operator variability study, but not an inter-laboratory study.
Tables 9 and 10 allude to EP15, which is 20 days of singlicate measurement, which is used to represent 1 month in time. So it is sort of folded into tables 10 and 11, although you didn't really deconvolve your 20. Did you do one replicate of each of the QCs over 20 days?
No.
But if you could turn back time, terrible musical reference, you would, right?
Yes.
So Steve, as you're getting the panelists to come up, certainly you are applauded, you and your colleagues, for this long effort and the extended time frame in which it occurred. The specifics around the project aside, especially since it's still under review, can you think of one or two things as a sort of best practices or lessons learned of what you would have done differently had you had the benefit to go back in time, as the reference went?
A couple of things. One of the things that was actually most important was the pre-analytical variation studies at the beginning, because that really let us get to where we wanted to go. The one thing that I think we all agree that we would like to have done a little bit differently is our long-term stability, because this is an issue of, we thought we knew how long this would take. The surprise, it took longer. So in qualifying these markers, even though I've already said we don't do this fresh, we do this frozen, there was a somewhat limited time frame of stability that we had initially set out for these. So we only set up enough samples to do analyses over a particular period of time, and we have samples now that are older than that. So that's a concern. I guess when you get to the point where you say, well, we got 6 markers, so we need 6 aliquots for each from each, and then we have the points at 0 and 3 and 6 and 9 and 12 months, and it gets to be a lot, and you say that's too much. I would urge you to err on the side of do more. That was difficult. Setting the error limits, again, we did not have the opportunity to do that because at the beginning we had to generate that data before we went into the extended qualification. So I think that's probably it, Chris. Thank you. Oh, and talk to the FDA early and often. And that's not a plug, it's the truth.
So I have a question about going back to the context of use. Now I saw, and I may not have caught everything, Steve, so you had healthy volunteers and I think cancer patients in your learning set, and then you have CF patients. Of course, these are renal biomarkers. So how do we explain, I mean, I saw the context of use definition, that was helpful, but sort of think, and I'm still thinking out loud actually, so which patient population does it apply to, and how did we qualify that context of use?
So the cancer patients there played into the larger context of use, which I didn't describe here. So it was the NHVs, which were in the limited context of use, which I showed on the earliest slide. So the cancer patients served different categories of kidney injury and non-injury in the same disease, so sets of controls for those. But they didn't play into the limited context of use for normal healthy volunteers. And actually, before we go, perhaps we should get the panel to introduce themselves, those who have not been here before. So
Let's see if I know how to turn this on.
it's on.
Oh, it's on. Hi, Eliza Thompson. I'm a clinical team leader in the Division of Cardiovascular and Renal Products. I have been at the agency about 10 years, and I think I've been in the biomarker qualification process at FDA for that, I think, whole time.
Amanda Baker, an old panelist.
Meena Subramanian.
Hi, I'm Kelly Kelm. Right now I'm acting Deputy Division Director for the Division of Chemistry and Toxicology Devices in CDRH. And I've also been involved in a few of the biomarker review efforts along 11 years I've been there.
I'm Frank Sestair from Merck. I head up the FNIH clinical biomarker qualification effort, and I was involved in actually the formation of the PSTC back in 2006. It actually started, and then the submission was in 2008. We did that really fast. This clinical stuff takes a long time.
Okay, now on to the question.
Right.
I'd like to help address that question too, though. But to address that question, the context of use, so the context of use is designed for early clinical trials in either normals or patients with normal renal function. So in our prospective study, while they are patients with cancer or patients with cystic fibrosis, they come in and they are disqualified if they don't have normal renal function. So they have to have normal renal function. And it does speak to the point, it's impossible to do the study you want to do, to take a normal healthy person and give them, an intentional, nephrotoxicant. You can't do that, right? So you have to take standard of care agents, which happen to be nephrotoxicant, and give them to patients who need them. However, in the learning set data, we did have patients that did have either grade 1 or grade 2, as well as normal renal function. So we do have some biomarker data responses in patients with some compromise of renal function as well.
Yeah, that's helpful. Thank you. I guess maybe I'm going to ask some minor questions really before starting to express opinions. You mentioned, Steve, multiplex, but read as singleplex. What did you mean by that?
So each test, there were 6 different kits that were used. Each test was, each result was generated on a different sample and independently. So there were 6 singleplex tests performed. Then mathematically, those numbers were combined post-testing to provide a composite measure, which is actually part of the submission.
And maybe my last question was, given that, and I agree with you, I mean, it still took shorter than longer, but these things do tend to take quite a long time. So I wondered what issues you ran across for, reference materials, lots, bridging. How was that?
So almost all of those were handled fairly reasonably, as you might expect. We have a current issue with manufacturers dropping tests. This is what happens when you go over a long period of time. But all of those things are, I think, fairly commonplace. We've learned how to bridge lots. We've learned how to look at method comparisons to do that. I don't think we have anything stunning as a lesson learned there, other than what's already done pretty widely and well accepted in the industry. It's going to take longer, it's going to require more, and you just have to get geared up to do it. And this was no exception.
Okay. No, that's a good, very important point. I don't recall if I had seen that, and I'm sorry if you have covered it in the white paper. That's something, industry has been still discussing. How do you bridge the commutability concept of it?
We did not.
Yeah.
We used same kit lots, actually, over most of the studies for most of the biomarkers for the testing phases.
Okay, thank you.
So in the context of use for this panel, it says that the data is being used in a supportive fashion. At least that's how it was stated in the context of use. And I guess how would it be different if it was like a decision-enabling marker or something like that. The way it was stated, it seems, it's really the other renal markers that are being used for the decision. I just wanted to know how the panel would have changed things if it was more of a decision-making marker that you were going to make a decision on.
No, it is a decision-making marker. I mean, our preclinical data demonstrates very clearly that these biomarkers outperform the standard BUN serum creatinine. We're just saying that we're not going to stop using BUN and serum creatinine. We're not going to replace BUN and serum creatinine. We're going to use it together with BUN and serum creatinine. But the proposal is that these biomarkers are expected to light up, and the data have shown that they will light up when there's no change in BUN and serum creatinine, when in animals you can see histopathology. So BUN and serum creatinine are not, they're not the be-all and the end-all. We know that there are better biomarkers than them, right?
So supportive is the safe word there, in addition to, rather than instead of.
Okay.
Your question is, if it were a context of use that isn't in addition to something.
Yeah, it was a standalone, yeah.
Yes. So that can clearly be the next step. Once we've shown that these outperform the standard biomarkers according to how we measure and interpret the data, then the next one can certainly be instead of. But no one ever gets rid of the old markers, even if they're not very effective.
Well, so another one that's chugging along is vascular injury. Vascular injury, there is no biomarker, and that's really stymieing drug development. And so there we will rely on a newcomer. Hopefully, someday, we will rely on a newcomer where there's not much else other than inflammatory biomarkers or something like that. So that will be a different context of use.
And I just actually wanted to make a comment also about how specifically or concretely, it would be informing decision making. So, for example, how it's sort of set up in this limited context of use, sort of this very circumscribed context of use, is if you were to get an abnormal finding, it should trigger further investigation, but not necessarily be interpreted as, yes, my drug is nephrotoxic. Similarly, if you weren't to get a signal on these biomarkers, but to get a signal on your other ones that are more traditional, you shouldn't be falsely reassured. I think as this progresses, hopefully you come to the point where it's not, you pause when you get something abnormal and investigate further. You actually believe that the abnormal is probably nephrotoxicity.
And we should probably say that limited context of use is a cohort level decision, not based on one signal in one individual as well.
Right, so you're not guiding decisions on an individual level. And again, the focus is early studies, first in human studies. It's not like I'm going to monitor this to ensure safety of an individual subject. It's that I'm going to look at it at the end of the day, maybe when my cohort's done or my study's done, and try to use the data to make better decisions about what doses to carry forward, et cetera.
So, to Kylie's point earlier, those would be the conditions for use. So the context of use is not the whole story.
So sorry, and to clarify that, just to separate the analytical from the sort of clinical data. So as you move along this continuum, it sounds like you're saying as there's more actual real-world clinical data that supports that, that's what would move it in that direction, not that you would be looking for some additional analytical validation. I think that's what...
No, I don't know. I don't think that's the case. I think, and maybe this is an issue we'll come to later or not, but I think that the sense of this is you want to use these early on to the level that the data support their use, right? You don't want to wait to the end of the day when you've required full analytical validation or full clinical validation. And so I think sort of early on, we build in a lot of risk mitigation strategies to allow use when there's uncertainty about the assay and uncertainty about the clinical performance. But you may not need those strategies built in once you make sure that the assays are better developed. And I mean, Kelly could speak to this obviously more than I, but there are still concerns with the analytical validation thus far, and though it may be sufficient for the proposed context of use, for a broader context of use, not clear that it would be sufficient. Or do we already know that it wouldn't be sufficient?
Okay, because I think that was getting to the original question Chris was asking, and that was, if it were going to be used for that higher bar, what is the difference in the analytical validation you're looking for?
So I think we had some questions, and some of it also came around the composite measure, and whether or not that was going to be used going forward or not, and whether or not when you use an algorithm, is that robust to the individual analytes as well? And I think that's still a question that's outstanding, especially given that in the limited data that we had, there was obviously pretty broad imprecision. And so I think that's why there was support for this, look at the cohort. But if we wanted to look for more, context of use in the future, whether or not we wanted to actually take a look at that and think about whether or not, but I think that was the question about whether or not the composite measure would still go forward or how that would work.
Just two quick comments. I want to make sure that we don't dismiss the comment that was made, which was around the lot bridging as well as the the stability. The stability of most of these RUO kits that we use that eventually we want to move them into biomarker qualification. The manufacturer tells you 12 months from day of purchase. Some they have data, some they don't have data behind it. So again, if your study is going beyond that 12 months, you do set up some lot bridging. But I think what maybe our paper, what is missing is that what are different approaches that we do to lot bridging? The reality is, depending on the vendor, you can have as much as 200% variability, twofold variability. Is that due to antibody, recombinant protein that is in there, whatever it is? It's out there. So I like what is mentioned here is that maybe we need to provide some of the general practices, one comment. The second one is I want to go back to the general comment that, why don't we include, for example, multiplexing into this? I mean, I'm just looking at this panel, this study, and I cannot imagine now you take this and mix all the 6 panels and the 12 antibodies together, make the quality control for each one. What if 1 or 2 of those did not pass, didn't meet the acceptance criteria? So this whole idea, again, I go back, I really encourage us, we need to keep this simple as we have done it and not muddy it yet.
Okay.
One response to your first, we actually did do QC material that bridged the kit lots as they went down, but we also talked to the manufacturers about their internal changes in those kits. So we did, in a fashion, we bridged every change that was done in the analysis time.
And Steve, what I'm getting at is, for example, we use, the endogenous samples to broad bridge. If that's not available, what is the next best thing? So I think we need to have something in there to provide some general guidance, maybe.
Yeah.
So also, I just wanted to highlight before the next question comes up, we're also mixing up when we're thinking about multiplexing. In this instance, they were run as individual assays, and they made decisions based on acceptance criteria for each individual method.
Correct.
Sometimes when you think about multiplexing, you're looking at them as a composite. So we need to be careful about what we are referring to in the context of multiplex.
But also we did look at multiplexes early on, and that was Steve's point, and we intentionally moved away from it because it didn't meet the criteria that we needed them to meet. But I remain an optimist, and I do believe someone will master that multiplex challenge, right? And they may be able to get us to a point where we could use a multiplex. I mean, we
do have IVDs that are multiplex.
Exactly.
So you can also talk to some of them and see lessons learned.
And we've got multiplexes in the animal studies that are working quite well for us.
Right. It's just that, I totally agree with you. I think we even, in the initial stages of writing this document, we talked about multiplexing different endpoints, right? Not just proteins, but you could have a genomic endpoint or a genetic endpoint and all of that. But in the context of this particular example, it's not the traditional multiplexing. So since we're throwing so many terminologies around, I want it to be crystal clear.
Yeah. And I do believe that, Steve did a really nice job of showing the different information you get from these biomarkers. They are reporting on different segments of the nephron. They're reporting on different histopathologic processes. They all have their different timing. Some come up fast, go down. Others come back a little bit later when regeneration is going on. You can almost look at those biomarkers and you can see what's happening under the microscope slide sometimes. In some of the animal studies, it's just outstanding. So we're going to lose that potentially. Because, one of the things is it's always this push and pull between how much do you need to spend, what's sufficient, what's necessary, how many biomarkers do we have to measure. The more we measure, the more chance we have of false positive, which ones are the best performing. So we have a statistical analysis plan, which is a step down to look at which of these biomarkers are doing the best job. And then, do we start with 6? Can we go to 4? Can we go to 3? Can we go to 2? How many do we need to pass our preset criteria? It may come out of here with 3 biomarkers that do all the work for us. But in a sense, it'll almost be a shame to not be able to look at all of them. And that's why a multiplex, you would look at all of them all the time for the same amount of money,
And I want to say that the FDA is used to, I know we've seen some composite biomarker tests where it's even across different modalities. I want to say we have some with fluid biomarkers as well as imaging biomarkers combined. I think the neuroscience space is really moving that direction. And that's another thing where, FDA would be more than happy to work with you on those. So don't feel like you have to stick with one type of technology, method, or biomarker, if you will.
Hi, Liz Mansfield, formerly at FDA for many years, and now at GRAIL, that has absolutely nothing to do with therapeutics yet. Anyhow, I want to go nitty gritty right now. I noticed in one of your slides, I believe you said that you set your acceptance criteria for the intra-assay and inter-assay variability.
Mm-hmm.
And I was really surprised that your intra-assay variability was set higher than your inter-assay variability. And I was curious at how you set those criteria.
Past practice and during the learning phase, looking at the reproducibility of those kits. And believe it or not, sometimes that's the way it works. It doesn't always make sense, at least to me, because you would think that your inter-assay has got to be better than your intra-assay, but it doesn't always work that way.
I think it's the other way around.
Sorry.
Intra-assay would be all the same conditions. Okay, so that's just the way it is.
Yep.
Okay, and the other one is maybe, I'm not sure, I wasn't here in the first of your talk, I'm sorry, and you don't want to talk about, but this combining the answers, is it an IVD-MIA? Is it a multivariate index assay? So are you weighting different ones differently? Or?
So, no, it's not an IVD-MIA in that sense of it.
Mm-hmm.
But from the data, we can tell which ones contribute most to it, but they are not being weighted differentially in the composite measure.
And are you doing sensitivity testing? Because some of those markers look super variable.
So some of them are, but we've already faced this issue because of, difficulties in some of the assays. So which is a more robust assay? Can we trust some of the lower levels of some of these biomarkers that we put in? And yes, it is true, but calculations were done to show that the variability that we determined that we actually had in the individual measurements of the biomarker did not change the overall composite measure output.
All right. Thank you.
You're welcome.
I think you just answered my question.
Okay.
I was just going to ask from the point of a biostatistician to look at multivariates.
Mm-hmm.
And if you tease it out and combine with the biology, like is it an acute, is it a chronic, and how does it process? Or is it also a probably pretty healthy subject, and they have the repair system going on? All this will be pretty involved.
Right.
And it is really complex to try to tease out. And so, where are the hopes in the future? Because we're all dealing with complex diseases.
Right. So maybe our FDA statistician would like to comment on that, standing behind you.
Yeah, so this probably is also a reason why I stand up here. I think there is a distinction between using multiple biomarkers as a panel versus using individual biomarkers. I haven't got a chance to discuss internally, but scientifically, I personally do have concerns of calling it simply just a composite biomarker, although the field is calling it that way. Statistically, in our field, we call them maybe a risk score if you weigh them unequally. And in this particular case study, the quote-unquote multiplex is actually not using an unequal weighting scheme. And so there is an exploratory qualification space versus that confirmatory that's coming later. And in that confirmatory phase, you are actually looking at individual biomarkers. Right. So there, in a clinical trial setting, when we talk about composite, we're saying either 1 or 2 or 3 or this or that equally without this unequal weighting. That's where a composite will make more sense. But the original exploratory qualification, it was submitted to us as an unequal weighting. So we did a lot of work. We means our stat team did a lot of work to look into whether you weigh them equally versus when you give an equal weighting. What's the benefit of unequal weighting when there's challenges of interpreting the results? And so the qualification recommendation will come later. I cannot talk about that. But scientifically, there is a pros and cons of how you are going to call this particular context of use, using them as individual biomarker or as a risk score, for example.
I want to make certain people in the room are probably confused. I want to make certain that we all understand there is no intent to confirm the composite measure at the end of the prospective study. There is no intent to do that. The composite measure is meant to stimulate the use during this interim period where we have learning data, where we have normal healthy volunteer data. It's just natural fear and tendency on the part of drug developers to apply something to a drug development program. So how can we reduce that risk while at the same time sort of stimulate the familiarity and the use of these biomarkers? So let's put them together as a composite. You measure all of them, and now you get a number. And the number, the claim that the number says is this is within normal variability or it's outside of normal variability. That's it. It's based on normal variation. There's no learning. Oh, by the way, we looked at these patients that were treated with cisplatin that had mesothelioma, and they deviate from normality. That's it. That's all we have, right? We're not going to try to reproduce that or confirm that or reset the threshold or anything like that. This is really a learning experience kind of a thing. So.
And just to clarify, and I think this is actually another nice aspect of it, it's not dichotomous like the result is normal, it's abnormal. It's actually a likelihood of getting. I think that's critical. It's not,
That's right. It's a probabilistic function, and if it deviates so much, it's a 30% chance or a 50% chance or 80% probability, And the nice thing about it is set on the number of patients that you have in your trial. It's a nice little table that our statistician put together there. So
following what Alisa mentioned, indeed, this is really a cohort estimate. So you're looking at the distribution.
It's not designed for an individual.
But still, there is a distinction between the benefit of using the unequal weighting versus equal weighting. When one doesn't really contribute much at all, then you might want to fall back to an equal weighting. The unequal weighting, in this case, if you can really justify, it's so much better in this exploratory space to know whether it's outside the range or not outside the range. That may be a useful tool, but if it doesn't really give you much benefit at all, then you really have to wonder whether we should be using them.
Doesn't that make sense, though, that in the patients where they're undergoing potential kidney injury, that the biomarkers could have differential influence and be much better than others? So it's nice to see from one point of view that the composite measure in the expanded context of use is measured individually, weighted individually to the biomarkers, assessing damage, while in the normal healthy volunteers, they're not assessing damage, they're assessing lack of damage.
Correct. In the exploratory, I'm only speaking to the exploratory space right now, because we haven't actually looked at the submission on the confirmatory phase yet.
So the other point, though, that was raised about acute, chronic, normal variability, context of use. This is where the context of use is so critical. The context of use is only in normals, only acute, early clinical trials, only acute, and only normal renal function. So we've set the boundaries very, very small, limited it.
So the historical slides, and especially I think these last comments, helped me to understand this example better than I was able to do from reading the white paper alone. And leading to this comment that it seemed that the analytical performance requirements that were put forward for this collection of markers going forward had been based just upon what was achievable with respect to the assays and what was achievable with respect to agreement within individuals kind of across, sitting at the level of what was observed performance. I would have anticipated they might have been informed, that those analytical performance requirements would have been informed by clinical information that you gathered from, by biological information that you would have gathered from the rat scenario. So what I would like to ask those, even though you're just talking about finding differences from normal, difference from reference range, if you might anticipate with biomarker qualification programs, analytically having revised analytical performance requirements evolving as you gain the clinical insights that are tied to the use of the biomarker. Here you're talking about different from normal. I mean, whether that actually has a live or die faster or shorter or not, okay? But so you're not really worrying about that in this program. But in many programs, you will be talking about what is the specific clinical insight and whether the analytical performance requirements might evolve, might tighten or might loosen based upon your correlation of the performance of the biomarker and the assays with respect to outcomes.
I think that's a fair statement, might loosen or tighten. So it may not only go one way.
No, I think that's a spot-on comment. I think in retrospect, Chris asked, what would we have done differently? I think we could have leveraged a lot more of that learning phase data and the variability, because I think the opposite. I think that maybe we were a little too rigid with our 20% CVs right out of the box. I mean, what we said, we're going to have 20% CV, and did we really need to meet 20% CV with some of these early assays? And I think we almost hung ourselves by being heroic and trying to develop different assays and more rigid assays, kind of a thing.
But we didn't actually have all of that data at the beginning. So we didn't get that until well after the NHV study was concluded.
But we had that, we have it, and we're still doing some things with stability that are probably not reasonable, right, based on the variability. But there's also things, I agree with you 100%. There's other decisions you make early on, like how do you set your thresholds? Not just where do you set your thresholds, but how do you set your thresholds? Is it a fold change from baseline? Is it an absolute delta, or is it an absolute amount, or is it an upper limit or normal? We don't have upper limit or normals because this is like coming out of the box. So you set your thresholds different ways, you set your magnitude of changes different ways. And we're going to learn differently and maybe go back and revisit some of that stuff, exactly how to do that. And then, the clinical data and the degree of changes that we see and how to set those things, it could have reverberations and come back to how we go forward with these assay specs going forward. So it's an iterative process, but we did put a line in the sand there. And I think some of the variability, we mitigate risk a lot by going with fold change from baseline. That takes care of some of the risk of lot-to-lot variability, and then measuring things across time and space. I mean, that helps a lot. And then there are things like just microalbumin. I mean, you go from not measuring anything to 30, whatever the units are, micrograms per deciliter, whatever, and then that's a huge range. That looks like a huge fold change, but that's all normal. That's all normal. It's only when it gives above 30. So some are above a threshold, some are a fold change from baseline where you have a sizable amount there. So there's ways to mitigate that variability that you see there in terms of the biological impact of it as well.
So one comment that I have to that, though, so it appears often that you sort of decide and choose how you're going to interpret it, for example, fold change, when you look at the clinical data. And sometimes my question is whether or not your assay at an analytical level is robust and accurate and reliable when you're looking at fold change. Because that is another thing that you're looking at. So is it a small fold change, or is it large enough that, if you're talking about something that's an imprecise, like urine biomarkers tend to be, and especially they tend to be impacted by conditions, pre-analytical, storage, et cetera. So, sometimes my question is it actually robust to all, like a fold change and all the things that could happen. Are you sure that's robust to that, or are you making the assumption that it is, without thinking about the assay and whether or not it really is robust to that sort of measurement?
To add another level of complexity to that, people have different urine volumes depending on when you last drank or ran or whatever, right? And so you'll have a normalizer, and that's your urine creatinine.
Just because I forgot to mention that, all of the values that we generated were creatinine normal. Too, just as you would assume that they should have been.
Right.
But we measured the normal values and then creatinine as well as correct.
So, but you have a numerator with variability, you have a denominator with variability, then you have a ratio.
Get more variability.
Then you get a ratio, right? And so yeah, all of those things enter into this question, and then you do a fold change on top of those ratios.
But I just want to piggyback on that, because I know that in internal discussions, Kelly would sometimes circle back to us because she was trying to figure out, what was sufficient in terms of the analytical validation, and would probe us with questions about, what would be a clinically meaningful change, or, and I think that where we kept getting stuck is, well, we didn't know. And, at some point, if there's time, maybe we can discuss how we tried to build into, these studies additional safeguards to mitigate, risk about the uncertainty about the assay or even the biomarker performance. But it is very hard, I think, early on when you get asked these questions and you just don't know.
Right. I'm trying to decide where I'm supposed to be looking, how much I'm supposed to be looking, what's the important point, and obviously that comes from the clinical side. Not always, but the analytical studies aren't always done the way the clinical studies were done, and then how do you bridge that?
So the loose criteria that, in an exploratory phase, which we use is, plus or minus 3 standard deviations, right? For what it's worth, to see if there's a trend towards a difference until the data gets solidified. So you have to start in a place where if you're using fold change, or if you're using really raw values, to say it has to be outside of the variability of the method, and that is where demonstrating robustly with large numbers of samples that you understand the assay capability becomes very important to say then whether there's a clinically meaningful change or not.
But the threshold that we set was each one of these biomarkers, almost by chance or whatever, it comes out to about like a 3.2-fold change. A patient will have normal variability, right? But if you look at 100 patients, only one of them will get above a 3-fold change by chance, right? And if you have 2 in a row, that just doesn't happen. And if you have 3 biomarkers that do that, that never happens unless you give them cisplatin, right? So you use a combination of the magnitude of change, how many times in a row do you see that elevation, and how many biomarkers. And so we have a chart which captures all 7 biomarkers, and we have a decision tree which we're going to be using going forward in a trial. Now, how you translate that to your confidence in your assay is not taking you above that 3-fold by chance because of the noise in the assay. That's why we asked Steve to make certain that the assay is working well enough that we don't do that, right? And he's got to convince you.
But I think this also gets back to an earlier question about when you talk about having your healthies versus your patient population. So the paradigm in which these are really going to be used initially, it's the concept of a study in normal healthy volunteers. And what you're trying to use them for is to figure out at a very early stage whether or not you've caused some hopefully reversible level of injury, and therefore to stop dosing at that setting. So, what would you want as your patient in that setting? You'd like to have someone who's healthy and has had a very low level of reversible injury. But that's not, in fact, what we used to set or to show what injury was. We used patients who were really sick, these mesothelioma patients who at baseline had markedly elevated levels, and they were getting hit with bad nephrotoxins, and we were judging nephrotoxicity by a substandard gold standard creatinine, which we already know is lacking. And so, even there with this question of, what's a clinically significant change, you could benchmark it on the mesothelioma data that we had, but that's not really the fold change or the standard that we really ideally would have wanted for these normal healthies where we're looking for really early reversible injury.
But that's why we're going in now in the prospective study where it's a much milder form of chemotherapy, much lower dose of cisplatin than those mesothelioma patients. And keep in mind, in those mesothelioma patients who are getting huge doses, they open up their abdomen, they give them a bath of warm cisplatin, right? 50% of those patients did not get an increase in creatinine. But everyone got increases in KIM1 and clusterin and osteopon and all that kind of stuff, right? So we're on a good track
No,
here.
and I just want to make clear, I think that there are a lot of strengths to the submission, and it wasn't pushing back on that, but it just, I think, speaks to some of the complexity of figuring out what's acceptable in terms of the analytical validation. Sometimes the data that we're relying on are not as good. It's why I think we need to build safeguards into these studies to mitigate risk related to the uncertainty about the analytical validation, for that matter, the clinical validation. That was more the point I was trying to make.
So also in the context of use that you mentioned sort of going forward, we're not going to be going into oncology patients, et cetera. We're going into patients on clinical trial. All of those drugs will have gone through rodent and non-rodent toxicology studies, and they will have demonstrated reversibility. So they will have demonstrated these biomarkers are coming up in the absence of serum creatinine or BUN with a grade 1 histopathology that reverses. So that will have been part of the package before that drug goes into the clinic. And then the proposal will be, we're going to add these biomarkers. We don't think this is going to happen. We have a 4-fold margin before we think we're going to get this injury. But just in case, we're going to add these biomarkers along with BUN and creatinine just to make certain for this particular drug. Now, if the drug is for an antihistamine, you're going to say no, but if it's, if it's an antibiotic for, one of these MRSAs or something like that, then you're going to say, yeah, go ahead. You don't even need the KIM1, but we'll do that just to make certain, because we don't want to induce chronic kidney disease or something like that.
Right, and actually how I want to tie that back to is the concept that I think is really valuable here of why, you can have more uncertainty about the analytical performance. And I think it's because we're working in these controlled settings. It's not clinical practice. One aspect of it is that it's a very constrained use. Mm-hmm. You're limiting the use to a setting where there's a priori a reasonably high level of concern for nephrotoxicity. We're not talking about deploying these widely across every normal healthy volunteer study that's out there. You're also maybe getting some belief, already that it's this biomarker at least is going to track with the injury. We're doing this in healthy people where you're not going to maybe have so many concerns about interference from concomitant medications or lack of specificity. So I think there's that aspect of it. And I also think that the other part of this working in a controlled setting is, in contrast to doing this in clinical practice, you can build features into the design of the study. A lot of internal discussion we had is whether or not you should only use these in early normal healthy volunteer studies if you also have a control group in that study, because that control group can signal early on whether or not you're getting unexpected findings from your assay. Because I think part of the initial proposal, and hopefully I'm not sharing too much, was the concept of you didn't necessarily need to have a control group. There's also these other ways to maybe mitigate risk in the studies related to either uncertainty about the assay or the clinical performance.
That looked like a better conversation over beer, by the way, or wine.
I'm sleep deprived. I got back from London, so maybe that's why.
I wanted to just nip off a few questions that Afshin pointed to. So you'll see there's quite a large reliance on EP or evaluation protocol documents. There's 3 I just want to highlight as they pertain to lot and reference material qualification. EP9, which we do reference, is a bias estimate with patient specimens. EP, and I'm going to make sure I get this right, EP25, evaluation of in vitro diagnostic reagents, and I know we aren't talking IVDs, but I do think many of those same principles do apply in terms of reagent stability. EP26 is perhaps even more germane. User evaluation of reference, my eyes. User evaluation of between reagent lot variation. So EP25, EP26, thank you, Mark. Are they yours or mine? All right. And EP30, in fact, is determination and qualification of commutable reference material. So there's a lot of retrospective materials and content that are in the EP documents. So that would be 9, 25, 26, and 30. I just wanted to make ask a question and then make a link to my colleague. So, why did you use creatinine and not 24-hour urine? And why did we use GLP guidance and not CVI-CVG as our acceptance? Those are BMV 2001 numbers. So why 24-hour?
Two good answers. 24-hour urines are a pain to collect. You cannot do it. You need to incentivize people more than we were ever able to.
You just label it, this is not milk or orange juice, leave it in the fridge, right? Isn't that what you do?
So there are actually a whole lot of people who don't like a gallon of urine in their refrigerator spiked with hydrochloric acid. It just doesn't go over well unless they have some burning reason to do it.
I think the other reason, we did do a 6-hour timed collection for a normal healthy volunteer study, but there was a concern about stability of the biomarkers and how to keep the urine in conditions where the biomarkers would stay stable.
When there's 7 or 8 additives that you can add to urine, and I get it, then you get into sixplex and then form a factorial designs. I get it. That's why multiplexing is bad, man. And it ain't cheap.
And we did look at that, and there were differences in how each biomarker performed with each stabilizer. So we went with NEAT to avoid that complexity.
Well, so then if they're unstable, then you're relying on creatinine for spot urine collection. And even collecting spot urines is complicated. Midstream, time of day, second void.
But there's nothing you can do. You're going to get those patients coming in randomly anytime. So why not start with the case that will be clinically used?
So why FDA and BMV guidance and not CVI, CVG?
We didn't have CVI, CVG at the beginning. So we started with BMV. And, you know, a lot of those numbers look familiar. That's because they were the best guess, the only guess. They were not scientifically proffered estimates because we didn't have anything until we were done with the first part of the study to do that.
And I'm probably just repeating things. So you've all read every single reference that we allude to in the paper. No, you haven't. I wanted to point out a couple, though, related to your decision-making threshold. The George Klee paper, K-L-E-E, talks about a 5-tier hierarchy in terms of standard of care versus community practice versus outcomes versus the 2-nested ANOVA that we have. So the Klee paper is a wonderful fabric of decision-making for how to make a decision point from clinical perspective, as is the Fraser paper in 1997 Fraser paper and the Ishihara and Boyd. So there's 3 or 4 papers that are all on page 44, and even Jan Krauer's paper on 45. Those papers as a composite of thinking are a really good foundation to answer some of these tricky questions. But I wanted to just make one other statement. You showed the RICO CVI-CVG data that's on the Westguard website. And I think you may expand on this point. Those numbers come from very small populations on tests that we think this is probably good enough to set a standard. For instance, testosterone for the world is defined on 4 measurements on 10 healthy men. For men, women, children, 100-year-old men, well, based on topical creams, that's the same as a 20-year-old man these days, I guess. But remember, those numbers come from very, very contrived settings. So even using those numbers is with a great degree of caution. One should always look to perhaps generate your own. I hope I didn't steal any of your thunder.
So one defense to that, the infinitesimally small number of samples, is that I believe each one of those came from at least 3 pitifully small samplings combined together to get those numbers. And secondarily, one thing, although I will be supportive of using CLSI guidelines wherever I feel the need to do so, that is not the function of this white paper, nor are we trying to make that a requirement of biomarker validation. Those are documents which are extraordinarily helpful in getting you answers to questions that you may not have to develop on your own. They are well tested and accepted around the world by industry and regulators alike. They are a go-to for doing something that you may not know what the best way to do is, and that's where we should leave that discussion.
Steve, but I want to add one more thing. I think, Russ, that's good to just balance the conversation. You recommend few of those documents to pertain to this lot bridging and qualification and so on. But there is 2 fundamental differences between, again, those CLSI guidelines, the assumptions they have taken, versus, again, some of the biomarkers we work with. One is that if you look at those documents, and you guys that are more familiar with that, you can correct me if I'm wrong, they concentrate, focus on pretty much a well-characterized calibrators that they use to establish a lot of their parameters. Well-characterized, okay? The world we live in, and we use some of these, we are not working with well-characterized calibrator. That's one. The way you set up your assays or your testing or your validation fit for purpose is going to make a big difference if you have that or not. The second thing is the entire documents of CLSI talks about calibrator stability. There is no way in there that really talk about patient sample stability or the samples. Why? Because the samples for the, a lot of time for these automated analyzer, you take the samples, you run it within 24 hours, you're done. You never freeze the samples. The world that most of us live, you take that sample, you freeze it. So now so I want to mention they're good. Again, I agree. I spent a lot of time reading that. I became a member a few months ago to see what is the deal about this CLSI. And when I went through that, and when I see the tools that we have in our hand, these research use kits, a lot of the concepts are good. The statistics is incredible, as much as I could understand it. And definitions are great. But how, in the real world on planet Earth, how can we apply some of those to what we do? I found it very difficult, so I just leave it as that.
So you can actually apply those to what we do, and the errors and things in calibrators will wash out in the end. You will get worse results than you would with an IVD calibrator or a certified reference material, no question, but you can measure that. And so I think that those things are very helpful, even though we don't have those in the biomarker world, those being the reference.
Exactly. You know what? The only thing I'll leave it as here is that if I'm putting putting an assay which will characterize calibrator on a Cobas instrument from Roche, which I'm going to deliver a 2-3%, 4% C from the instrument. The way I'm going to end up doing a validation, a statistical analysis is going to be very different when I'm doing a 96-well play.
Absolutely.
So I just want to make sure we're throwing the CLSI guideline in this mix, not guideline, document. I want us to be careful. It's a useful document, but from a practicality, we got to be careful. Does it apply to some things? Maybe it does, but just be careful.
So here's scientific consideration again. They are not designed exactly for what we want to do here, so we need to use our scientific judgment about what parts of those are appropriate to apply to the drug development process for biomarker qualification. Keep that in mind. This is not a blanket endorsement of using them in this process. It is, however, I think, an endorsement of using them as a reference guide.
Well, and it's a lot of laboratory people that have just thought through the problem. And sometimes I know we have people, statisticians on our side, who love doing it because they're thinking about, there's this problem, how do we solve it? And there are people that come from the lab perspective. But we have a wide range of technologies. I mean, I've reviewed anything from a manual method where somebody had to, this is an IVD kit where you actually have to make your calibrators, and they said take graph paper, draw your calibration curve and do testing, up to obviously the automated systems. And we also have things like newborn screening where the imprecision is a lot higher, and we have a lot of assays where there is no reference material. I mean, even troponin, for example, is not standardized, harmonized, or anything like that. I mean, it's something that we cope with on a regular basis. And I think no matter what, we like the CLSI documents just because it, they tend to take the problems that are there and describe, how to think about things from, the best down to maybe the acceptable, but not as good. So I just think sometimes they are helpful because it's things that people have thought through, if you will. But I forgot where we started with all that. But anyway,
they're not helpful. They are your big brother when you're pounding your head against a wall, or big sister trying to figure something out. So I do want to argue against the fact that they're based on well-qualified calibration material. Absolutely not. No, they are a huge slice of pragmatism with a wonderful relish of statistics distilled down into the pie that we live in this wonderful world. And they are your big brother or big sister or your lost uncle or your great-great-grandmother who you still miss, or grandfather. So they are a friend, and they will help you logic check your convoluted journey on this path. So I can only recommend that you get to know and love them like you did your great-grandfather or great-grandmother, because they will be a friend of yours.
I'll call them distant cousins.
John.
Yeah, thanks. Just an observation and maybe a note of surprise, because as a service provider, I'm doing all sorts of different biomarker projects, and I'm always trying to get our clients to get the best value out of their data. So I was somewhat surprised to hear that you had not weighted statistically your data based on the analytical performance, and then even perhaps more surprised, given that you had the data, that you didn't weight it based on physiological variability either. And particularly where you're looking at multiple markers, because I have case studies over going back a long time where with a 6-marker composite, a single assay changing its CV by 10% can change what you may want to call the diagnostic sensitivity or specificity by 25 or 30%. So in terms of getting the maximum amount of value out of a data set that you have, those are exactly the sort of things I think I see not happening in clinical protocols. Most people are going either for percentage change from baseline or fold change, but they do not take into account the variability behind that data. And if we always have the analytical variability, but nobody seems to take any notice of it. But when we have the physiological variability, well, you actually got the best of both worlds. So I just wondered if there was a plan moving forward as you do this, and you perhaps widen the scope that you've put in place, that you'd see value in actually doing that.
So, go ahead, Frank.
I was going to say, so with the composite measure, again, you got to keep the composite measure from the ultimate game. The ultimate game is to look at the individual biomarkers on a single plex, all by themselves, right? So now let's talk about the limited context of use, which is interim strategy. So we'll talk about that. There, we're looking at a fold change from baseline for a subject, right? And we look at several times of urine collections, and we look at the maximum change from baseline, wherever the peak was, right? And then you look across, 20 subjects. So you look at all those fold changes, right? And to ask whether any of those subjects that were treated, what's the likelihood that there is a change from normal, all right? So, all right, I fully agree that there's a time and a place for weighting. The way I use weighting back in our toxicology space is when I have a lot of experience with toxicants showing different biomarkers changing with different intensities, different magnitudes of change, and they are better performers, like you say, for giving me the diagnosis or the prediction that I'm looking for. But I have a wealth of experience. Here, we're not doing that. We're not intentionally taking a number of different neurotoxins given to a number of different populations of people and then looking at which one's performing best, et cetera. This is all around normal variability. So we don't necessarily know. You're right. You could take the one that's the most variable and penalize it, give it less variability, right? And there was some discussion about that, but at the end of the day, we just decided to just equally weight them. So it was a long discussion, and it was swinging back and forth, going back and forth. At the end of the day, we kept it simple and said, we're just not going to do weighting. But I agree with you. There's a time and a place to do a weighting and to draw on all the experience you have with it. But it has to be a data-driven approach with a lot of experience.
Yeah, and I agree.
We're in alignment.
I agree. I think that the comment came from the fact that I've seen so many cases where you're looking at multiple biomarkers. What appear to be the best biomarkers actually come out to be the worst when you take those two factors into consideration.
Right, I agree.
Thomas Joos from the Natural Medical Sciences Institute at the University of Tübingen. I would like to add our experience during the Innovative Medicine Initiative project, Drug-Induced Organ Injury Safety. We were in discussion with the regulators to what type of assay validation procedures we wanted to do, and we got the feedback, stick to the CLSI guidelines. Then we discussed internally with close to 70 biomarker candidates, that's not economically feasible. So we wrote back and explained why we started with a low bar validation procedure. And coming back to your case study, you have been in this luxury position that you could select from a variety of kit providers, very well-established kits. Maybe in this white paper, you should also include examples how to address new biomarkers for which you have poor reagent material available. What would be the minimal requirement which could bring the biomarker qualification process to the next level. We have to stick to first to a technical type of validation prior addressing the biomarker qualification process. So I would propose that due to this big case studies, maybe adding few examples of really low bar validation procedures coming to a decision, and then if you expand your screening sample, your sample cohorts, going to we called it a high bar validation procedure.
Steve, can I address that?
So we actually have that in here. We have the limited qualification and the expanded COU. So it's really a bar from one to the other. Although we did not, as the limited context of use is not qualified yet, we were not able to use that in a progressive fashion. But it's possible that we can use that as an example of what could have happened.
So can I also address, do we actually try to address that philosophically in the analytical requirement section in terms of use what reagent materials and assay you've got to use to measure pools of normals over days or the, the two different disease states, and determine what you incorporate the variation in your assay into the variation in population because you're going to get an additive, or validate the best you can, define what your precision and bias is, and then go and have a conversation with the biostatistician and say, what size of treatment or control arms do I need to have, or how do we design a clinical study, but to statistically differentiate based on this analytic performance. So we actually try to write it two-way, one a priori, one a posteriori communication to with the biostatistician, because they're wonderful people, right? So we did try and philosophically address that conundrum.
I agree, though. I think the point being made here too, and it reminded me of something, is that there's almost two buckets of what are we using? Not performance standard. What were we just talking about? The last thing was expectations for
Performance expectations.
Performance expectations or
performance standard?
No. Anyway, it'll probably the right word. Sorry about that. This happens a lot to me. There's almost like two buckets of expectation setting. One is there's a performance standard, and I think that the document does lay that out nicely in terms of the amount of variability you get from your assay and your biological variability versus the magnitude of the effect you want to be able to see, right? But then there's other things that I think the document's soft on, which could be better, is how many times do I have to run it? And things like linearity and stuff like that, I get that. But 200 samples or 6 users over 50 days, or what is that low, like you said, what was it, low bar? A low bar for like the first entry into the regulatory eye. It's like how many times do I have to do that before, that kind of stuff. I think there should be no debate on. I'd like to see an attempt at alignment in terms of what that expectation would be for qualification.
So
right?
two things for that. CLSI EP10 will actually do that for you. So that's a preliminary evaluation of a test.
Do we all align on that? If we all align on that, then put it in here.
What I would suggest
We
more
don't align
appropriately
on it, then
is find
okay,
a
we're
good
not aligned on it.
statistician and talk to her, because the question you have hasn't got a simple answer. It depends on the variability in the test. It depends on...
No. Okay, the performance bar you have to meet depends on the performance of the assay, right? But just to benchmark the performance of the assay, that's all I'm saying.
Okay.
How many times you have to do it to determine whether you can't do the same assay in 2 different labs, or 2 people on 2 different days can't get the same answer. How many times you have to do that? Just tell us in this document. That's a
So, and I would just
We'll
want to
put that
speak
in
up
consideration.
I mean, although we recommend CLSI because we think it's a good document, I think especially the study here as well for precision was not, was a few runs, few samples. It does not have to. I think you'd want more than one day. You'd want more than one run, machine, operator. But there's no expectation or minimum requirements, and I think it's one of those things that the more that you can discuss a proposal, the better, and tell us, this is what our expectation is in terms of precision. We've run a small number. I can tell you the ballpark. And that's when generally we sort of put our heads together and talk about the context of use and what we think, whether or not we think that that's reasonable or not. So I know there's no, here they did not follow EP5. It was not close. And I think the idea is just, what does it tell us about the biomarker? What do we know about it? And what don't we know about it? And will it support the context of use that we're discussing? So I don't want people to think that we're expecting a 20-day, or anything like that. And I think the idea is obviously just talking to us about it and giving us a ballpark of what you think you're seeing in terms of performance. This is always helpful. But there's never been anything of, hey, if you don't come in with this, we won't even look at you. Because it's not that way at all.
Can I make hold on a second, because chair prerogative here. We have 3 minutes left, and I have a gentleman who's going to give us the best question of the day to go out on. And
this is a good topic. Mark Arnold from Children's. I think the discussion early on and through this last period has been really focused on helping us understand what the biological variability of the biomarker is. Some around what does the assay have to look at. Your case study, you talked about setting criteria early on and following it through. So the question I have is, in retrospect now, at what point would you have gone back and said, this assay performance is good enough or not good enough, and we need a better assay? Because for many of us in drug development, we're looking at this as an end game to qualify a biomarker, but we need to know as we move along that there should be steps. And your case example is probably one of the good places to start with retrospectively. How much data did you have and when could you have said this?
so that's a very pertinent question, because we actually have come to that point where we have arrived for one or two of the biomarkers, we need better assays. And we are firmly convinced of that. And so now the question is, well, we can't go back and retest the same samples, but we know that going forward, we have to do something different. So it was not an intentional look at the data to develop that, but it became obvious later on in the game, didn't it, that we needed better assays. And so that's what we've got to do before we go ahead and begin the next phase of this. So I don't know if that's a fully satisfactory answer, but I think that had we been a little more concerned with performance over time, keeping in mind that it took quite a long time to do that, and it's very difficult to measure RUO performance on a kid over a couple of years, we would have been able to look at this sooner and perhaps change some specs or tighten up some performance criteria to allow us to get better data.
I really like the idea, though, of doing that total error analysis, contribution on each of our biomarkers and see where we fall as it relates now to the magnitude of change that we are seeing. I think it's a really good idea to put that.
Well, and we all, often in the IVD space, especially when you're thinking about a new biomarker test for clinical use, being able to have a small snapshot of data and realize, so that you can think about the total error that you should have before you even start working on your assay. When the more that you know about that as you're developing it, and so you don't go too far because you realize, a few days in, or weeks into development that this is just not going to meet the bar. The more you know what you want to meet, rather than looking back too late and realizing that you should have set some sort of a bar. This is what I want, and these are the specifications of the assay, this is how well it should perform, and so you can look at it earlier rather than later.
I think this goes back to all of the discussions we've been having is, even though we are playing towards an end game, it's a continuous process, right? So we can never stop evaluating whether the criteria that we establish for fitness of purpose, right? Whether we are continuously meeting it and whether it was on target, whether we need to change it on the basis of, are we really addressing the question about the utility of the biomarker? You're taking that information from, are we demonstrating its clinical validation to say, is the data that is being generated from the assay supporting what we're trying to prove, right? So I agree with Mark. That is implicit, but if we have to really highlight that, I think we should be calling that out in this white paper, because that's really integral to this biomarker development. It's not once and done. Absolutely not.
So with that, I would like to thank the panel for facilitating a wonderful discussion. And we will now adjourn for 15 minutes, and 15 minutes only, not like the hour at lunch, 15 minutes only, and come back for an extended facilitated discussion of the framework. Thank you.
point a little less at the ceiling speaker right above you.
Oh. I think John's going to introduce everybody, right? Yeah. John, you're going to introduce everybody on the next session, is that right? Okay. I think what I'll have is go ahead and ask. I'll ask if the people can come up and sit down and then have John introduce the session.
Okay, sure. Yeah, that makes sense.
Okay, we're going to go ahead and get started on our next session. If I could ask the panelists for the next session to come on up and make your way to the front line of seats here. Yes, you are. Yes, you are. Yeah, so panelists for the next session, just come on up. Yeah, that's exactly what that means. Great, thank you. And for this next and final session, I'd like to introduce, as you all know, John Michael Sauer, who's going to be moderating this session and will help kick things off. John Michael? Yeah.
Okay, great. Wow, what a great set of sessions and conversation we've had. Clearly, we're making progress. I know it's hard. Hang in there. I mean, we're trying to all come together with a similar language, right? A lexicon where we can talk about this, working through a pretty difficult subject. I mean, it's really clear to me from the conversation that we've been merging a bunch of different groups, right? We have folks who are trying to develop in vitro diagnostics. We have folks that want to use biomarkers and INDs, and biomarker qualification sitting somewhere in the middle, right? And so it's hard to keep that line, right? We want to go ahead and dodge over there. I have questions about the project I'm working on and dodge back over to the other side. But I think we've done a pretty good job in beginning to define what we need to do to advance this forward. I have a couple quick slides. But before I go into those slides, again, I think we know most of these panelists. Who hasn't been up here? I think Lisa and Liz, sorry.
Lisa and Liz.
Yes, Lisa and Liz.
Yeah.
Would you like to introduce yourselves?
Yeah.
That one's on. All right. So you heard me from the audience, but I'm Lisa McShane. I'm a statistician at the National Cancer Institute. I've been there longer than I want to admit. And I've been working pretty much my entire career on statistical analysis of biomarkers.
Hi, I'm Liz Mansfield. I introduced myself earlier. I was at FDA for, I don't know, over 10 years on my last stint working in personalized medicine. And of course, biomarkers always came up and analytical measurements always came up. I'm now at GRAIL, which is working on early detection of cancer. Hope we get into this space someday.
Great, thank you. Okay, so this session is really an open discussion, just like we've been doing, but I think what we really want to do is go ahead and fill in any spaces that we haven't talked about. We can go ahead and revisit previous topics, that's fine. But let's go ahead and begin the open discussion. But what I'd like to do is I'd like to refine a couple thoughts that we've been having during the sessions, just to make sure we're all on the same page. And so one of the big things here, and it's not supposed to be a cop-out or anything, but this idea that context of use defines assay performance expectations. I think what we're saying there is it's just not what you want to use the biomarker for, which that's what the context of use says. It's not just the population that you want to use the biomarker in, but it's then also that biological embedded performance of the biomarker, right? The inter-subject variability or the variability across subjects, the response of the biomarker to whatever stimulus, right? Is it a 2% response or is it a 300% response? Clearly, that's going to define how much variability you're going to allow your assay to have. And what about where those cut points are? Right? Are again, are they really far away from where your variability is for normal subjects? Or again, do you have a pretty significant distance there where the biomarkers has to respond quite a bit? That all has to be put together. And so it was funny at the break, one of the conversations was fit for purpose got a bad reputation. It did. It did in a lot of ways, right? It seemed like, in many ways, a cop-out in my mind. I think by using the context of use, we're saying the same thing as fit for purpose in many ways. Make sure that that assay matches what the performance that we need from it. The other thing that happened during a couple conversations was around, for every assay, do we need these performance characteristics, right? Or these big 7 or 8 or 9 different characteristics, are they really required across every assay? And, you know, we've been talking about, okay, risk benefit, COU changes, so which ones drop out if it's easier? One of the answers was they don't, right? That it's really the degree and the rigor that's around these different factors that are going to be put in place for an assay potentially that requires less, or a biomarker that requires, you know, a less intense context of use, or has a higher risk benefit risk ratio. And with that, and these are things we've talked about all throughout, right? And I just put it up. And those expecting a checklist at the end of this, we're not going to get a checklist, right? I think we already figured that out. We're going to have a framework. But with that, I'll go ahead and open this up to questions or comments from the panel.
So just as a comment, and this goes back to something that Chevray said at the beginning of today, and that is a difference between where we think biomarker qualification is going to be in the future versus what we've done in the past under what we're calling the legacy process, which was pre-21st century. That I really think that we can leverage the new submission of the qualification plan, which was sort of in context of use, but not formally called out as such, that when you're trying to plan out your biomarker development effort, including your analytics pieces, go from soup to nuts, from your needs to your context of use, and kind of map it out internally, and then let's have a discussion around it. Because I think that will help. One, you're not going to spin yourselves around a lot of excess work if your context of use changes early on because we haven't reacted to it, and likewise not to do too much research investment too early until you've had a chance to talk to us. So I think it was Steve that said early, talk to us early and often. Certainly after a letter of intent with a qualification plan, this is a real opportunity to do that. Around how analytics fit into that 21st century process, even at the letter of intent stage, we'd like to have some conversation or some information around the analytics. We don't want, we have this novel biomarker and you don't have any concept of what an assay would look like. That's just not terribly helpful, I think, for either party. So as part of that feasibility assessment, just tell us what you do know about your assay. And for the other components that you maybe haven't looked at, well, you know, what is your plan? And then to emphasize what Chevray had also said, once you get from the letter of intent into your qualification plan stage, the intent is that at that level, your assay validation should be pretty far along. Maybe not complete. You maybe have 1 or 2 outliers or things that are ongoing, but we really want to know that analytical performance at the qualification plan stage. Because ultimately, if your assay is not working and you don't know that you're adequately measuring your biomarker, then it's kind of hard for us to have a conversation around its clinical utility in a drug program. We've kind of had some learnings over the years that if we try to kind of let the analytics go along in parallel with the clinical utility piece, we kind of get into these conundrums at the end. We have too many moving parts. So I think doing the analytics early is helpful.
But Chris, to be able to do that, we're going to have a lot of information around this biomarker, right? From the standpoint, I think we're going to have to understand what the reference range needs to be, say, in healthy volunteers, and then in folks where it responds to be able to go ahead and have cut points. So there's a lot of clinical data we're going to need before we even jump into qualification. So I think you're really defining in many ways kind of this idea of regulatory readiness for that biomarker, where we know it's a good biomarker, we know that it's performing well, we've done studies on it already, maybe using assays that aren't, you know, fully validated, and then we're going to come in, talk with the agency about, okay, now this is how we're going to actually do this, and here goes our validated assay. It's going to be very different from how we proceeded in the past.
True, and the scenario that you laid out especially for the case study that was just presented, there was the good fortune of already having research use only assays. We have examples where projects are brought forward where there is no assay, it's a homegrown, it's going to be developed. And so to have that conversation is helpful to have it early. Now, certainly with 21st Century, it describes the 3 submissions that would come into the agency: the letter of intent, which we've been doing already, the qualification plan, which is formalized and new, and then the full qualification package. What is not described in the legislation that we have to work on internally is, well, what happens in what we call the spaces between? Certainly in consultation and advice under our legacy program, there was an opportunity to have periodic conversations as data was being accrued. So the opportunity for the spaces between may be that we would have a very focused meeting between letter of intent and qualification plan around the analytics, just the analytics. Let's get that nailed down and get to an agreement before we go to the next step. But those are all things to be determined.
Great, thanks, Chris. Any other comments from the panel?
Maybe I can make one comment. You know, for me, you know, I'm looking at this 2-day workshop. I want to basically make sure that, at least for me, that we build on the framework on this, on the Crystal City 5 and 6, as well as the white paper that is out there. So what I encourage the audience to speak up, okay? And so let me give you my view on this, and we can have a debate back and forth on this. When I look at 99% of the clients that I support, there are over 100 of them, that are in drug development, okay? They go through the phase 1, through the phase 2, at some point the biomarker may or may not make it to the biomarker, biological biomarker validation for clinical qualification. They start and they adhere to the recommendation with regard to number of runs and some of the acceptance criteria that was mentioned in Dr. Jean Lee's paper, okay? When I look at these 7 parameters we put in, now for the biomarker qualification, right? The way I look at it, it's a continuous process. I can see, and I foresee, and I do it every day, it's a continuation of Jean Lee's paper with regard to the acceptance criteria, with regard to, again, what it was missing, in my opinion, was that it was missing, again, some of these variability, the patient variability. How do you take those variability and add it to the bioanalytical variability and eventually coming with a total error? And I think we're addressing it very nicely in this paper. So, again, what I hope that we accomplish is that I don't know if we need to reinvent the wheel. In my mindset, we already have system and process that is in a white paper and most of you guys are using. So the question is having a discussion around those, and as one of the panelists mentioned, what is missing from our current white paper is, again, those, how many runs do you want me to do? And I think we can put the minimum, or what is the industry standard, at least. And again, I go back, a lot of it is already in Jean Lee's paper. So I will stop there, for looking forward to your comments.
Yeah, if I could elaborate on that a little bit. I think we have to start working backwards a little bit and think about clinically, what do we want this biomarker to do? And as we're doing the analytical validation piece, we need to put that into the context of what we want it to do clinically. And you can sit down with a statistician and you can say, I know that if this biomarker can't achieve a precision of at least 20% or less, that this is just not going to be useful clinically. And you can plan studies, a series of studies, saying, all right, let me do the little pilot, and can I say that this is just wildly out of control, and I can just quit right here with this assay. It's going to need a major overhaul. So I think you do have to think of it incrementally, and it's all doable statistically. If you can tell me where you want to get at the end of the day, we can work backwards and say these are the benchmarks we need to meet as we go through this process. So I do think it's doable, and I think trying to come up with a magic number of, the CV has to be a certain amount is maybe not the thing we need to focus on most. What we need to focus on most is put it all in this big context, and how do we define these milestones? What are the important sources of variability to be worrying about? And think of it as this whole broad process. And one thing that always strikes me as a statistician, and I deal mostly with the academic community, but I suspect it probably happens to some degree also in industry, that when people are developing therapeutics, they bring the statistician in immediately, and they have the statistician help them decide what sample size do they need in order to show that they have a certain response rate and that is different from some control, or you think about that immediately. Whereas my experience has been that with biomarker work, it's often somebody's collected a bunch of data, they then come to the statistician, and often they come to the statistician because they've submitted a paper to a journal and it got rejected, and then they come pleading saying, can you help me fix this? And you can be a co-author if you help me fix it. So I think we do have to change the culture, and I think industry has done better at this than many in academia, and maybe it's just because of the way we're resourced in academia versus industry. But if you run into a statistician from industry, one of the first things they will often tell you is, I do preclinical work or I do clinical work. In academia, you run into a statistician, and it's often, I work in this clinical trial center. You don't very often find statisticians say, I work closely with people in the lab developing assays. So I think that's part of the thing that we have to deal with, and that we still do have a lot of collaborations between industry and academia as we're doing these kinds of projects. And so I'm just making a pitch, not because I want to have employed statisticians. We actually don't have a problem with that. We have more jobs than we have people, but I do think we need to have them involved in the process from the start. And these really are problems you can tackle. You can develop some statistics that'll tell you some no-go, no-go decisions. So I think that that's something we ought to keep in mind as we go through this paper.
Yeah, I think that the idea of is this live? Yes.
Yes.
I think the idea of being able to get the analytical validation keyed to the clinical expectations is essential for it. Understanding what the bioanalytical variability is for the test is helpful. Understanding what the biological variability of the marker is just within the individual patient is also helpful, but not prescriptive in its own sense. You might find that given a certain biological variability that you've got to find a way to reduce that by oversampling. As one example. Or you might decide that given the context of use that you want to apply this biomarker to, you just need to find a better biomarker. So there are some decisions you can have to make based upon what you've been able to learn about the analytical performance of the test and about the biological variability of the marker in the patient. But it all has to be going in an iterative way against what you can practically do and what your context of use is expected to deliver for helping the drug to be developed efficiently. Realizing that you've got, again, a whole lot of scenarios across that drug development where you can take much higher risk or much lesser risk in terms of what you're going to get out of the marker and the test.
I think what you guys are stating is absolutely correct. Okay, so let me play this scenario now. That's in the ideal world, that's exactly what we want to do, right? The reality of it here is most of the pharma will go either to a CRO or they will buy a commercial kit. When a CRO validates or analytically validate an assay, they don't validate for just one indication. They do a bioanalytical work. They go set up based on saying if the assay is good enough or not good enough, or here's basically how the assay is performing. A lot of that again is it's what was evolving from the PK world, right? Those acceptance criteria. So we're talking about some other additional. We're trying to again open up the acceptance criteria a little bit based on context of use. Now, how many of the folks in pharma actually go in and at that point they got the order from the clinical team, go find me a VEGFR2 assay that is sensitive enough to measure my clinical sample? What is your process internally? We would like to hear that. Is it that you go and talk to a clinician and a statistician and start taking the data from the validation report, bioanalytical validation report, and put it together and say, does this meet my need or not? Or do you go and take a small sample set and run it or not? So again, what I'm trying to get at, and even the example of these 6 NEF biomarkers, we had the best expert, and I'm not putting that down. I think it's a good paper and it's a good work. But look at it this way, where did we land at? That if we were going to go and design this after all these years of work, all these resources, we got to go back and now reestablish those acceptance criteria, right? So again, this is good, but I just want to make sure as we talk about, and whatever we put in this white paper, I hope it's not a philosophical discussion, but it's practical of what we can do on planet Earth. So that's what I want to just mention. Go ahead.
May I
have
add
a
a
question.
point, please? I totally agree with Dr. McShane about what you say involved the biostatistician in the very early beginning. But not only that, I often get a point too. It's difficult to be a CRO, but you still have to ask the question, have you met with your team, namely the biostatistician and the clinician? Exactly what are you trying to achieve? And then you can go to the clinical design. Because a lot of times, for example, for rare disease, you may not even have enough patient samples to provide you with the test samples. So you have to, in the very early beginning, know what is the limitation before you can say, that's the best I can do. And so you need to understand the risk, but if there is such a huge benefit, look, I'm going to save a lot of children from this rare disease. And you got the passion to do that. You have to do the best. And what you would then meet with the FDA, what can I let go, what can I not let go?
Well said. Agreed.
I mean, so that's a bit of a behavioral change, right, from what we do now, right? I hear that. And qualification gives us the opportunity to go ahead and create that change. I mean, but it's going to take a lot of work up front, right? So we're changing with these ideas how submitters are going to go into qualifications. Sorry, Russ.
So I have a question for you, Lisa. So you bear the burden of statisticians globally, based on your answer. No pressure. So we try to model two engagements, measurement to define CVI and CVG, or this is what I can confidently measure in terms of precision and bias, which is actually quite complicated to get out without reference materials. I get that. We try to model two of those scenarios. Which of those two scenarios is the best set of information to come to yourself and your colleagues with to say, okay, I can either measure with this degree of precision and bias, or this is the CVI, CVG, what quality of measurement are these numbers robust enough? So one is a priori measure, one is validation only characteristics. What do we want to change behavior come forward with in terms of preference for people like yourself and your colleagues to define the effect size and as it relates to the powering of studies? Does my question sort of get to the right place? Which one of those two scenarios would you prefer us to come to as a representative of the whole world right now?
Okay, let me make sure I understood your question. So you're referring to knowing things about like the biological variability in addition to some of the assay variability, as opposed to having a beautifully characterized assay with all of the performance characteristics of the assay?
So the way we tried to write the performance requirements section was one of some measurement, and in that measurement of CVI and CVG, we could pull out a tiny bit of the pure component of analytical variance, but in a very, very small way.
Mm-hmm.
Is that the right sort of approach to come with that information with an existing assay? Or ignoring CVI and CVG because we can't get at the patient population groups, just characterize the assay through standard and maybe some unstandard analytic measurements of performance, and then say, okay, this is how good my assay is. Can I use it to measure an effect size, or what effect size do I feel confident that I can use this assay for? And that was in that sort of fold change power function section that was written into the doc. I'm just
Yeah. Well, I mean, I think you really need a combination of both. You have to know what effect size you think you're going to have that you want to be able to detect in order to put your analytical performance in any kind of context. So, you could come to me and say, I want to be able to show that you pick up a 20% change in this biomarker. And I could say to you, okay, here are the analytical performance characteristics you're going to need to have on that assay to pick up that kind of change. But then you may come back to me a week later and say, well, actually, the change that I expect is going to be very subtle, and therefore it was a useless conversation we had last week. So I really think you need to have a handle on both. And I've seen cases where people get really hung up on, for example, getting very precise estimates of a sample taken on a particular day from a particular patient, and then only to realize later in the process that the patient's sort of natural biomarker level can be wildly fluctuating during the day or from month to month, season to season. So I really think you need to have bits and pieces of as much of the picture as possible and not get too hung up on one component.
So can I take that one more layer down in the onion then? So there's some subtlety in the way we tried to write that, and that is everything. You made a wonderful comment earlier, Afshin, and I started thinking about true north and magnetic north. We're trying to polarize everybody to an end product with some of the examples, and we're all going to take this tortuous path, realizing that true north isn't accurate. Sorry, magnetic north isn't accurate. So we've already got relative accuracy. I thought it was a great example. But one of the challenges that we had then is that's still, I know what fold effect, I know what effect size I need to be able to measure. That doesn't help us in early translation and very, very early discovery translation. And so what we're trying to get to is another layer in the onion in the document. In that section, measure and fully characterize, or one or the other. How do we help our colleagues that have not got RUO and Biotechny and R&D systems reagents or FDA-approved diagnostics that are developing multiplexed or single-plexed assays in their own laboratory because there's a dearth of reagents? What of those two flavors, or is it all of that again for early discovery and discovery translation for new biomarkers? I know I ran a monologue. Was it clear? No.
I guess I would go back to what I said earlier, that it would seem futile to try and pick up effects that are of the same magnitude of just random day-to-day fluctuations in somebody's, quote, true biomarker level. So that's why I think you do need to get that kind of information fairly early in the process. And so I would rather have someone spend time take 5 patients and measure them, over a couple of weeks and do a couple replicates of the assay at each of those samples that you've drawn. That would give me more information than you're coming forward with some arbitrarily chosen effect size you think you want to be able to pick up with tons of data on the intra-batch assay variability.
Right. So if I'm to paraphrase, you want a bit of both.
Yes.
Some patients.
I want my cake and icing too.
Characterization, even for discovery, and I think you serviced your colleagues well.
Yeah, part of that discovery aspect, though, you ask how much work is necessary up front. Another question to ask is how much work has already been done for you before you get there in terms of what was known about the biomarker in other contexts as it might relate to this or other disease processes in ways that might give you some insight about what you're going to need from it now. Because the ones that are extremely innovative, I mean, the biomarker completely out of the blue, who thought that it existed, okay? There you got some work to do, okay? Because now you're trying to figure out sometimes what it is. What is the biomarker? Let alone how it might be applied.
And you also have the chicken and egg problem is how do I actually understand the biology of the biomarker if I don't have an assay that I can count on? Right.
So. Just one thing from what I can tell you, 10 years ago when I was doing biomarker support, what I saw the pharma doing is that here's all my samples, here's a single drop of blood, do the 45-plex for me, get me all the data. We have changed now. The trend that I see, and I would say the most successful program I've seen are the ones that they say, whatever we want to call it, partially validate the assay, semi-validate the assay, whatever terminology we want to throw at it, but make sure the assay is good enough that we can rely on the number, right? We are not going to get an absolute number. And what is that? Again, you look at the small set of samples to look at precision, accuracy, upper and lower limit of quantification, parallelism, minimum required dilution, some of to establish it. Run a handful of samples, and based on that, go back and actually then refine, do a full biological assay validation, and then you also have had some real sample. Yeah. Right? But how many pharma actually have the time to go stepwise? Most of it is that the samples are in a freezer, get the assay going, right? So in terms of cultural change, that's one thing. So I see where you're going, but again, the culture change, it's got to be at so many different levels there. The second thing we're talking about, what if you have to develop the assay yourself? To me, actually, if you develop your assay yourself, Good job. You know what? You're going to have control over at least your recombinant protein that goes in there. You know which vendor you bought it. You know which antibody you have. You know you're not, if you have so much more control on it. So it's actually, those assays in general, long term, they work better than when you just buy a kit that it's a black box. You don't know what is in it exactly sometimes.
One question which is kind of related to this. I think with the assay validation, we set up probabilities, total error, and so on, but no validation is worth it without the clinical study at the end, right? And so during such a clinical study, maybe going on for 3 years, you observe suddenly weird results, and then you think about it, and then you may have an idea of what could be the problem. And then you think about, okay, I have 3 samples in the fridge. I could just look at it. I didn't think about statistics at that time, but that's all I have, so I look into this. Now, my question is more what I understand, or my question actually to the FDA would be, okay, although this might be not, statistically significant, but would you still like to see those results that are based on available samples at that time? Or is it not worth it to provide this information? Because for us, that's always a problem. At the end, you think, well, it was not 100% properly done, so you may not want to provide these data. Or so my question also with regard to the white paper would be, could it be like phrased like, okay, and if you have additional information, please provide, or just leave it out or something?
I like the scenario you're bringing up. If I could just say one thing. I think what this is showing is that assay isn't ready for qualification, right? And that biomarker probably isn't ready for qualification. I think that what the expectation has to be is that it has to be at some state where you have enough information around the biomarker, around the assay, to then be able to go forward with qualification. I didn't answer your question, I know, but I think it illustrates what we're talking about, the difference between using that biomarker in an IND versus using it for qualification or qualifying that biomarker. I'm sorry.
I certainly can't speak for the CDER folks with respect to what they'd want to see in terms of particular kinds of information brought forward. But if I understood the scenario you described, you thought you knew something about the marker and the test. You had it ongoing with respect to collecting clinically relevant data, and then you saw something you didn't expect.
Yeah, like very huge, unexpected data. Somehow BLQ data, which you would not expect from what you've seen before.
So you're always looking for a learning opportunity.
Yeah, exactly.
And the question is,
so random errors.
Do you find that it was a bullet, or do you find that it was something that was an outlier, that was maybe there was something about the way in which those patients were selected?
Sample collection, whatever.
Some problem popped up that then informs your opportunities to revise your approach to the marker or the test. As to how that would factor into the ongoing status of if this was already a qualified marker or if it was one that was bucking for qualification, I can't say. I mean, it depends upon how serious the issues are. But you never want to throw away the opportunity to learn something more about what your marker and what your test was trying to tell you.
And it depends on the context of use that the biomarker is in that program. It sounds like the scenario you're laying out is more kind of in the IND space, perhaps, whether it's qualified or something that's being drug-specific that's being developed. But depending on what the peak or that unexpected result of the biomarker was, what is the consequence? What was the trial design? Is it more for your measuring safety? Where there, as Lisa keeps pointing out, there's ways that the trials can be designed that you can mitigate risks based on some understanding in advance. But I would say there's always opportunity to learn from that, and I wouldn't keep it to yourself per se. I mean, there's this perception of don't share things with the FDA because things will go bad.
Exactly.
And I think we're really trying to get away from that, especially around biomarker discovery and scientific understanding. So to the extent that it was a surprise to you, it would likely be a surprise to us, and let's have a conversation about it.
Yeah, I'd like to follow up on that. And I think it's the unexpected data, the failures that often are the most educational. And so it is important that you share that information. And what I have seen many times is that you get some unexpected result, and you're hanging out in a meeting like this and standing in the hall talking with Joe or Jane, lab person at another institution, and you say, I got this really strange result, and they say, oh yeah, I actually got that too, and I didn't quite know what it meant, but we did this and that, and we think it's this. And so there's probably a lot of weird data hidden in closets. And so I think we really need to be transparent and get that data out there. And it's okay if you can't explain it right away. And it can be really wild things that happen. I remember very early on when I was a grad student, we had this really wild measurement show up in some HPLC vitamin E results. And we thought, what went wrong here? Well, it turned out the school I went to had a veterinary school, and someone decided to run a horse sample in with some human samples. So had I not known that, had I not questioned that big value, my subject-to-subject variance would have been way off because I had a horse in there. But there, sometimes you do find these explanations, and it often takes more than one person's fluky data to figure out what might be driving some of these things.
But would then the clinical report be the right place to put these information in, or what would be the best way to?
Well, okay, when you say clinical report, you're not talking about for a patient, or are you? You're talking about a qualification report or something.
No. Then for the study report, if I don't want to show that my biomarker is not ready for qualification, which I don't want to, then the question is where I put this information to share it. So would then be the clinical study report the right position to put this information in, or what would be then the best way? Because the qualification document would not be the right way, obviously.
Right, unless there's a concern for your drug safety board about your design of your study and the safety to the patients, I think it would be okay to wait and figure out if you get more data around that signal with more patients, and then to put it in as part of your report analysis. I think that would be, well, I know Lisa's no longer here, but I think that would be the more appropriate way than to, as your study is still enrolling and you happen to be starting to run samples and you see a spurious result, just like you don't have the context of where that fits into the broad results of your study, we wouldn't either. So the more you could analyze it and then present what you think it means, that helps.
I was just going to make a comment. We've decided that we need a biostatistician in the boat. We need a good bioanalytical person. We need a good clinician. The people that I think we haven't put in the boat yet is good biologists, people that understand the systems biology behind these things. I think that's where you get the other side of this. You can define your analytics to meet criteria, but to understand the biology. And I'm no offense to any clinicians in the room. A lot of clinicians don't know that. They know they know how to treat patients. They know all that, but they don't know the system biology. And that's the other person you need to bring into the discussion. And then you can really define these parameters.
Well, and when it comes to the biology piece, I mean, those of us that spent years getting those degrees, ultimately, what is the outcome of our efforts? It's some sort of a pictorial diagram with arrows connecting a bunch of figures together. What's the likelihood that those figures stand the test of time? They never do, or very few of them do, and that's okay. Biology is messy. And so as you kind of piece apart that scientific understanding, just know that it's a snapshot in time of this is what we understand at this point in time, but that's okay. But then we get challenged in drug development biology where time passes, scientific understanding passes. So to the extent that you can think in advance by storing samples appropriately, et cetera, that will be to your best advantage.
And I'm not really talking about someone who just understands the chart. I'm talking about someone who's been working in the therapeutic area as an expert. They know how that chart is changing, and that's who also need in that boat.
Jeff, I would add, you also need a VP to sign off on the cost and timeline, and a procurement that is outsourcing
I'm
looking
learning
for quality.
about that part.
So anyway.
Okay, well, just remember that the two examples that we're going to present during this workshop, in both cases, the person leading the project is a toxicologist. They're safety biomarkers, right? So I think that fits that biology example.
Yeah, I think one thing that was difficult in the paper is putting together really an operational framework. I think there was a really good understanding of some new concepts. The idea of, we've talked about learn and confirm for a long time, but integrating, statisticians into the process. But I think knowing when those points to where you can start to build performance, the performance standard, et cetera, what is that delta that is needed to see that? If we could come up with not necessarily a guideline, but a representative example of how you would, take a biomarker through this process, because I think we have been caught up in more of the traditional biomarker validation. But if we want to really move this into qualifying a biomarker, we need to move to some of these concepts you've put forward. But how do we make that operationally feasible? I think that's where AFSHN's driving us. What do we need to do to make that happen? But maybe if we could just give a process map, a representative, that might give people a better understanding of what's needed and how we might make this a reality.
That's a great idea. Comments?
Chad, process map is exactly what we need, because but I worry about example, because when you give an example, people basically gonna stick to, that's the only way I gotta do it. Right. That's my only worries, but it helps to explain, right? So it's difficult. What do we do?
Well, I think you call it a representative example, because I'm thinking back to when companion diagnostics were rolled out. Nobody could figure out how to really put it into practice. It took time. It took meetings like this where people, looked at different scenarios and how you do actually operationalize it. And now it seems to be a pretty not straightforward, but an effective path. So I don't know. I think this is an opportunity for us to put something like that on paper. Again, representative, not the only path forward, but a way to go about it.
Yeah, I mean, you're talking about the difference between philosophy or principles and ordered steps.
Yeah.
And the case of ordered steps is a path, which you have to be somehow flexible. I mean, a lot of times the path loops back on itself. That's a really challenging thing to do in ways that will be comprehensive.
Yeah.
So there's some blend there between the ordered steps and the way in which you have the insights to change the order or to emphasize one thing more than another.
Yeah, and I don't think it could ever be all-encompassing, but it would give somebody a starting point. Because, for example, do you need a phase 0 study to understand the variance components, but also if you were in a disease population to get that delta? Without those parameters, we really can't assess what that performance standard is. So I think that's where everybody's caught up right now. We're going back to the default mechanism of we'll just go with a raw analytical approach to doing this, knowing all of we've outlined all the issues around relative accuracy, et cetera. So I just think we could probably brainstorm and come up with a representative map.
I think you're going to see in the next example around GLDH a creative way of getting samples. And I think with qualification, we've been thinking about these large prospective studies to be able to show exactly what we want to show. Maybe we can be a little bit more creative. And so keep that in mind when you see the GLDH example.
Yeah, John Allinson, LGC. So just maybe a quick observation on that comment. I think there's a lot to be learned just from diagnostics development and how they've developed over the years. And for my sins back then, I can remember a lot of that. And, we did start with the approach of doing the analytical validation, set a reference interval so we can see if people are falling within that, and then start looking at biological variability. And that often meant that in a cyclical approach, you're actually going back to refine the method to make it better. You have to remember that once you've done that, your physiological variability data is going to look different if you reassess it. So it is a very much cyclical in development approach, and things like introducing reference materials that are well characterized and understood are all part of that. So I think that information is all out there. It's how you want to pull it together. But the point I wanted to go back to in answer to your introduction about things, do we need all of those parameters validating? I know we're getting out of time this morning, and just one observation I wanted to make on the draft document, which, by the way, I think was a very nice document, and it's a massive improvement in taking us forward in this area. But is that of how we actually look at data, and particularly all of the examples, Afshin, that you give for parallelism. And I know there's lots and lots of talk about it. Lauren's put a really great paper together as well. But I just have concerns when we look at the data. I think one of the recommendations was looking at CVs around the mean of a data set. And the thing is, I've seen so many examples of people that do that actually then include data that they shouldn't include because it's clearly demonstrating matrix interference effects. So I always grew up and learned about parallelism as being recovery in dilution first and foremost. And if you actually do plot it, and you did give it as an example of percentage recovery for your dilutional adjusted values, I think it's also getting people into the sort of routine and of not just expecting that the neat result's going to be the right one. So actually recalculating that recovery against different ones as we go up and see data coming into consensus. And that chart that you saw, I mean, that would clearly fail your CV criteria. But I have seen some CV criteria that has passed when if you actually look at the data really in depth, then you'll be taking some of that data set out because it's clearly from results that show matrix interference effects. So in terms of acceptance, I think the easy way of doing that is actually set a confidence limit approach around that percentage recovery based on your analytical performance. So look for statistically significant differences from what you expect from the variance within the assay, whether that's a 95% or a 99.4%. You know, this is where your context of use can actually describe and help you tailor that to meet what your context of use is.
John, one thing is that maybe, my apology if I did not present that properly, but in the white paper, I believe we have actually, again, talked about the context of use with the parallelism, again, with a 95% confidence interval. So it's in there. If it's not clear, again, we probably clarify it some more, but we have included that. Good point. You know, the one thing is that if you look at 2-3 years ago with EBF at least, I know there was a paper out there were discussions, you know, that they put parallelism as pass or fail. I really worry about putting something like that as pass or fail. The way I look at it here is that going back to, you know, when you're doing these validations, I'm talking about phase 1, phase 2 early on before we get to the biomarker qualification. You want to learn about the good, bad, and ugly about your assay. And those ugly stuff is actually very helpful. So document those in your report, whatever you want to call it. And so these acceptance of pass or failing for parallelism to me is all relative. What do you want to use it for? Set up your acceptance criteria. John, exactly what you said is correct. It's another way to do it. You know, the way I look at this white paper is that we've got to come up with different system methods. Some going to be more sophisticated and more sophisticated companies that have access to clinician and statistician and everybody else, great, use it. But we also got to introduce, in my opinion, some basic method in there for you know, maybe early stage assessment. I'll leave it as that.
Hi, Dan Kranek, FDA and CDRH, mostly imaging actually. But I heard a comment this morning about iteration on the assay design. And I heard another comment, I think here, and there was the introductory remark before the question about parallelism touched on the same thing. But my question would be, when do you need to do it? And if you refine your assay, do you need to redo all 7 elements again? Can you borrow partial from the previous design? How do you do that? Because what I've heard throughout the day is, you know, not only do you have legacy samples, but you might have legacy data that you're trying to borrow for reproducibility or physiological variability. I just saw a lot of heads nod that said, hey, when you change the design of the assay, you should go collect all the data again, which I agree that's probably a great best practice. But I guess in our role, a lot of times at FDA, we're faced with interpreting data that exists. So you can say, I'd really like to see this, but I have to interpret what you have. So I was just wondering if you could comment a little bit on either in the paper here, like how to handle that, like as the assay is changing, or is there an expectation that the assay is totally final finished, you don't change it at all, which I think is probably unrealistic. But how does that impact kind of this? So, thanks.
Maybe I can address that since I was dealing with that quickly. So to me, at least, and again, you guys agree or disagree, if you change a critical component of your assay, your antibodies, for example, detection or capture, or your recombinant protein that you use as a calibrator, in my opinion, it's a no-brainer. You got to go ahead and revalidate because that is changing. Now, here comes the I guess the mystery a little bit. I've had assays that the carrier protein that I use for blocking, a blocking buffer, it doesn't matter if I went with this blocking buffer or another blocking buffer. This guy went out of business, I'm going with somebody else. I do a quick test, everything is still recovering. The assays seem to be, assay based on few runs, performing the same, based on upper lower limit of quantification and so on. I've also had assays that I changed the blocking buffer, and that blocking buffer was actually causing or minimizing the interference about, between some of the biological molecules that were interacting with the biomarker. And that changed the assay. So there is some, I guess, a easy pathway with the critical reagent. If you change it, you gotta revalidate. But there are other areas when you do change something that is not a critical reagent, however, it makes a major enough change in your assay performance. In my opinion, you have to revalidate, but I'm always on a cautious side.
You're always having to assess the risks associated with what you know and what you don't know. You find that this happens with tests that are developed in the commercial set as well as in the research set. You're having to figure out when you made a tweak, is it something which you think is modest or is it something which is key? And if it's key, then you've got a lot more work to do.
And the biggest challenge, again, I go back, that's why I love these companies that actually make their own critical reagent. With a homebrew, with these commercial kits, I have seen so many crazy things that basically sometimes the capture antibody concentration was changed over time. And all of a sudden your curve, it's not a matter of just shifting curve left and right. You start losing your upper or lower limit of quantification. So that's why, again, even when you get these, doesn't matter what stage you're 1, 2, 3, or so on, in my opinion, you've got to have an internal practice to check the reagent that you get before you do anything with it.
And after you've placed your bet, which is to say you chose not to revalidate on a certain feature, then you just have to be essentially keeping careful track of how that assay is performing going forward. I mean, you can't just fire and forget and say, I think that it was okay. And you're now looking for signals, as you should be doing all across the time of use of the assay, to say, are there things showing up where you ended up with, where was the lady? Okay, found the aberrant results. Didn't see them before. Now you're seeing aberrant results. Get on those quick.
The easy part of that answer, if you change a critical reagent, you have to do some serious work. But the more difficult part of that is if you change something. I have had personal experience where a 510K cleared, actually PMA approved, prostate cancer test, had the blocking buffer changed, and all of a sudden started spuriously, false positively diagnosing patients as having prostate cancer based on a change in the formulation of the blocking buffer from the manufacturer which supplied it to the test manufacturer. So that is a lesson in no matter what you change, you have to be fairly rigorous day to day about what is going on in the exam. There are excellent stories about how assays have drifted over time, leading to inappropriate diagnoses where all the intermediate quality control tests have passed, and they still are winding up wrong. So it really is, no matter what you do, and even if you don't change anything over a period of time, you still have to look and check, is this behaving as I think it should? And that's not as simple as running quality control charts every day and looking at them and saying that's good. So I think the hard thing is if there's a critical dependency on that test, if there is a risk associated with it, that no matter what you do, and if you do nothing, you still have to do a little bit work to make sure it's behaving properly.
Right, and an extension of that also is one approach is to have a set of proficiency samples, what we refer to as samples in which you have previously characterized the level of the reagent, or level of the biomarker. So post change of any reagent in the assay, it's good practice to go back and check if you're reading the same sorts of results. So you have to a priori define what is your tolerance, in terms of recovery. So what we refer to as concordance. So sometimes if it's qualitative, you can say you expect greater than 95% concordance in your yes or no results, or it could be quantitative to say I have to recover within 10% if that's the precision in your assay.
So I think those are excellent comments, but I just want to reframe that in the context of submitting the biomarker qualification package and when the analytical validation expectation is in terms of acquiring that data in comparison to the clinical validation data, which you're suggesting or perhaps are, also including what are the quality assurances to make sure that over the course of the clinical validation, the assay doesn't change. So in addition to the analytical performance, what assurances do you have to make sure that as you go forward and collect the other data or compare between studies or otherwise, how do you do that? So that's why I wanted to ask the question. So, thanks.
Thank you. I think we have some questions from online.
Just a few questions here. Is that live? Okay. First, this is for Afshin. You mentioned stability testing, which is an integral part of any validation. There's currently quite a bit of debate across industry about using nominal versus observed concentration as t equals 0, and therefore the target concentration for later time points. Since biomarker reference standards are often recombinant, this makes the topic even less clean-cut. What are your thoughts?
So I give my personal opinion and at least my practice where I see out there as well. Look, we're talking about relevant accuracy. Okay, so to me, and we're using a recombinant protein as a calibrator. So to me, again, and then we're talking about stability that we're not collecting really T0, T1. A lot of these are relative, right? They're not a true absolute measurements of what is in there. And so we're going back to the stability now, with a lot of these factors that we have very little control of compared to ideal what we like to do or we can do in the PK world. To me, the first measurements, you give that 100%. If it was supposed to be, at 100 picogram per ml and you're reading it at 85 picogram per ml, to me, again, that first reading that you do is assign 100% and then follow from there. Now, I've seen the industry in both ways. There are individuals, there are companies that they're adamant that no, it was supposed to be 100, you read it at 80%, and then the next one you follow the actual concentration, relative accuracy. And there are the ones that are doing percentages. To me, it really, again, we're looking at trend of stability trend over time. And to me, again, going with a percentage should be adequate. But again, we have regulatory folks here and other companies. So please speak up if you think otherwise. We're in agreement.
So again, the document addresses that in two ways. One is the serial time point from first freeze, and then also the true recommendation to bridge from fresh to first freeze with perturbation. So we understand table 3, which had all of those pre-analytical variables. Start off with what you can do, but don't forget the gap and plug the gap. And there are many, many commercial companies that will procure and generate freshly drawn samples for you all over the world without wanting to advocate many. There are at least 3 or 4 in the United States that we used all the time. Or engage with a clinician at a hospital. Seriously. So we do recommend start with what you can do, but bridge back to time 0 and perturb. I just wanted to point again, clarifying it. And if I've come across as defending, I'm just trying to clarify the points in our intent. We are all welcome of all feedback, by the way.
No, I think that's really good. And so you guys, again, it all comes back to, if you look at it, is the level of risk you're taking, right? What is important, what is not important. And again, there is an ideal way of doing it, and then there is a more, easier way to do it. At some point, you got to fill in the gap. I think that's what we're trying to. You see the different views, even with the meetings that we have had for a year now. They've been different views, right, of how you go about doing it. So, good point.
So for long-term stability, how do we create acceptance criteria, right? How do we weave that in?
Yep. Now, with regard to the acceptance criteria, I think I think I gave an example, right? You got a biomarker, in my opinion, that a 10% change. If you're looking for a 10% change in between pre versus post dose, and your biomarker stability after 3 months is 20% of the, it's degraded. Okay, that's one. Another way, if you're looking for a 3-fold change, and after 6 months you got a 40% degradation, do I want to be, punished for why did my biomarker go down to 40%? So I think it really comes back to context of use. What do you want to use the data for? And again, I really hope, if nothing else from this meeting, we understand that, the PK world was a good place to start, but we really got to move toward this context of use and give ourselves a little break with some of these acceptance criteria. Sometimes we put, do it statistically, biologically, based on, all the stuff that we talked about.
Can I just quickly put 10 penneth in there for that? I think what we're looking for when we prove stability is data that is changing in a statistically significant way outside of the analytical variability of the method. If it's within the analytical variability method, within certain confidence limits, you can't prove it's unstable. The question is the precision of the method good enough for the context of use that you have from a clinical perspective?
So, can I steer the conversation?
Are you advocating Westgard rules? Okay, so that's.
In terms of stability, I think it's looking for statistically significant changes in the data results you're getting back that are outside the analytical variability, or you can't actually claim with whatever degree of statistical significance you're looking at that it is an unstable result that you've got. If it's within analytical variability, how do you claim it's unstable? If it's outside the analytical variability, which is what I often see using the bioanalytical guidance, you can often have results that are outside the analytical variability that we still call stable. And that's the bit that I don't get.
So I have a procedural question because we had...
Did we put Westgard rules in? We did, didn't we?
We actually have them referenced, yes. But we did not talk about Westgard rules in detail. Sorry.
Okay, you can't interrupt me another time. So since we're talking about analytical validation for biomarker qualification, right? And within the drug development paradigm, it's very unusual for a biomarker assay validation per se to be brought up to the agency when it's not in the context of a general biomarker qualification. So when the biomarker is embedded within a drug development program, essentially the drug development company is making decisions on its performance, and then they try to evaluate it as part of the phase 1, 2, 3 studies. So do you see a change in that paradigm happening with this initiative? So is this like a new beginning where there is going to be different touch points for PK and immunogenicity assays, which typically goes to the CMC section, right, only at the time of a BLA or an NDA review? For biomarkers, do you see this, touch points with the agency changing? And how frequently do we come to you? And do you really review the analytical validation?
Right. So to John Michael's final bullet here of the interplay of these various spaces, certainly I think each of these spheres have lessons that can inform the others. And I guess that's where I've been kind of trying to be more the devil's advocate of not having such a high bar in this space for all contexts of use. And even though we're not talking about biomarker development in the IND drug specific space, I wouldn't want this to go into that space to raise that bar, especially in early phase development. As you rightfully point out, in many respects, it's more of a financial decision on the part of the drug developer and how they allocate their resources over their various programs, of to what extent they feel that the biomarker has value, that they're willing to roll the dice. And then make a decision internal to the company about what happens to the future of that program. That's not us.
And I would
Liz,
also add,
quick comment.
my time in CDRH, it was not that unusual for CDER to ask us, this company did this assay, is it valid? Is this a valid assay? Which would just be a consult to CDRH. So I don't think that needs to change either. I think the maximum flexibility is that manufacturers actually can come up with their own biomarkers and so on, and that they would be validated internally and analyzed by CDRH. Yeah.
And in large part, it all comes out of the context of use. So in an IND program, where you're doing a proof of concept phase 1, 2, the biomarker is being interplayed, it's adding additional value to what would be normal, traditional clinical care, and the risk to the patients in that trial is no different than if there was no biomarker, then in all likelihood, we're not doing the full court press. And so I think an analogous context of use in BQ, I'm not saying that it should be the same, but there shouldn't be a huge disparity.
Right, I understand. It's just the procedural things that, we were talking about in terms of where do you begin, how do you approach the design, and how do you power the studies? Because mostly in drug development, the powering is based on your clinical endpoints, unless you plan to use the biomarker for primary decision making. So.
Okay, I have to jump in as the moderator. We're 4 minutes over. Sorry. We're going to do more of this tomorrow, so keep those questions. I know we have more questions from online. We'll go ahead and feed those into various discussions, but let's thank the panel, and we'll close for today. Sorry, I had to take that.
Thanks, John Michael, and thanks to all of you. I'm going to go ahead and wrap things up for today. And boy, are you guys an engaged group. And you can get into the weeds, which is good, right, Martha? That's what we needed for today. So we heard a lot of great things. The conversation was really helpful, and it was interesting to hear all of the really challenging and remaining questions that arise once you start thinking about actual case examples and how this framework might apply. So we're going to, as John Michael just mentioned, we're going to continue this kind of discussion tomorrow morning with a different case example that'll focus on GLDH as a liver toxicity biomarker, and then we're going to have a series of discussions that'll get a bit broader and talk about what's next in terms of the framework, how to refine it, and also to disseminate this framework more broadly. So I hope you can join us all tomorrow at the same time and same location. With that, thanks again to all of you here and all of you online. I've heard we've had around 200 people joining us online, so this is a really important topic, and we do appreciate all of your engagement. So we'll see you bright and early tomorrow. Thank you.